Welcome back. Kindly please take your seats. We are starting with session 1. It will be chaired by Professor Rana Pratap Singh, School of Life Sciences, JNU, and co-chaired by Professor Bindu Mangla, Department of Chemistry, JC Bose University, Faridabad. Now I would invite Professor Rama Jayasundar, Professor and Head, Department of NMR and MRI, AIMS, Delhi, for the presentation.
So, good morning. So, let me give a very brief background of my academics. This is just to put my, help you all see my lecture in context. So, I'm basically a physicist. I have a PhD in physics. But I also have a professional medical degree in Ayurveda. So I have done my BA MS as well. This is after my PhD in physics. So at the outset, I congratulate JNU and ICGEB for organizing this very important workshop. And I thank them for inviting me here, and also for giving me the freedom to choose the aspect of Ayurveda I would like to touch upon. I hope what I am going to share with you all serves as a food for thought for the deliberations that will take place later on. So we are all living in the midst of data revolution. On one hand, we have the multi-omics data, and on the other hand, we have increasing use of artificial intelligence, machine learning, and deep learning. Some of us are actually working in these areas as well. So information is flowing in thick and fast, changing the landscape of healthcare. There are very many areas which have become niche areas in healthcare. And in the context of medicine, I have just highlighted some of them. Systems approach to health and disease care is being talked about extensively. And mental health, as you all know, is assuming proportions of epidemic throughout the world, including our country. So if we want to handle mental health, there are two prerequisites. One is that we need to have an understanding of mind, and mind has to be factored into the framework that is available right now for modern medicine. And we need to increase the scope of the metrics that can help a multi-pronged approach to mental health. The other topics are personalized medicine, sustainable medicine. All these are niche areas in modern healthcare. Now, where and how should Ayurveda be positioned in such a scenario is the question. On one hand, we have the biological sciences and medical sciences being heavily backed by science and technology and very rigorous scientific research. On the other hand, we have Ayurveda, which is thousands of years old. Yes, there is a lot of research, interest in Ayurveda. This is supported by the fact that Institutes like JNU and ICGEB, which are at the forefront of biological sciences, research in biological sciences, they are hosting this such a workshop. So there is a lot of interest, not only interest, there's a lot of research work as well being carried out. Nevertheless, the core principles of Ayurveda have been developed thousands of years ago. That has not changed. That will not change as well. What we are doing is trying to do research to understand the concepts better or for validation, right? So what I am going to address today is the core principles of Ayurveda, whether it has any whether it makes any sense in the contemporary science. Comforting thought is that there is a common point of interest, which is health and disease. Modern medicine also is focusing on health and disease, and Ayurveda also does. And Ayurveda simply means science of life, and because it is science of life, you see inclusion of both health and disease here. When it talks about health, Ayurveda talks about preventive and promotive health. Do these words ring any bell? Yes. Modern medicine is also talking of preventive and promotive health. Let us remember that modern medicine started off as a disease care system. Only in the last few decades, it is realizing the importance of health. It is realizing the importance of preventive and promotive health. So Ayurveda talks about preventive and promotive health, and of course, when it talks about disease, it does talk about how to treat the diseases. So far, so good. Let us see how Ayurveda understands health. Is there a systematic way in which it defines health? It says health is a complete balance of functioning of tissues, their metabolic end products, senses, mind and consciousness, and social and ecological well-being. Absence of disease alone is not health. WHO, in the initial stages, it's defined health as absence of disease. But now it says that health is a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity. You can see the convergence of thought, the way health is understood and defined, and if we are objective, we realize that Ayurveda is one step ahead in its comprehensive way of understanding health. So Ayurveda, let us look at the way Ayurveda has understood health, not in depth, just to give a glimpse of how it looks at health as in different layers. So it starts off with self. There is a physical health, physiological health, psychological health, emotional health, social health, environmental and ecological health. So there are layers of health that Ayurveda talks about, and it not only talks about the layers of health, it also gives solutions to how to handle each of these different layers. There are also metrics to handle it. So Ayurveda has a multidimensional understanding of health in health. So you can see that physical, physiological, psychological, and all these health, and also it talks about sensorial health, both in physical, gross, and nuanced terms. Food and lifestyle, we know that modern medicine has included many, many diseases in the ambit of diet and lifestyle disorders. And modern medicine is also talking about the importance of sleep. The quality of sleep is very, very important. So all these various dimensions are what Ayurveda looks at when it talks not only about health but also diseases. So there are a number of parameters that Ayurveda uses as health metrics. VPK stands for Vata, Pitta, Kapha. Slightly later on, I will give you all an idea as to what this is. Phenotypes, this is called Prakriti. So modern medicine talks predominantly of genotypes. Of course, it's also beginning to talk of phenotypes. Ayurveda doesn't talk of genotype, but it uses this concept of Prakriti or phenotyping to personalize health as well as disease treatment. And you look at these other parameters, seasons, circadian rhythm, activities, age, gender, food, all these give a bunch of basket of metrics to assess health and disease in Ayurveda. And health is a multimodal delivery. So there are many number of ways in which health is delivered to people. So there are general rules that everybody should follow to stay healthy. There are regional rules in sync with the climatic condition of the region. There are family traditions. There are festivals through which, we know that different cuisine are associated with different festivals. And these cuisines are in sync with the weather during those festivals. And age-dependent regimens, Prakriti-dependent regimens, and so on. So far, so good. Yes, okay, we all agree that Ayurveda has a slightly in-depth understanding of health, but we know that Ayurveda uses terminologies which sound very archaic, and their management strategies are very different to what we are all used to. Of course, just because something has different terminologies, it does not make it unscientific. What would make it unscientific is if we are not able to explain what these terminologies are. So that's what I'm going to try and do. So let us start from the very scratch. The endpoint of any clinical medicine is management or treatment. And this is defined by the metrics used for diagnosis, and this in turn is defined by how the human system is understood. If the human system is understood in terms of proteins and cells and molecules and genes, the diagnostic metrics will speak the same language, right? And the treatment will have to adhere to the metrics. Right? Now, something defines how a human system is going to be understood. Human system is such a complex system. It would be very naive to believe that there is only one way of looking at the human system, right? There should be more than one way of looking at the complex system that the human system is. What is going to decide how the human system is understood is the worldview adopted. So what is a worldview? It's how one looks and understands the physical and non-physical realities around us. And what determines what the worldview is going to be? It is the basic science which provides the worldview. So let us look at, so if we have systems which are different from each other, the culprit is the worldview. That decides what terminologies are going to be used, that decides what is going to be prioritized, that decides how the strategy is going to be evolved. So let us look at the worldviews. The modern medicine, or let's say contemporary medicine, not only medicine, all the subjects, all the disciplines, including the biology, is based on the worldview of classical physics, which considers reality as being made of discrete material entities which can be reduced to fundamental building block. This is the worldview of classical or Newtonian physics, and this is the worldview adopted by the biological sciences. And the worldview of Ayurveda is the Indic worldview, which says that reality is interconnected and cannot be separated or reduced. So you see how different worldviews have led to different understanding of the same biological system. So what you see on the left side is something we are all very familiar with. Anything physical in the world, including the human system, biological system, is made of atom. Atoms make molecules, molecules make organelles. You have cells, tissue, organ system, and the entire organism. Here the large object, that is the biological system, is reduced to smaller components. And based on the worldview Ayurveda has adopted, it has conceptualized the human system as a seamless, interconnected whole. Let us look at it in slightly more detail. So Ayurveda talks of 4 different domains. One is a structural domain. The entire structural domain is a network of channels. These are called Shrotas. You may remember that Dr. Upadhyaya, in the first session, he did mention this word Shrotas. Shrotas means channels. This refers to gross and subtle channels. Subtle channels or ion channels, the channels in the cell membrane, all these come under these Shrotas. So the physical aspect of human system is a network of channel. In fact, Ayurveda, one of the definition for human body in Ayurveda is Srotomaya, which means that a body is nothing but a network of channels. Ayurveda talks about a physiological domain which is defined by parameters called Vata, Pitta and Kapha. I will tell you all what it is. Not now, later slides. Here you see that the structural and the physiological domain is interconnected with these parameters VPK. Then there is a psychological domain which is defined by parameters which you all may have heard in a different context. This is called Sattva, Rajas and Tamas. And again, you see the psychological and the physiological domain connected by these parameters VPK. Then you have the subtlest aspect of human existence, which is consciousness. This deals with levels of awareness. You can see Sattva, Rajas and Tamas acting as a connect between the psychological and the domain of consciousness. But I think what is very interesting is that the grossest aspect of human existence, which is that physical body, is connected to the subtlest aspect of human existence, that is consciousness, through the first level of awareness, which is the physical body. So you see how Ayurveda has conceptualized the human system as an intra and interconnected entity. This is in stark contrast to the structural hierarchical understanding of the human system in modern biology and medicine. I'm not telling one is superior or one is inferior, not at all. These are 2 different ways of understanding the human system. We should be very happy because both has its own advantages and disadvantages. There is a wealth of information from both these viewpoints, but I want you all to understand that these different ways of looking at the human system is because of the different worldview. So much about how a human system is conceptualized. How is it translated? We now talk of translation medicine, right? How did Ayurveda translate these to a clinical practice? So for this, Ayurveda has a number of classifications. The first level classification is gross and subtle body. And the gross body is subclassified into 3 types. One is based on structures, which deals with the physical aspect. The other is a functional classification, which deals with physiological and psychological aspect of human existence. Then you have the system-wise classification. Please, this is not the system-wise classification we are familiar with in modern medicine, like reproductive system, digestive system. This is slightly different. Nevertheless, this is system-wise classification. This deals with physical, physiological, and psychological aspect of humans. And then the subtle aspect deals with levels of awareness and psychological and realms of awareness. Now, I am going to give you all a peep into the functional classification because this runs as an undercurrent to the entire understanding and management of health and disease in Ayurveda. When you say functions, immediately you all realize that it is an emergent property, right? There is no single parameter or metric you can say is solely responsible for functions. Structures play an important role. For a function to manifest, structure has to be there. Biochemistry is important. Activities ranging from electrical, magnetic, mental, emotional, and if you are all comfortable with it, even spiritual activities, all these contribute to a function. It is interesting to see that structure and biochemistry, which plays a very important role in modern biology and medicine, is factored into the functions. So Ayurveda, for its functional classification, has identified 3 functions, which it thinks is very important: movement, metabolism, and transformation and growth. In the context of Ayurveda, these are called Vata, Pitta, and Kapha. I am going to refer to these as V, P, and K just for convenience. So VPK refers to the 3 functions, but what makes it a practical tool is these parameters it has under them. So VPK is functions, refers to functions, and a group of parameters which are biophysical, physiological, and psychological in nature, which impact these functions. So these is just an example of some of the parameters. You can see, of course, pH. Ayurveda does not mention the word pH, but it talks about acidity. The logical inference is that it refers to pH. pH and temperature, we know the important role it plays from a modern science standpoint as well in metabolism and transformation. These are not completely new words, dryness, fluidity, adhesivity, size, weight, stiffness, viscosity. These are not new words, except that they are not used as a metrics that as Ayurveda uses it, right? And psychological, of course, you can see some familiar terms here. So what is important to note here is Ayurveda never delinks mind and matter. In the worldview of classical physics, mind and matter are 2 different entities. Whereas in Ayurvedic understanding, mind is part of its understanding of humans. So the parameters under VPK are connected with each other because you remember that the worldview adopted by Ayurveda is interconnectedness. You see this running as an undercurrent to everything that Ayurveda talks about and uses. So V1 to V7 or some parameters under Vata, P1 to P7 under Pitta, and K1 to K7 under Kapha. So the job of the Ayurvedic Vaidya is to make sure that this network is in balance. When it is in balance, it denotes health, and disease is a system perturbation in Ayurveda. And how do they use this to manage health and disease? It's very clever of our ancestors to have done what they have done. Since VPK is used for understanding the human health, the human system, they have brought all factors which impact health and disease onto the same platform of VPK. Everything that plays an important role in understanding and impacting and managing health and disease are classified, understood in terms of VPK. So for example, activities are all classified in terms of what changes it does to VPK. So exercise increases Vata, exercising in hot weather increases Pitta as well. Happy frame of mind and sedentary lifestyle will increase Kapha. Stress, strain and worry will increase Vata. Negative emotions like anger and jealousy will increase Pitta. So when I say Vata, Pitta or Kapha increased or decreased, it means that certain parameters under them are increasing and decreasing. Seasons are also classified in terms of Vata, Pitta, Kapha. You can see that as the seasons change, There are parameters under Vata, Pitta, Kapha which undergoes subtle change. These are not gross changes, but these are subtle changes. Clinical symptoms are also classified in terms of Vata, Pitta, Kapha. I have given some examples here. If it's a shifting pain, then it is Vata associated. If it's a burning pain, it's Pitta associated. If it is a dull and heavy pain, it's Kapha. So all clinical symptoms, it could be rheumatoid arthritis, it could be multiple sclerosis, it could be any autoimmune disease, it could be a simple fever, cold, cough, headache, loose motion, constipation, inflammation, ama. Everything, all the symptoms are classified in terms of VPK. We should remember that the symptoms we see now or experience now, they have not undergone any change in the 5,000 odd years. The same symptoms were there 5,000 years ago as well. So there are no clinical symptoms which can come outside the purview of this classification of VPK. In fact, VPK is a theory. It's not just a concept, it's a theory of Vata, Pitta, Kapha. There is always a question as to how Ayurvedic Vaidyas can diagnose diseases which were not known during the time of Charaka and Sushruta, but which are recent phenomena. The answer is very simple. We all study the theory of addition, subtraction, multiplication, and division in our primary school. It is this very simple theory which is used for solving complex problems as well, right? So, to you all, the theory of Vata, Pitta, Kapha may sound very broad-based. It is broad-based, very simple and naive, considering the complexity of the diseases, but these are used for diagnosis. How do they do it? So when a patient comes to the Ayurvedic Vaidya showing with a set of clinical symptoms, from the symptoms, the Ayurvedic Vaidya finds out which of these have gone out of balance, because at the end of the day, as far as Ayurveda is concerned, an imbalance in Vata, Pitta, Kapha causes diseases, right? So they will know whether Vata or Pitta or Kapha or a combination of these have gone out of balance. In fact, from here they can go further back and identify the diet and lifestyle which have caused this imbalance. So from the symptoms, Ayurvedic Vaidya goes to the root cause of the problem and diagnosis is made. So this is just a small example of how symptoms in cardiovascular diseases would be looked at by Ayurvedic Vaidya to make their diagnosis based on Vata, Pitta, Kapha. So once the diagnosis is made, everything becomes very easy. So for example, if it's a Vata disease, then medicines would be made of ingredients which decrease Vata, and food and lifestyle activities which decrease Vata have to be followed, and which increase Vata have to be kept out. So this is how treatment is strategized. So there are physical interventions and there are also non-pharmacological interventions. In terms of diet, I'll wind down in another 3-4 minutes. So let me change gear and talk about sustainable medicine, which is the future of healthcare. Why you look at this? I don't have to, it's self-evident. Why there is a lot of talk on sustainable medicine. One of the most significant contributors to climate change is the healthcare industry. And it is an emerging field that addresses healthcare's environmental, social, and economic impacts, right? So sustainable health and clinical care, they are now talking of preventive measures, patient self-care, lean management, and low-carbon alternatives. So what does Ayurveda have got to say about this? Ayurveda uses resources in a very, very sustainable way. It is very strong on preventive measures. It uses non-pharmacological interventions. There is no energy required for it. There is no equipment required for it. It talks about therapeutic nutrition. Again, there is no need of extra charges for this. It uses the medicinal plants maximally, and it talks about using the local flora and fauna for the medicines. So if you look at sustainable health and clinical care, there is a tick for all these factors from an Ayurvedic standpoint. So I'm coming back to this ending with these focus areas we started off with, and you can see that Ayurveda is pretty contemporary. All those niche areas that modern medicine is talking about is being dealt with in Ayurveda. So let me stop here, and I thank you all for listening to me and for giving the opportunity to the organizers.
Thank you, ma'am, for such a wonderful lecture and making us understand about the concept of Ayurveda and how holistic the health concept is there, especially about learning the concept of Vata, Pitta and Kapha. And you rightly said about the Shotha and the channels. And we also know that our body is actually made up of membranes. If you know the area of membrane, it is going to be 10 million square meter. So, and all the membranes are equipped with the channels. So you can understand the concept. So if there are some quick questions for ma'am, if you want to understand a little bit more about the presentation. Anyone? No? Yeah, yes.
Hello, ma'am. Thank you. Very nice talk, ma'am. Ma'am, myself, Dr. Raghvendra. Ma'am, I just want to educate myself. I don't have question. Ma'am, is there any traditional knowledge-based things which provide knowledge about this Ayurvedic science, Ayurveda?
See, there are
Because
classical.
there is a Directorate Traditional Knowledge Digital Library. So this thing included in that library?
See, there are in TKDL.
Yeah. Yeah.
You're talking about?
Yeah, TKDL.
TKDL.
Yeah.
It's an initiative of CSIR, wonderful initiative where they are documenting, many of the texts and a lot of information.
Yeah.
But as of now, I think if I'm right, it's not. It cannot be accessed by everybody as of now.
Yeah.
They are trying to open it up, but it's a wonderful initiative of CSIR, where there's extensive documentation of not only textual information, but also from folk knowledge. Yeah.
Thank you very much.
Thank you. Thank you, Professor Ajaya Sundar, ma'am.
Sir, I have. Ma'am, you said you explained the multi-dimensional understanding of health with Ayurveda, where you said it's a physical, mental, and social well-being. So, may you please throw some light on social well-being through Ayurveda? And another question from my side is you focused mainly on VPK. So, our emotions, our behavior, they all lead to these changes in VPK. And a similar concept is with the release of hormones through our emotions. And so can we correlate this VPK release or with the hormones?
Yeah. Thank you for the interesting questions. So social well-being is interpersonal relationship and how an individual behaves in a society, because how an individual behaves in a society will affect the health of the society as well, right? So there is the kind of discipline that needs to be followed and how a mind has to be tuned to positive thinking. All these are dealt with in Ayurveda, because Ayurveda also talks of non-pharmacological interventions. right? So yoga is there, meditation is there, right? And Ayurveda also talks about counseling. So individual's behavior is something that Ayurveda lays a lot of emphasis on, and also how a society has to function. Remember that, Ayurveda is closely related to the Vedas, our Vedic knowledge. And so how a society has to function, what kind of things are right, the dharma, the right and wrong, who should do what, who should not do what, all these are embedded in the well-being of the society. So these are dealt with in Ayurveda. You may not find elaborate discussion of these in the classical texts, but you have information on them, and then we should link it up with the other text, right? And as far as correlating hormones with VPK, see, as I said, VPK denotes a functional classification, right? So when you say function, structures are gross and subtle are part of it, right? And so there can be, it should not be difficult to to correlate the two. But I would also like to make a point that these kind of correlations may mean nothing for Ayurvedic Vaidya. So these are of either intellectual interest, academic interest, and it may lead to something more as far as modern medicine is concerned, because these are all topics that's coming up in a big way in modern medicine, and modern medicine is still trying to understand what mind is, right? So one needs to have an understanding of mind to be able to talk about behavior and mental health. And Indic sciences have dwelled into mind and consciousness extensively. So their understanding of mind and consciousness is factored into the healthcare framework that they have evolved. That people may not want to believe it or understand, that is a different thing. So there is something called the limitations of people's understanding is not the limitation of the science. The limitations of the Ayurvedic Vaidyas is not the limitation of the science. So what I talked about today is the science. What are all the metrics it uses, what is the framework it has, and what are all the factors it has factored into the framework, and how it utilizes that to understand and manage health is what I understand. So, it's a area wide open for all kinds of very, very interesting research and work. And as Dr. Bharati also said, these kind of research will also bring in clarity for the Ayurvedic community. And also as scientists, these are all very interesting questions for us.
Okay, thank you, ma'am. Thank you so much.
Ma'am, may I ask one personal question? Because initially you explained that you did your doctorate in physics and then turned to Ayurveda. So what drives you from physics to Ayurveda? Just it's a personal question.
See, although so I did my PhD in physics from University of Cambridge, UK. And I was into
25 minutes
biomedical
presentation, 5 minutes
physics,
per discussion.
biomedical NMR. So at some point in time, I felt I should have a medical degree. So my first thought was, of course, for studying MBBS. Although I got admission for MBBS, I decided I will do Ayurveda. I just wanted to know what it is. I wanted to know whether it's actually a science, as Ayurvedic people keep telling, or whether it's not a science. So it was a big risk because I was at the peak of my career at that time, and somebody told me it's a professional suicide you're doing. Anyway, I told them that we live our life once, and I may as well do what I felt I should do. And I think it was a good decision. It has really opened up my...
Thank you so much, ma'am. Thank you so much. It was really a lovely
Thank you so much,
presentation.
ma'am.
Thank you so much, ma'am. Before we invite our next speaker, I would like to inform the registered participants that you can collect your kits from the registration desk after we finish this session. On-spot registration participants. Now I would like to invite Dr. Santosh S.R. Nair, Department of Basic Principles, CBPACS, New Delhi.
Good morning, everybody. Chairman, sir, I have a request. The ambience is very dark, it is very intimidating, and it is very depressive, sir. Yeah. It is winter, depressive season. The ambience is also depressive. And the subject Ayurveda, the Ayurvedic people, we speak in a very archaic language, very primitive language. All these things leads to the focal topic today is Ama and Shotha. I am focusing only on Ama. I don't know how many of you have a flavor of Ayurveda, I don't know. I already said that the Ayurvedic people speak in a very primitive language, dosha, dushya, dhatu, mala, all these things. Anyway, 3 basic components are there in a human body. Already she has explained. The VPK, that is known as dosha, then comes dhatu, then mala. The dosha, they are very functional one. They are not tangible entity actually. They are not functional. Dhatu and mala, they are structural one. For example, the muscle, we Ayurvedic people call it mamsa. The bone, we call it asti, like that. 7 kinds of dhatu are there, and mainly there are 3 malas. Where is the light? Oh. Can you put off this one? Oh. Now it is fine. I can see your eyes. Because eye contact is very essential component of a. So I can see how many of you are sleeping. By that parameter, I can stop my presentation. So the pitta, the VPK, the second one is pitta. Pitta is related with Agni, and it is the main function of pitta is transformation. Without Agni, the transformation is not possible. To understand Ama, you should know the concept of digestion and metabolism mentioned by Ayurveda. According to Ayurveda, the digestion and metabolism has 2 stages. The first stage happens in your GIT, gastrointestinal tract. We call it Jadhara Agnipaka. Ayurvedic, we say Jadhara Agnipaka. Jadhara means the stomach, the intestine. The Agni means the corresponding pitta functioning in that area. So the function of Jadaragni, Jadaragni-paka. In modern science, she call it digestion. Then comes the metabolism, what she called. Ayurvedic people, we call it as Dhatogni-paka. It happens in the cellular level. So, according to Ayurveda, there are 3 kinds of Agnis. One working at the GIT level, we call it Jadaragni. And second, we call it Dhatogni, that functions at the cellular level. One more kind of Agni is there, that is we call Bhutagni. According to Ayurveda and according to philosophy, Indian philosophy, each and every object is made up of 5 elements. Akasha, Vayu, Agni, Jal, Prithvi. We call it Pancha Bhautika. So each and every object is Pancha Bhuta in nature, Pancha Bhautika in nature. It has an Agni component. So any philosophy student who has learned Nyaya Vaisheshika? No. Nyaya Vaisheshika is one of the Indian philosophies. According to them, any transformation needs two kinds of Agni. One, an external source of temperature, and second, an internal source of temperature. For example, mango. It is green mango. The external source of temperature for the ripening of the mango is sunlight. The sunlight stimulates, the mango is Pancha Bhautika in nature. It has an Agni component. It stimulates, it disintegrates the mango and kindles the Agni component of that mango. And the further changes happens. The green color turns into yellow or something else, and the smell of the mango changes, and the taste of the mango changes. So any transformation, two kinds of temperature or two kinds of Agni is essential. Same with digestion and metabolism. When you eat a mango, the Agni in your GIT functions an external source of Agni. We call it Jadaragni. And the mango has an Agni component. We call it Bhutagni. So Jadaragni stimulates, disintegrates the mango and kindles the Agni component of the mango. Then the function of both Agnis goes together, and at the end of the digestion, that is Jadaragnipaka, and the simultaneous process of Jadaragnipaka and Bhudagnipaka, the mango changes into a dhatu. We call it Annarasa or Poshakarasa dhatu. Annarasa or Poshakarasa dhatu. According to Ayurveda, this Poshakarasa dhatu is further subjected into Dhatugnipaka. There are 7 kinds of dhatu. Corresponding to 7 kinds of dhatu, 7 types of Agni are there. These Agni stimulate the corresponding Poshaka dhatus, and changes happens. The Annarasa, the mango, will become muscle, tissue, bone, your blood, your bone marrow, like that. So according to Ayurveda, there are 2 levels, Jadaragnipaka and Dhatugnipaka, completed by 3 kinds of Agni, Jadaragni, Dhatugni and Bhutagni. Bhutagni means the Agni of that object. So when, according to Ayurveda, when this Agni is weak, the proper transformation does not happen. So the product, the partially digested, partially transformed product is known as Ama. So Ama happens at any level, at the Jadaragni level or at the Dhatugni level, mean at the cellular level. For example, the general example is fevers, the common cold and fever. You feel an indigestion. Some belching will be there. It shows the Ama at the Koshta level. Dhatugni level, creatinine. It's a mala produced at the cellular level. It should be removed, it should be expelled. To remove this Ama, Ayurveda has some group of drugs we call Deepana and Pachana drugs. The Deepana drug stimulates the Agni at any level, at the Dhatugni or Koshtagni level, and Pachana drugs helps to remove that Ama. According to Ayurveda, at least 4 entities can be identified as Ama. Amam Annarasam Kechit. Amam Annarasam Kechit. Annarasam means the product, the end product of Jadaragnipaka is known as Ama. So in her language, the chyle or chyme may be called as ama. It's physiological entities, not a pathological one. Because chyme and is partially digested and they are the end product of digestion at the stomach, then comes the small intestine. Malasanchaya, kechittu malasanchaya. The accumulation of excretive product is also known as ama. So the urine, when the retention of urine happens, that particular urine you can call it as ama. When uric acid increases in your blood, you can call it as ama. According to Ayurveda, the dosha, they have some specific seat in the GIT. Ayurveda classifies GIT into 3 parts. Broadly classified into Amashaya and Pakvashaya. The Amashaya has again 2 parts, Urdhva Amashaya and Adhoga Amashaya. Urdhva Amashaya means stomach and above. The duodenum, etc. is known as the Grahani we call. It is Adhoga Amashaya. Then the large intestine, it is known as Pakvashaya. According to Ayurveda, Urdhva Amashaya is the main functional seat of Pitta, Kapha. Adhoga Amashaya is the main functional area of Pitta, and Pakvashaya is the main functional area of Vayu. Some etiological factors, due to some etiological factors, these doshas increase in the GIT. All these are imaginary. You cannot see in a dead body whether whether Vayu is in the Pakvashaya, you cannot see it. So, due to etiological factors which increases Kapha, the increase of Kapha happens in the Urdhva Amashaya, in the thoracic area. Technically, we call it as Chaya. Still, you follow that etiological factor, the Kapha will again increase. It will come up out of the GIT. We call it as Kopa. Then it spread all over the body. We call it Prasara. And when it gets some structural deformity, it will accumulate in that area. We call it Sthana Samshraya. Then these dosha, then dhatu, etc. will take part in a chemical action. We call it dosha dushya samurchana. According to some authorities, the third meaning of ama is the initial product of dosha dushya samurchana. It is a pathological one. So, according to Ayurveda, each and every disease, not each and every disease, most of the diseases are caused by Agni Mandya, which leads to ama, and ama leads to various kinds of diseases. In Ayurveda, specific symptomatology is available to identify which dosha is in ama, whether vata is in ama state, pitta is in ama state, or kapha is in ama state. So, all these things comes under ama. Some entities, in some context, ama is used as a physiological meaning to indicate the primary product of digestion. In some cases, it is an accumulation of the waste product or excretory product of your body. In some context, it is used to refer the initial pathological process, and generally, we use it for indigestion. According to Ayurveda, there are 3 kinds of indigestion caused by vata and pitta and kapha. The indigestion due to kapha is known as ama. So, that term is also used to indicate a very specific disease. So, I think this is a small introduction of ama. My time is over, I think. Actually, Ayurveda has very unique and funny concept. One of the concept is Pratinidhi Dravya. Pratinidhi Dravya means, in Ayurveda, I am completing. Pratinidhi Dravya means, one oil is there, we call it Chandanadi Taila. Chandanadi Taila. So many ingredients are there. The main component ingredient is Chandana. Chandan is very costly and it is not easily available. So, according to Ayurveda, you can substitute chandana with another easily available ingredient. Usually we substitute with the Rakta Chandana, which is cost effective and easily available. Actually, I am a Rakta Chandana here because the School of Microbiology, JNU, CCRS were planned to prepare a Chandanadi oil today. But yesterday evening they came to know that the main ingredient Chandana is not available. They searched all over Delhi and found that one Rakta Chandana is available, and I am that Rakta Chandana, and I am very grateful to the original Chandana. I don't know who are. And I am also grateful to the organizers that they thought that I am a substitute, ideal substitute for them. Sometimes the substitute was better than its original one. Thank you very much.
Okay, thank you so much for nicely explaining the ama and also giving nice examples. So, do you have any query for Dr. Santosh? Any comments?
Hello, sir. Firstly, thank you for such a wonderful discussion on this topic. So my question is, जो हम लोग अग्नि मानदेय की जो बात करते हैं, which is causing this आम, तो मेरा ये प्रश्न है कि पित्त कफ प्रकृति टाइप हो गया या पित्त वात प्रकृति टाइप हो गया, तो क्या उसमें ये आम का जो फॉर्मेशन है वो कम देखने को मिलेगा? और दूसरा ये है कि वैसे सीज़न जहाँ पे पित्त में कमी आती है सीजनल वेरिएशन के कारण से, वहाँ पे क्या हम लोग को आम में कमी देखने को मिलेगी बनने में?
बिल्कुल. According to Ayurveda, there are basically three kinds of prakritis classified based on Vata, Pitta and Kapha. Pitta prakriti, generally ama is less. In a Kapha prakriti, more chances are there. And during Grishma Ritu, means summer, we have a very less digestive power, ama is again possible.
Thank you, sir.
I think it's a very nice conversation, I would say that. So, my question is regarding ama. If you look at the way you defined ama and what we have, although I didn't get a chance to hear your whole talk, but ama seems like it's a set of all the excretory product, or the catabolic product of
not all the
cellular
excretory
physiology.
product.
Yeah.
If an excretory product, it is properly excreted out from our body, it cannot be called as ama. It is mala. When a mala is accumulated in our body, we can call it as ama. It is a condition of ama. It is a type of ama.
So any non-physiological accumulation of biomolecules could be called as a ama.
Pardon?
So non-physiological accumulation of
Yes.
any excretory or metabolic or catabolic product
It
could
can be
be
called as
considered
ama.
as
Okay.
ama.
Ama, yeah.
Okay, thank you.
Thank you very much.
Yeah, Professor Santosh, it was really a lovely talk, and you said the Panch Bhut, the Panch Tattva, and they are related to Ayurveda, and you explained Agni in detail. So I just want to know that whether other fours, they are related with Ayurveda or physiological system of the body?
According to Ayurveda and according to Indian philosophy, each and every object has all these 5 components, including my body. For example, muscle tissue. It is predominant in Jala and Prithvi, less in Akasha and Vayu. In urine, it is Jala and Agni. It is not easy to understand. For example, ghee, it seems it is a Jala predominant. No. According to Ayurveda, it is Prithvi predominant. Not according to Ayurveda and even philosophy, it is Prithvi predominant. I think it is gold, very risky. Gold, according to Ayurveda, it is not Parthiva. It is Agneya, it is predominant with Agni.
Okay, thank you, Dr. Santosh, for the nice discussion and making us understand about the ama. So the message is, don't only focus on the intake, but also it's a proper digestion and excretion that is also important. So our focus should be there as well for the holistic health. So thank you very much. Next speaker you can make.
Thank you so much, sir. Now I would like to invite Dr. Neel Sarovar Bhavesh, Group Leader, Transcription Regulation, ICGEB, Delhi.
So thank you, and it's nice to be here at my host. We are the host, and I'm standing here, and it's a CCRS workshop. We have a couple of projects, so I'll mostly talk on what we have been doing in the first one. And one of the big challenge which we face nowadays is the mainstreaming the traditional knowledge into modern health practices. So how we integrate them, that's just one of the challenges and how we can overcome those things. And so this is, those who are from ICGEB know this is the monsoon view of ICGEB from the top, and you can see somewhere here JNU also, we can actually see them. So why it is necessary? Because if you look at the Ayurveda economy, bioeconomy, it is growing at almost, it is expected to grow at a 12% CAGR. So that is a quite good rate, and it would contribute immensely to our GDP. And there are products already in the market, and there are different things associated with this commercialization which are also generating revenue in that. And there are companies who are able to raise good amount of money from the VCs, so it's a growing sector. Now coming to the present part, precision medicine. The precision medicine means what kind of a definition which ex-president of the United States has given, the providing the right treatment at the right time to the right person, taking into account patient's health history, gene, environment, and lifestyle. So this is what he was talking about, the precision medicine. And when it comes to Ayurveda, we already think of these things quite often. This is what is the basic fundamental principle which Ayurveda actually works on. So precision medicine, when we talk about, when you look at pharmacokinetics and pharmacodynamics variability, there are subject phenotypes, weight, body, and age, organs. The subject genotypes are polymorphism and enzymes. Pharmacodynamics variability are polymorphism in drug target. In morning, Dr. Padhe was talking about different drugs and downplay pathway of target polymorphism. Disease phenotype, expression level of drug targets, disease response or progression, indirect treatment effects, comes to pharmacodynamics, heterogeneity of disease mechanism. Disease genotype comes to driver mutations of disease heterogeneity, which most of you must have seen during the COVID, the mutations, how it was coming very fast. This is disease genotype. Lifestyle environment, medications, diet, smoking, and there are others, adherence to drug formulation routes. So when it comes to precision medicine, all these things have to be taken into account when we deliver the medicine precisely. The current global status of the present thing is precision medicine is mainly in cancer, mostly which are being used in breast cancer, colorectal cancer, GI cancer, tumors, kidney, and other things. Other diseases which things are being tried are type 1 diabetes, which have these mutations, or in cystic fibrosis. One of the big case which for the precision medicine has worked for this lady, she was diagnosed with the stage 4 lung cancer in both lungs. It kept growing after standard chemotherapy. participated in a clinical trial of this casinotinib, and it worked very well for her. So it was, what they did actually, they sequenced the whole genome of this lady and found out what mutations she was carrying, and they used this medicine for that. And it worked for her. And it worked so much that almost 50% of tumors disappear. So this is the power of genetic information and the precision medicine. Why doesn't casinotinib work in all cases? Because it doesn't have that target. So it cannot be prescribed for everybody. It only depends on the ALK fusion, which this is kinase, and demonstrates the potential for personalized medicine. So precision medicine ecosystem is you not only the genotype part, but you take all the health informations, data storage, analytical visualization, and finally this goes iteratively, and outcome is the health outcome is that person should get healed very fast. This is the outcome of the health. The precision wellness is another metaverse where you want that person should remain healthy, and this is the sphere where you see the wellness is interrelated to physical, mental, social, and spiritual well-being, not only the physical what we talk. And this is the concept taken from the Ayurveda. Defined as the integration of all these factors, focus on designing treatment strategy, disfering the individual genetic, social, and other status. Evolution of precision medicine, it evolved from the precision medicine part and provide practitioner a chance to improve human well-being, not only cure the person, but improve the well-being, taking everything in this account, and facilitates the combined analysis of various genes like all the omics, including the microbiomes, which has more number of cells in our own cells in the body. So Indian traditional knowledge actually encompasses the complete realm of science, starting from physics, chemistry, and biology it goes, and there's what is integrated by this computational biology. So you have different analogy from the modern and the traditional knowledge system. Indian traditional knowledge, you see all these people who have contributed immensely in the different areas of physics, chemistry, and biology, and medicines. So the medicines and physiology, what we see from Charak and Patanjali and Sushrut, the father of surgery and anesthesia, Patanjali Yoga came, Charak Ayurveda we have, which we are talking in today. And human body, according to the two different kind of traditional knowledge, one is European, when the Vinci has actually talked of the human body, and what Charak has taught. So this is evolving, and it's growing even in ancient times. So coming back to the Ayurveda, what it talks actually, everything in the universe is made of Panch Mahabhutas, Akash, Vayu, which I think has been discussed, Agni, Jal, and Prithvi. And this correspond to spaces, where Akash actually correspond to space, Vayu denotes to the movement, Agni functioning of enzymes and other thing, Jala makes the plasma, saliva, and all the biofluids, and Prithvi makes the rigid structure of the body. They are governed by three fundamental energy, namely the Sattva, Rajas, Tamas, which makes Trigunas. The Malas are Purusa, fecal sample, urine, and sweat. Sometimes many of us actually forget about this sweat in our daily life, because we are used to living in our AC, air-conditioned environment, and we are not able to actually get this thing out easily. So this is what Charak had talked about about Prakriti. So when you look at this part, actually all these things actually relates to the modern biology which we talks. Like sucrasonate is allelic segregation, the meiosis part. Karl Garbha says intrauterine epigenetic changes. What are the epigenetic changes taking place? Atur Ahar Vihar is talking of the metabolic selection of the alleles and epigenetics. And finally, the environmental is Mahabhut Vikar, environmental epigenetics. So that's why all this epigenetics comes into the picture. Even this was there in traditional, if you look at the Manusmriti, that said marriage should not be the woman who is related anywhere in the last 6 generations of the man's mother. This was because you don't want inbreeding problems to arise. So these concepts were actually there. So Prakriti, if we talk, it's determined by following factors, mainly the condition of the sperm and ovum at the time of conception, condition of uterus, food and regimes adopted by mother during the pregnancy, and predominance of Mahabhutas is comprising the fetus. So this actually, what is the concept? Basic, I'm talking, it's a big concept, but I'm trying to summarize in a small nut that what it means to Prakriti, how it is this. So these are the things, Vata, Pitta, Kapha, which we're talking, and accordingly these Prakriti types are, they are pure Prakritis ones, but there are mixed prakritis like Vata Pitta, Pitta Kapha, Vata Kapha, and some prakriti. If it comes to molecular biology part, the modern science, what it says, that your DNA is like a hard disk, all the information is stored, and from this hard disk, the selective information is actually released into the cell in forms of RNA during transcription. And these things actually translate into the protein, which are the workhorses of our body. So these are like information storage, which pass on from generation to generation. These are the information carrier, time dependent and space dependent carriers, and finally the information executed the proteins. And they are not in isolation. They talk to each other. Everybody is talking to each other inside the cell. And it's a complex network of biochemical pathway, which anybody who reads know how they are talking to each other. And every day, new and new additions are being made in this biochemical pathway. And it leads to different kind of studies which we do. If we want to study the genes, we do genome part. If we want to do transcription, the information carrier, we do transcriptomics. On the protein, then it's a proteome and metabolome for understanding all the metabolites. So human pyramid of life is like you have the genes, then you have the proteomes and enzymes, only few of the proteomes can be bigger, and then very small number of the chemicals, which is the end product of all the metabolic pathway, which comes inside. So generally what we see nowadays means we have to accept the harsh reality. We see of all the people talking about traditional knowledge versus our allopathy medicine, which is a fight. Most of the times what you see in traditional medicine is a host-directed therapy. You allow the host to modulate itself, autophagy or whatever way, to get cured, while in allopathy medicine it's a pathogen or the tumor-directed therapy which tries to kill the pathogen or the tumor. So generally that's why it is a fight. I give an analogy with the high pollution which we all live actually in Delhi. So if high pollution, if you have something like a short-term remedial solution is, this is what you see in November, December, October, November, everybody is coming to that. Or everybody has in this house this type of things. But this is a short-term solution. A long-term solutions are like this. You have to have more number 3. So this is what is required. You cannot have a short-term solution every time. And same thing, instead of this verses for a long-term thing, it has to, both the thing has to shake hand. That is for the well-being of human being. If both comes together, because they work in different realms of the thing. One is host and one is pathogen director. So they have to come together and then talk and then go together. In this regard, one of the project which was initiated by the CCRS, I think Dr. Srikant was there, he actually helped us a lot in that. And these institutes actually, JNU, TDU and ICGEB combined together to explore this thing and at least find out how we can go about it. So we have a kind of a MOU signing later that time. One of the early stages, which was published from ICGEB, in the group of Mitali and Bhavna, who will be talking today. So they worked on this EGLN1 involvement in high altitude adaptation. So this was a kind of how Ayurvedic Prakriti is related to the modern science actually. They can actually confirm that. Now this is the last year, in February has come. It is time to admit that genes are not the sole blueprint of life. And this is what Ayurveda has been talking about all the time. There are many things which comes into the picture. And now even modern science is thinking that it is not that only that you read all the letters of our gene and you understand the life. It's not like that. So where we thought, okay, let's look from the modern perspective, the molecular signatures of Prakriti. All the omics, proteomics, transcriptomics, long non-coding RNA exomes, gut metagenomics, integromics, and finally building up the model and see how they relate to Prakriti. Hypothesis is the physical feature, clinical and biochemical parameters and molecular determinants. So all these things are like transcriptomes or Prakriti subtypes are qualitatively and quantitatively different from the other subtypes. It means each Prakriti should have a different molecular signature. That was the hypothesis when we started the project. And aim was to identify these things actually, to see if there are different signatures or not. So to identify potential biological markers for different Prakriti, to identify pathways and predictors for different Prakriti, and establish a system correlation between metabolome, proteome, transcriptome, offer different prakriti. So a good quality sample is required. If you don't have a good quality and right subject, everything can fail. And this is what is this lays all the solid foundations of good quality, correct sample collection and prakriti. And this way CCRS has actually worked very hard with all the centers, different places, to have a very good sample quality. And finally, it helped us, like JNU and us, to go into a different omics. And if you don't have this thing, good quality sample, this kind of phenomena can happen. So samples quality is really the key to things. And we had really made a very robust inclusion and exclusion criteria for that study. Since it was the first study of its kind, we have included only male volunteers between healthy adult type, this 18 to 60 years, willing to provide, of course, written consent. And we have excluded all these things who may be not healthy. And this is, now we will see, we have to coin a word apparently healthy, because it's very rare to find a healthy person. I think Rana Pratap Singh will talk on this detail, they're apparently healthy, because most of us are actually in some or other problems. So apparently healthy, but generally excluding people like this from the study. And the sampling sites were different places. There are 7 centers in India, including a high altitude center in Gangtok. So different centers like Jalandhar, Lucknow, Bhubaneswar, Mumbai, Shillong. So all these things different. So to avoid any kind of problem coming out from a bias toward a site in India. CCRS in this project developed a nice automated prakriti assessment protocol, and this is freely available thanks to the CCRS. You can go and log in and determine. Lita is also sitting here, so you can go and log in and you can objectively determine your own prakriti from this portal. So this is one of the very good thing developed by CCRS. It is publicly available nowadays. In addition, we have selected all the humans for this study. Routine biochemical examination, which is generally done in clinical setting, were done like hematology, LFT, KFT, other assessment, X-ray, urine routine and microbial examination, physical examination, lipid profile, stool test, all things were done to find out the parameters of that particular human beings. So we had 6 participants from 7 prakriti at 7 centers. So total 294 humans were enrolled, apparently healthy humans were enrolled in this study. 10 ml of blood, urine, 2 to 4 grams of fecal samples were collected in the morning between morning hours in both times of the year, Uttarayan and Dakshinayan. Participants were instructed not to eat before sampling. Collected samples were transported to JNU ICGEB in a cold chain maintained logged environment, so the temperature at every 1 hour is logged in the sample transportation. All the sample collected were trained medical practitioners at the CCRS sites. The hospital with proper monitoring. Information sheets were collected with questionnaires. Why 2 ions? People will ask, why you have selected 2 ions? What is the rationale? Of course, Ayurveda has this rationale, but even in the modern medicine, which was published in 2015, they found this in Nature Communication paper that widespread seasonal gene expression reveals annual differences in human immunity and physiology. So there is a difference in the transcription level of this human immune system. So the seasonal variations are seen. So this is validated through the modern analysis, and that's why, and we also, this is a good basis for that, we selected the Dakshinayan and Uttarayan also. Sample collection, trait analysis, questionnaire, sample collection, initial biochemical analysis, sample storage, and sample transportation, all were completely done very well. The approach, what was the approach for that? We use NMR-based metabolomics because you really don't play around with the samples. In mass spectrometry, you play around with the samples, you do chromatography, and then you do. So there are chances of modification and other things. So NMR is a native, you take that same thing and you do metabolomics. You have a level-free proteomics for finding out what proteins are there and what is the quantity of that. Metagenomics was done for sequencing, NGS metagenomics for the things, and NGS was also used for the transcriptomics, which Rupesh has done. So a kind of a workflow, you have the sampling site from the 7 CCRS institutions. Ethical and biosafety approvals were taken, 294 individuals, both in Uttarayan and Dakshinayan, so it multiplies So 588 sample collections were done, and all these analysis was done later. And finally, the multi-omics data integration is being done. I think Dr. Hemant Kushwaha is standing back the back. He is doing the integration part, and finally, statistical analysis. All of this will be find out to see the pathways. So data integration, although it looks very simple, means it sounds very simple that you have a nice genome, transcriptome, proteome, and you can just integrate them. And this integration, you think that, okay, genomics is you have epigenomics, proteomics, transcriptome, metabolomics, and you can build this network which are actually you get it from different things. And microbiome actually adds completely a new dimension because what information you get all this information for roughly around 30 trillion cells. But this one has something like more than 38, 39 trillion cells, microbiome. So you have a completely new set of things coming from this. And this will integrate to that, and you get information from either NGS, mass spectrometry, NMR, or mass spectrometry. And you can try to build it, but it's not that easy. You have to have a complex differential equations and complex matrix analysis. I think sometime Hemant can talk on this, to build up this network and find out how they look like. So what are the deliverables in this things? Deliverables, what we proposed at that time, that new methodology for iteration of different omics data according to Prakriti. So according to Prakriti, we can well have a subsets. Establishment of big data analysis method to convert Prakritic phenomics data in a structural data set, baseline data set, and correlation with omics data set, how to correlate the omics data set with the Prakriti part. And finally, this is the like a holy grail here. Find out the identifier signature of Prakriti, whether we can look at from the data, different data, and say, okay, this Prakriti has this data, mapping two sets directly, set A and set B. This is the ultimate goal of this project. I'll just talk to a couple of slides on what we were doing here, the metabolomics part. We have taken urine, simple urine. You add kind of a buffer so that pH remains during the long time. You add azide so that nothing grows during storage time. You add D2 and TSP for the electronic purposes. And simply you take the urine and put the 60 ml buffer. So we are not doing any processing. The urine as it is, we are taking urine directly in the NMR tube. And once you measure that kind, you get this kind of a spectra. So urine has around something like more than 200 metabolites. It's a complex mixture. And for each compounds, you get a separate lines without purifying, without separating. So you get this kind of thing, and you identify each and every metabolites present in the urine. And finally, you do the PC and PLSD analysis. And this is from our preliminary data. On one hand, we do see a nice clustering. And this I think Hemant has done. Nice clustering of different Prakriti. So we do see a separate clustering of different Prakriti from the metabolomics part, and they have similar result. Transcriptomics, I think Rupesh had, and Sunil had from proteomics, and Arun Kharat had from metagenomics part. So all kind of clusterings are there, and this is, so it is more than what you think. It's a more than 5 TB of data we have generated. It's a huge data, and probably is the biggest amount of data for healthy humans in India in one experiment thing we have collected. This is kind of thing, and analysis is going on, so this will take some time to come up the final data. But this is the preliminary data look quite promising that we do have molecular identifiers for each and every Prakriti. Relevance, of course, the biological, this marker signature of Prakriti will entail a new era in Ayurvedic medicine, where modern molecular evidences would be used to assess Prakriti along with the phenology. So both can go together very well. The traditional part, Ayurveda, and modern can go together. These molecular signatures will be useful in diagnosis, prognosis, and treatment purposes. So, how it is relevant for the quality of life? So now you have traditional medicine, allopathy medicine, Prakriti analysis does for humans, and multi-omics actually identify markers for the human. So multi-omics can segregate humans from their omics data. Prakriti can also segregate from this 10 Prakritis. This can be very well used for precision wellness. This can be used for the precision medicine. And once we merge them, we can actually improve quality of life. And this is the ultimate goal for all the sciences, that to improve quality of life. With that, I thank you all, and if questions, I'll invite you.
Thank you, Dr. Neil, for discussing the Prakriti project and how to have molecular signatures for different Prakriti. So this is a huge project, and the message is the hand holding. You may not imagine how many people were involved in having the data, what is presented. So it's a huge exercise, but the outcome is going to have a huge relevance as well. What we're trying to understand, the precision wellness along with precision medicine, and we are going to have now some more projects based on this pipeline which we are generating for the healthy humans, how it is going to happen in different disease cases, so that will be more relevant. So if there is any question or clarification with Dr. Neil on this presentation.
Yeah.
नमस्ते सर, फर्स्टली थैंक यू फॉर सच एन इन्फॉर्मेटिव सेशन. सर मेरा प्रश्न ये है कि मल्टीओमिक्स डेटा को जनरेट करना इज़ अ टाइम टेकिंग प्रोसेस. तो सर अभी जैसा हमारे देश में पेशेंट लोड बहुत ज़्यादा है पब्लिक हॉस्पिटल्स पे ये, तो मेरा पहला प्रश्न इसी से संबंधित है कि हम इससे कैसे प्रिसीजन मेडिसिन को, जो ये चैलेंजिंग एस्पेक्ट है, इसको डील करें कि पेशेंट लोड को भी मैनेज हो जाए और ये वाला भी रहे. एंड सेकंड थिंग कि जब हम लोग क्लिनिकल ट्रायल डिज़ाइन करते हैं तो उसको उसमें इस एस्पेक्ट को कैसे हम लोग इंक्लूड करेंगे प्रिसीजन मेडिसिन वाले एस्पेक्ट को.
ये काफ़ी रेलिवेंट. सेकंड पार्ट, मैंने फर्स्ट पार्ट में बोलता हूँ पहले. एज पर द जहाँ तक कॉस्ट की बात है या फैसिलिटी की बात है, फैसिलिटीज़ अब काफ़ी तेज़ी से ग्रो कर रही हैं और कॉस्ट भी काफ़ी कम होता जा रहा है. अगर आप देखोगे कि जब पहला ह्यूमन जीनोम सीक्वेंस हुआ था, आई थिंक $1 मिलियन के आसपास लगा. अगर आज आप में से कोई अपने आप को सीक्वेंस करना चाहेगा तो $400 से $500 में आप सीक्वेंस कर सकते हो. तो ये कॉस्ट अब इतना पे आ चुका है, और इस कारण से जहाँ भी कैंसर या इस तरह के चीज़ हो रहे हैं तो लोग सीक्वेंस करें. जहाँ पर ट्रेडिशनल मेडिसिन जो है, एंटीबॉडीज़ हैं, ये नहीं काम कर रहे हैं, वहाँ पर लोग कर रहे हैं. to find out, if you look at the centers in New York, MSKCC, रेगुलर सीक्वेंसिंग वहाँ पर करते हैं. अब एम्स में भी लोग कर ही रहे हैं, सीक्वेंसिंग हो ही रही है. तो इस ढंग से वो हो रही है, फैसिलिटीज़ अब आ रही हैं और फैसिलिटीज़ के अनुसार हम काम कर रहे हैं. ये अब और ये काफ़ी वाइड स्प्रेड हो जाएगा, इसमें कोई प्रॉब्लम नहीं. रही बात क्लिनिकल ट्रायल की प्रिसिजन मेडिसिन की, ये काफ़ी रेलेवेंट क्वेश्चन है, क्योंकि आपको क्लिनिकल ट्रायल कैसे करोगे? एक म्यूटेशन आपको मिला और काफ़ी दिन तक वो म्यूटेशन किसी और पॉपुलेशन में मिलेगा नहीं. एन आपका एन साइज वो होगा नहीं, इतना बड़ा होगा नहीं. सो ये एक उस समय ट्रायल किया जाएगा, या तो फिर हम एक सेट ऑफ ड्रग्स ऑलरेडी हो जिसका पीके, पीडी, टॉक्सिसिटी सब कुछ हो कि ये वर्क करता है, और अगर ये म्यूटेशन होगा जैसे एल, कोई भी जिसमें एल के था फ्यूजन, उसमें बाइंड कर सकता है. ऑलरेडी वो स्टडी हो चुकी हो एनिमल मॉडल्स में या ह्यूमनाइज एनिमल मॉडल सिस्टम में, तब हम उसको वहाँ ले जा सकते हैं. लेकिन हाँ, पर से अगर उस म्यूटेशंस के लिए क्लिनिकल ट्रायल करना हो तो इट्स अ डिफिकल्ट, इट्स अ वेरी वेरी चैलेंजिंग टास्क. आई थिंक इट्स करेक्ट. You don't have an end, you don't have that kind of end, do a clinical trial, because you may find maybe 1 or 2, तो ये नहीं हो पाएगा. ये चैलेंज है, ये प्रिसीजन मेडिसिन में ये रहेगा चैलेंज. हाँ,
थैंक यू सर.
हाँ, हाँ, हम्म.
So how we can cure this lifestyle disorder by using this precision medicine?
एक चीज़ है, अगर आप जैसे लाइफ, एक सिंपल मैं एग्जांपल देता हूँ, मैडम हैं यहाँ पे इसलिए. अगर जैसे कैंसर का है, कैंसर इज ऑल्सो काइंड ऑफ ए म्यूटेशन विच कम्स बिकॉज ऑफ लाइफस्टाइल डिजीज, और 1 या 2 म्यूटेशंस नहीं है, 2-3 म्यूटेशन के बाद अगर आपने पहले अगर सीक्वेंस किया है, आपको लगता है कि 2 म्यूटेशंस हो चुके हैं, 3rd म्यूटेशन कैन बी कैटास्ट्रॉफिक, ऐसा हो सकता है, तो प्रिवेंटिव स्टेप लिए जा सकते हैं. पहली चीज़ ये प्रिसीजन में ये इस हिसाब से हुआ मल्टीओम. आयुर्वेद के हिसाब से आप चलेंगे, अगर प्रकृति के हिसाब से आप आइडेंटिफाई करते हैं आपका प्रकृति ये है, और आपको इस ढंग का लाइफ स्टाइल जो खान पान या रहन वो होना चाहिए, आप उससे डेविएट कर रहे हो तब आप पे समस्या आएंगे. अगर आपको बता सकते आपका ये होना चाहिए तो शायद आप उसको अवॉइड कर सकते हो. तो दोनों तरीकों को यूज करके हम एक ऐसी जगह रह सकते हैं जहां पर हमें शायद वो बीमारी ना हो, इस चीज को हम बता सकते हैं.
So if there are no questions, we thank Dr. Neil and his This talk was very much focused on the Prakriti, and the question what you asked, I think it is going to have a great relevance for the risk prediction. And I think overall goal is to have the Prakriti subtype of each and every individual on the OPD card when we go to the health center. So let's see what will be the time when we reach to that stage, and that is how the precision medicine is going to work. On your OPD card, you have your names, age, blood group, and so many details. But as Dr. Neil said that we all are not same, we are all different. So he had showed you one resolution that at least all of us can be divided into 10 groups, 10 Prakriti subtypes. So whatever problems we have in the modern medicine of either delayed diagnosis, misdiagnosis, or even failure in the treatment, overdose, underdose. So all those things are going to improve if we have such kind of resolution and classification at precision level, what he is talking. So this is what we are looking forward. Thank you very much.
Actually, this has, means for you guys, because you guys are young, it has a big legal and regulatory issues also. If we sequence everybody and we start to segregate that persons on that basis, so how far that information should be accessible in public and data protection, this is again a big thing one has to, because we have the omics data, and you know I have a mutations for something and that's, and I go and apply for a job or something, say, okay, this guy is carrying that kind of mutation, he may not be fit. So that kind of segregation and discrimination. So this also has a regulatory and legal issue which we all have to keep in mind, and we need to have a framework in future that how to protect, because this will come, this data will be coming up. You may have sequenced your own self, and you may be keeping your sequence data in your mobile, and someday your mobile is hacked and that can be misused, your sequence data. What about that? How to protect the data? This is a big challenge. I don't know how we, I don't have an answer for that. Maybe in future, maybe.
So thank you, Neil. I also thank my co-chair, Dr. Bindu from JC Bose University, Faridabad, and all the speakers for their wonderful discussion. Now I think we'll break for the lunch, and then we'll come back after lunch. Thank you very much. There are some announcements.
Thank you, sir. Thank you, sir. Now I would request Professor Rana Pratap Singh to present a memento to Professor Bindu Mangla, Department of Chemistry, JC Bose University, Faridabad. Sir, kindly present a memento to Professor Rama Jayasundar. Thank you, sir. Thank you, ma'am. Now I would request Professor Rupesh Chaturvedi to present a memento to Dr. Santosh S.R. Nair. Professor Rupesh, you are requested to kindly present a memento to Dr. Neil Sarovar Bhavesh. Thank you, sirs. Now we'll proceed for lunch, but important announcement for on-the-spot registered participants. Kindly collect your kits from registration desk now. Thank you.
Good afternoon to all. So लंच के बाद थोड़ी नींद आएगी, थोड़ा बोरिंग है आयुर्वेद, हाँ, लेकिन आज आयुर्वेद थोड़ा समझेंगे, आम का कंसेप्ट समझेंगे. And as I was just discussed with my colleagues that in the morning session you have also. Just give you 1 minute. We'll call the co-chair and chair, and then we can start. Okay. Okay. Who is chair? Okay. Sure. Hello. Yeah. So I will introduce myself first. I am Dr. Babita Yadav. I am a research officer having my qualifications in Ayurveda only. And presently I am working in Central Ayurveda Research Institute, that is under CCRS, Ministry of Ayush. So the topic, the theme was arm and shof. So in Ayurveda, arm is an easy topic. So we choose arm first. And so basically shof is a very vast topic. So we don't touch shof, we touch shof, but in the perspective of arm only. So this is what Gyanpurvak Karmana Samarambham Prashansanti Kushla. Means if you want to become a good researcher, then you must have a knowledge of the concept on which you want to plan your research. So I suppose that everybody must be aware with this term arm. Is it so? Is this word clear to you? For everybody? Can anybody define what is arm? Anybody, please raise your hand and you can say. Dr. Priyanka, no, we will not ask you. Yeah. Anybody? Just the meaning of this word, what is arm? Arm is mango, one thing. Arm is Aam Aadmi Party, another thing. Arm is very arm, that is common. So here the arm is very common. Arm is just like a mango, arm is just like an ordinary person. It is a very ordinary term in Ayurveda. The name is coined arm because it is very common. Without this, there is no disease. So this is so important and this is so common. So arm is that which is partially digested or undigested is known as arm. So this is a definition which is mentioned here. That is the nirukti. Nirukti means how this term derived from. That is nirukti. So the undigested, immature, incomplete, unaccomplished or imperceivable. That is arm. Means anything which is not complete. That is arm. Right? So if we took food, then it convert into ahar, convert into ahar ras. I suppose it is already in the knowledge. My previous speakers have already informed you that how the we took ahar and converted to ahar, then ahar ras to ras and uttar dhatu. So which is incomplete, which is undigested product, that we called it arm. So it is type of disease or a product of the metabolic impairment. The smell coming out of the unformed product or raw substance, it means amasya apakvasya gandha iv gandho yatra. Means there is something which is not good in smell. There is something which is not good for health. Okay, so that is arm. So the smell resembles the smell from burning body at cremation. Chita dhumadi gandhyuktaha means the smell which is very foul, which is very bad. Okay, so how a common man can identify that he or she has arm or not? Okay, have you ever observed that sometimes what happened if we took the food like pizza and all junk food and we eat lot of drinks, then we go to fresh and the next day morning we have very foul smell. Very foul smell. Have you observed it? कभी महसूस किया आपने? So क्यों हुआ ऐसा? वो खाना तो टेस्टी था, स्मेल भी अच्छी थी उसकी, बट अभी यहाँ सब गंदा है. So it's a very foul smelling. So it is अपक्व अन्न, मीन्स जो खाना ठीक से पचा नहीं और जो अनमेटाबोलाइट या पार्शली मेटाबोलाइट जो प्रोडक्ट्स हैं वो स्टूल में आ गए. That's why it is foul smelling or it's smell like a dead body. So this is how it is like. अग्नेयल बलत्वेन दौर्बल्य मीन्स आम क्या है? आम वो है जो उष्ण है, मीन्स which may produce heat. It is not a cold thing. That is the property mentioned. Adhya means the pratham, means it's the first product of, it is the first product of impaired metabolism. Impaired metabolism. If the metabolism is fine, then there will be no formation of arm. So it's the first product of impaired metabolism. Dhatu ras rasakhyam apachit आम प्रचक्षते means जो उत्तरोत्तर धातु की पुष्टि है, जो नरिशमेंट है, वह आफ्टर टेकिंग अ फूड it nourish our body after assimilation, digestion and metabolism and assimilation, it give nutrition to our body. If it is not there, it is harm. Due to the inability of the residual digestive fire, the essence of ahar, that is unrest is not formed appropriately, is known as arm. So is that clear? So now you can define what is arm. Is it fine? Can you define what is arm? Who will tell me what is arm? It is just a revision of the concept only. I suppose in the morning session we already discussed about this in detail. Yeah, please. The food which is not digested properly is arm. Okay, correct. Anybody else? Is this food only? No. So what else? Pardon? Metabolism. So the end product of digestion plus metabolism. Is that arm? No. We must know about this term, then only we will think about the research perspective. If we don't know what is arm, then we can't frame our question, research question. So who else want to tell me what is arm? Where is Santosh Nair sir? He left? Nair sir? Okay. Yeah. Dr. Priyanka, you want to add something in arm? Apakvras. Apakvras is unras. So the apakvanras is the same. It is an unmetabolized product. It is an unmetabolized intermediate product. So that may be arm. Means something which is not good. Something which may be antigen like thing. Something which is toxic to our body. That is arm. So these are the first symptom. How we can identify that we have arm? Very first thing. So very first thing is, this is pain in head and body. So body ache and headache. If we feel after having a heavy meal or after having incompatible food items, if we feel that we have headache, heaviness in head and heaviness in abdomen, it means you may suffer from this arm. So you have the presence of arm. Okay? So this is, these are the other symptoms, the other way of formation of arm. So one way was this, that whatever we eat, it must be completely digested, it must be completely assimilated, and the digestion in the metabolism process will be normal. Then will be no arm. But if there is the impairment, then who is responsible for this impairment? Do you know? Arm is not, it alone is important without Agni. Like without Kejriwal, Aam Aadmi Party has no existence. So same here, the Kejriwal is Agni. If we have Kejriwal, then we have Aam Aadmi Party. If we have Agni, then only we can talk about the Aam. Okay? Got it? अगर अग्नि अच्छी है, तीक्ष्ण अग्नि है, कुछ भी खा लो सब डाइजेस्ट हो जाएगा, there will be no formation of आम. Remember it. If our digestive fire is not at all good, तो थोड़ा कुछ खिचड़ी खा के भी आपको आम हो सकता है. Right? So the digestive juices must be present in a sufficient quantity. Then there will be no impairment in the metabolism. Okay? So other theories, one theory is that is due to the impaired metabolism. Like digestion proper नहीं हुआ, अग्नि नहीं है, अग्नि खराब है, impair है, इसलिए आम बन गया. Second theory ये कहती है, these theories are very necessary to know because it gives you a lead for the research from where you can start your research question. The another theory is, according to Acharya, the Ati Dusht, Vatadi Dush get mutually integrated with one another, and this also result in utpatti of aam, like the formation of toxins happens with the combination of kodrava with the other substance in the same way. That dosh is called aam. Means there is another concept we get. There is an involvement of dosha also. The first was there is an involvement of Agni. The second is involvement of dosha. If the two things means केजरीवाल के साथ साथ मनीष सिसोदिया भी चाहिए आपको, ठीक है, करेक्ट. तो आप नेक्स्ट. So this is what, due to the insufficiency of Jatragni, the digestive juice which is incompletely digested is called aam. Now you get it? Now you get it or not? This is a very simple thing. This is a very simple thing. कुछ भी हमने खाया और वो ठीक से पचा नहीं, तो there is a notorious component आम रहेगा वहाँ. So we must be careful about our digestion. So once you are planning for the research on this particular concept, so you have to start from the digestive tract. Okay? Okay, this is for the lead from where we can start. So you have to look after all the inflammatory or pro-inflammatory markers which are present in the GIT. Okay, so think about this first. So there are the different opinions among the acharyas regarding the formation of aam. Some says called it as apakvan ras. Dr. Priyanka told us कि apakvan ras. Unras means हमने खाया, digestive juices के साथ mix हुआ, अन्न तो हमें पता है जो हमने खाया, फिर वो जो digestive juices के साथ mix हुआ वो एक semi solid और liquid form में आ गया. That is the anras, right? तो जो improper formation of anras है, that is called aam. This is due to mandagni. अभी यहाँ केजरीवाल का रोल है. See, mandagni is agni. I suppose पहले वाले lectures में हम अग्नि के बारे में जान चुके हैं. Our advisor had just informed in the inaugural remark that we have 13 types of Agni. Jatragni, Bhutagni and Dhatva Agni. So Jatragni which is present in the Jatar. So we are all our science students. So we know what is present in the Jatar means the intestine, the complete food tract, what is present in the GI. We have the pancreatic juices, we have the juices from duodenum, we have the amylase mixed food. So we have so many juices, so many enzymes. So इनके साथ मिलके जो food है, that is the anras, right? If all the juices are working in a proper way, it will be metabolized completely, right? There will be no formation of aam. But if the agni is mand, if the digestive juices are less, insufficient in quantity, then what will happen? It will not metabolize properly. The food will not metabolize properly, and hence it will be the root cause of this aam. So then what will happen? This undigested anras gets extremely vitiated by the tridosh, cause abchand in the shotas due to its hyper viscous nature. The appearance of multiple color of aam is the result of combination of multiple dosha. So see, there is a beautiful term here, shrotasya abhishandi. means there will be blockage. There is a concept in science that is a gut leaking syndrome. Everybody must be aware with this. So आप उसे gut leaking syndrome बोलते हैं, हम बोलते हैं श्रोता सभिशंध हो गया, right? So there will be श्रोता सभिशंध. What will happen? या तो वो पूरा बाहर निकलेगा या तो वो कुछ absorb हो जाएगा. पूरा बाहर निकल जाएगा तो कुछ नहीं है, there is no harm. Otherwise, अगर वो unmetabolized particles absorb हो गए through intestine, what will happen? They will vitiate dosha, 3 दोष, वात पित्त कफ, and accordingly they will work further. हम बोलेंगे आम के साथ जब दोष की इन्वॉल्वमेंट हो गई तो साम हो गया, से आम के साथ दोष आ गया, right? So it is very dangerous. Then ama is produced because of inability of the digestive fire for digesting indigested food. It becomes the core causative factor for the manifestation of all disease and also cause heaviness in the heart region. Why in the heart region? Because this arm formation may start from the esophagus. That's why it's a post-sternal structure. So it may start from here. So it is written it's hridadrav or heaviness in chest region. So it is mentioned here. It's the first symptom. Shiroruk, headache, heaviness. Something is not good with our body. Then it may cause body ache. It may cause heaviness in chest. It may cause heaviness in abdomen. There are so many symptoms mentioned in Ayurveda about this. Then what will happen? If it is an ati matra, see the shlok, ati matra pradushtascha doshaha, then what will happen? अगर ज़्यादा मात्रा में आएंगे तो कुछ तो जाएंगे कोश से बाहर, एक्सक्रीट हो जाएंगे और कुछ हो जाएंगे एब्जॉर्ब, कुछ फूड के साथ एसिमिलेट हो जाएंगे और कुछ बाहर निकल जाएंगे, राइट? आइडियल सिचुएशन क्या होना चाहिए? इफ वी फील दैट वी हैव आर्म, तो हमें क्या करना चाहिए? ओवरलोड नहीं करना है, कुछ फर्दर नहीं खाना है, जितना है उसको छोड़ दो वहाँ पे, लेट गिव सम टाइम फॉर द डाइजेशन, बट इट विल नेवर हैपन. हमारा पेट खराब है, हमने हेवी फूड खाया, उसके बाद भी वी ऑलवेज टेक द नेक्स्ट फूड. घड़ी देख के खाना खाते हैं आजकल हम, जैसे ही 1 बज गया हम खाना खा लेंगे, पता नहीं अंदर क्या हो रहा है बॉडी में. सो सी दिस इज वेरी डेंजरस. सो व्हाट हैपन देन? अगर एक्सेस में हो गया, पहले से था, आपने ओवरलोड कर दिया, फिर से वो आम की उत्पत्ति हो गई, व्हाट विल हैपन? अब तो बॉडी में जाएगा, isn't it? And then it will mix up with the dosha, it will vitiate dosha and it will cause pathology. So in the context of Ulcer and Dandalsak, this is Ulcer and Dandalsak are the 2 diseases mentioned in Ayurveda, particularly in reference to the digestive system. Okay? The combination of excessive vitiated dosha and the arm dosha forms stringent accumulation, result in hard pillar-like resemblance of the trunk of the body. Have you ever observed this symptom? Anybody? किसी को लगा है खाना खाने की बाद इतना स्टिफनेस हो गया कि ऐसे लग रहा है कि हम बिल्कुल we can't bend, we can't sit, we can't move. So this is दंडालसक, डंडे के जैसे we feel कि हमारी बॉडी इतनी रिजिड हो गई है, इतनी स्टिफनेस हो गई. It means there is some factor which is responsible for the stiffness. If there is a stiffness, there is inflammation. So this means there is something. So very clear कि inflammation start हो चुकी है already. उसी level पे जब तक कि हम GI tract की बात कर रहे हैं. So the result in hard pillar-like resemblance of the trunk of the body. For this, the Chakrapani define arm as apakva ahar, unrest is nothing but arm. So he has named the term. There is nothing undigested. There is arm only. The term he has coined. This is arm only. Mrs. which is not good. Which is something not good for the body's arm. Which is causing this dandalsak. The different etiological factors makes the residual digestive fire incapable of digesting even a small quantity of light food. It happens, it's a very practical thing. अगर आपको ये symptom आया है, अगर उसके बाद आप खिचड़ी भी खाएंगे, वो भी आपको पेट खराब करेगी. Because it's already there. आपने निकाला नहीं उसको body से. It is already there. And the absorption of such unmetabolized products is very slow. So they start absorbing slowly. Assimilation very slow. Okay? So this is when the ingested food is not digested, that is due to insufficient digestive fire. The result, unformed digestive juice is known as am. So it is tad udbhav amma adhya sama iti बुधे स्मृता, मतलब वो जो उससे उत्पत्ति हुई वो अब साम दोष हो गया जब वो दोषों के साथ इन्वॉल्व हुआ, राइट? Then these are the etiological factors. These are the very few. There are hundreds of etiological factors mentioned in Shastra for this. What can cause this arm? So these are the factors which may cause formation of arm. अभोजन मीन्स आपने खाना ही नहीं खाया, लोग बोलते हैं हम तो बहुत फास्ट रखते हैं, लाइक मी, मैं 2 दिन हफ्ते में फास्ट रखती हूँ, बट स्टिल आई एम ओबीज. व्हाई? डू यू नो व्हाई? बिकॉज आई हैव आर्म. I can say I have Shotha. It means abhojan is not good. भोजन करना है. Abhojan is not at all good. अभोजन जो खाना नहीं खाएंगे, अजीर्ण भोजन खा लिया और फिर किसी की पार्टी ऑफिस में थोड़ा फिर खा लेंगे. अजीर्ण भोजन पहले वाला पचा नहीं. विषमासन मतलब आप कभी भी खाएंगे, काल नहीं है भोजन का, पर जब आ जाएगा तब खा लेंगे. ठीक है? Then अध्यसन एसात्म्य भोजन, these are like पशु वतु भुज्यते भी लिखा है कि जानवरों की तरह खाएंगे. जानवरों की तरह समोसा आ गया पसंद है तो खाएंगे, बस खाएंगे. We will not think about what is going inside. So see, these are the common causes of this. So likewise, vihar means the lifestyle modifications factor. So it is viharaj, then the mansik, the mental factors which cause Isha. If you have jealous with your colleague, then you may suffer from arm. So don't keep any jealous. Okay? Then if you have fear, then you may have arm. If you are angry, then also you may suffer from arm. So these are the mental factors. So this is about the arm lakshan. So before framing a research question, we must know about what is arm. In general, what is this term, what symptoms there are present due to this term, and what may be the other symptoms, prodromal symptoms as well as other systematic symptoms in the body. So, I have categorized these symptoms into 3. There is another category, 4th, I have not categorized here. That is very important, that is related with the gut microbiome only. So there is another a big lead for the research. So I have just categorized the generalized symptom form, what it can be. Generalized means something which is in the whole body, means the inflammatory markers present in every cell. You can think about this, what it can be, what it can be. It may be at cellular level. अगर cellular level पे तभी पूरी body में होगा, so it can be. The मुखगत आम लक्षण means anything which is present in oral cavity. मुखगत oral cavity only. Don't go beyond this. Then आमाशयगत just थोड़ा उससे नीचे आ जाओ. आमाशयगत means we can talk about the intestine, we can talk about the esophagus, we can talk about the stomach, we can talk about the small intestine, right? Then कोष्ठगत and lakshan. कोष्ठगत means we can talk about the second part of small intestine to large intestine. So first symptom is स्थिमित्य, rigidity. अभी बताया दंडाल्सक. If you feel rigidity, so what factor is responsible for the rigidity in the body? You may think about this. हम तो बोलेंगे आम है, आप साइंस में बोलो अब क्या है ये? What is the factor which is responsible for the rigidity, for the stiffness? So this is sthimitya, stambh. That is almost the same term, right? So anything which is, the thing is, the person is not able to move, so immovable, rigid, fixed. Then kati prasth grahe. Kati prasth grahe, then what does it mean? Means the stiffness, grahe is stiffness. Kati prasth, kati and prasth matlab back. So back में अगर stiffness है complete उसमें, so it may be a symptom of this. So you can think about this. If you are aware with the term ankylosing spondylosis, अगर किसी ने सुना है ये disease सुना है, so ankylosing spondylosis में जो inflammatory markers responsible होते हैं, they are due to this arm. Okay, so think about this. So then Darbelya, loss of strength. Why loss of strength? Because the food is not assimilated completely. That's why. It is loss of strength will be there. Of course, because undigested food is there. अगर बोर हो रहे तो बोल देना. I can stop any time. If you are not getting or if you are feeling bored, then you can say, okay. Then, why I have mentioned one symptom on one slide? Do you know this? This is just to trigger your mind, just to think about this. That see, this is the beauty of Ayurveda, how beautifully they have mentioned the theory of pro-inflammatory markers and inflammatory markers. So see, we have to think about this. Then, loss of physical strength, of course, because there is no proper assimilation of food. Gaurav, heaviness, I just told, Shiro Gaurav hai, sar mein bharipan hai, पेट में भारीपन है. Then heaviness in the body parts and heaviness in the body due to arm. Then Shiro Gaurav, heaviness in the head region. Then alasya, why alasya? Why alasya? No interest to work. Can anybody tell why alasya is there? खाना तो खाया था पूरा हमने, फिर भी आलस्य हो गया. Because there is no proper assimilation, so we don't get the macro and micronutrients. We don't get the energy from the assimilated food. That's why it is alasya. There is no energy, there is no way to work. Okay? Then, tandra. Tandra is fatigue, exhaustion. We feel fatigue because we don't have any energy. Then jrimbha. Have you ever observed your colleague, your teacher or anybody जो हर 2 मिनट में जम्भाई लेते बैठे बैठे ऑफिस में, क्लास में, और यहाँ भी 1-2 लोगों ने लिया अभी लंच किया इसलिए शायद. So yawning is a symptom of harm. See, देख लो, warning है. Okay? So excessive. Yawn करना is normal thing ना, किसी को भी नींद के लिए जम्भाई आ सकती है, नहीं सोया रात को तो it doesn't mean कि उसे आम है. But अगर continuous yawning है, in spite of taking a good meal, in spite of taking a good sleep, if you are suffering from this symptom, so it's a warning. Then clum and glani. It's the same thing, exhaustion, fatigue of the body, then depression of mind, debility or sickness. Continuous से chronic हो गया, शाम के बाद आम, मतलब पहले आम था, फिर शाम हो गया, दोष भी हो गए, धातु भी इन्वॉल्व हो गई, तो टिश्यू लेवल पे आ गया. So what you can do? So the symptoms were going on aggravating. Angmad, crushing pain, pressing pain and kneading pain. These are very important things. See, the crushing pain, something is crushing inside the body. If you feel this symptom, not in an acute condition, not in a renal colic, right? Not in intestinal obstruction. Remember it. If we feel this symptom, crushing pain, without any reason, without any disease, so it's a warning thing for this. Crushing means something is, what do you understand by crushing? Something is breaking inside. Something is getting into small particles. Is that so? With the force. That may be a crushing. It may cause pain. It may cause a pressing feeling. Then it may be a kneading. Kneading, everybody must be aware about this term. So this is important. This is a theme. Shof. I took only one word, arm. ये छोड़ दिया सर के लिए. Next speakers के लिए. They will talk about shof. So see, the shof is a symptom of arm. Means if there is no arm, there is no shof. So means every inflammation inside the body is due to arm. That is important. Now you get the lead. Got it? What factors are responsible for any inflammatory reaction? Can they may be arm? Of course they may be arm, because it is mentioned here that Shotha is there, swelling or morbid swelling or tumor. Then Shotha Gandakshi Koot. This is a very prominent feature. If you want to, we are the physicians basically, we are the clinicians. So how we diagnose that arm is present or not? If somebody has Akshi Koot Shotha, means Akshi Koot means the area surrounding the eye. This is Akshi Koot Shotha. If somebody has this swelling, so first we think about this arm, presence of. Second we think about the renal disease. First we think about arm only, that there may be a presence of arm. So see, when it comes, Akshi Koot Shotha, generally in a renal disorder, so it means there are some factors which are causing renal disorders. So you can think about this. वो भी आप सोच सकते हो जब रिसर्च में जा रहे हो कि what are the factors responsible for chronic renal failure? What are the factors which are causing this pathway responsible for this disease? And what is the pathway? So we can think about this. Then jwar. So then if jwar is there, means it is a good symptom, it's a good thing. If we have fever, it is a positive sign that our body is reacting against the antigen, isn't it? So it is very clear that this arm is behaving like an antigen, isn't it? तभी ज्वर हुआ ना, when it get into systemic circulation, जब आप cellular level पे आ गए, जब आप धातु level पे बात कर रहे हो, तब आप बोल रहे हो कि fever है, ज्वर है. So it means something which may be antigen, which is acting like antigen. So can it be like a protein like structure? What are the antigens basically? It can be anything. It can be anything. So out of anything, there is one thing, which is arm. Okay? So penis, the chronic rhinitis and sinusitis. Why I am telling all these? So that you can get a lead, that from where I can start. You can just take a lead from the pathogenesis of chronic rhinitis. You can take a lead from the pathogenesis of CKD. Okay? So it's a symptom of, these are the general symptoms. When the arm is in a complete body. This is the arthi. This is about the not knowing pleasure of dissatisfaction. Of course, if we have arm, we don't get energy, we have no interest, we feel fatigue, then obviously we may feel earthy, no interest, lack of interest. We don't want to work. Okay? Then भ्रम. So भ्रम is, एक वो भ्रम है कि मुझे ऐसा भ्रम हुआ, वो वाला भ्रम नहीं है ये दूसरा वाला भ्रम है. मुझे ऐसा भ्रम हुआ कि आज मेरी लॉटरी खुल गई, हाँ, वो वाला भ्रम नहीं. That is a delusion. So see, the भ्रम here is a turning round, then revolving, circular motion or rotation. Why it will happen? एक छोटे से आम ने इतना बड़ा symptom कर दिया. This is a very serious symptom if somebody is feeling vertigo and giddiness. Do you know why this is? So see, we are eating. The food is also digesting, right? But we are not getting whatever we need to work, isn't it? So half metabolite मिला ना हमें तो, हमें वो proper nutrition ही नहीं मिला. So it is due to weakness. It may be a symptom of anemia. You don't get enough iron, so that may be, vertigo may be present. You don't get enough vitamins from the food you eat, so you may suffer from this. So you can think about this. Duodenum के level पे पहुँच जाओ. So you can think about this. कि अगर ये आया है, so there is some pathogenesis at the level of duodenum, which may be responsible for this arm, and we can search out arm there, right? Then मूर्छा अति हो गई अब तो, इतने दिन से आम है, कुछ जा नहीं रहे बॉडी में, तो ultimately it will be happen. So this is increased desire to eat spicy and dry food. Why it will happen after this? See, these are the grading, severity of the symptoms. See, पहले बोला आलस्य है, फिर बोला चक्कर भी आ रहा है, फिर बोला काम नहीं करेंगे, गिर भी सकते, और फिर लास्ट में बोला कि ये सब हो रहा है लेकिन हमारा मन कर रहा है कुछ स्पाइसी खाने का. So it is a pikka, we called in medical term, pikka, to eat like this. Yeah, 2 minutes left. Thank you, sir. So this is about this. Then feeling mentally very low and stressed due to this arm. Then these are the category of Mukhgat arm lakshan. So what symptoms we can get here? This is again the bad taste of mouth. So you can study about the oral microbiome. You may get the lead this. Then dryness of mouth. Again oral microbiome. Then Nishtivana las, excessive salivation. Again oral microbiome, because particularly the local symptoms of arm is mentioned here. So Mukhlep, this is again the unguent or ointment plaster like feeling. Then loss of appetite, of course, there is excessive salivation, no interest in food, so loss of appetite may be. Avipak, indigestion due to loss of digestive fire. Agni nahi hai, वापस वही आगे, Agni nahi hai तो indigestion hai. Then excessive sour belching, because the food is not mixed well, the food is not digested well. So then Antardaha, burning sensation inside, it may be due to the presence of excessive pith, means when arm is linked with pith, it get associated with pith. Then feeling excessive nausea, the cough dosh presence is there. Then abdominal pain, the dull abdominal pain, not the acute abdomen. Acute abdomen is something different. Then heaviness in your abdomen all the time. And this is a third category, Koshtgat, means we talk about the intestine, the markers which may be present. You can just think about those markers after observing these symptoms, right? Cause sound within the abdominal cavity, then obstructive or constipation. So we will talk about this gut microbiome. We can say Kosht means this. Then passing excessive loose stool, that it may again be due to gut microbiome, unhealthy. Then arm mal pariksha. So this is about the how we can evaluate the arm pariksha. So it is in detail mentioned in Ayurveda. If you want to know, you can know. It needs a, so this is about the best physician who can be. So here, शास्त्रित सतर्क साधना नाम मीन्स शास्त्र में जो लिखा है वो हमने डिलीवर किया. वैद्य समूह में बैठ के आपकी जो आपका डाउट है, शंका है, वो आप पूछ सकते हैं. किससे? आयुर्वेद वाले आदमी से. देन ऑब्वियसली जब हम ये करेंगे तो हमारा नॉलेज एनहेंस होगा. देन वी मे गेट सम गुड रिसर्च क्वेश्चन. सो दिस इज अबाउट ऑल. बिकॉज 2 मिनट है तो ये सब मैंने लिखा है. Slide will be shared with all. How we can go further for the studies, it is already mentioned here. So thank you all. Sir, 2 minutes ही बना.
Thank you very much, Dr. Babita.
Thank you.
We are in a session 2 of this conference. The chairperson of this session is Dr. Professor Kashyap. Kumar Dubey from School of Biotechnology, JNU, and Dr. Jitendra Varsania from CBPAX. Now we have next speaker, Dr. Professor Rupesh Chaturvedi, School of Biotechnology, JNU Delhi. We request speakers to limit this talk to the 20 minutes. So we have 20 minutes for each speaker.
Just go from here. Okay, so good afternoon. This is a session after lunch. And you guys heard about, importance of digestion. If it's not well digested, whether it's a lecture or it's a food, it's going to give you a sleep. So just try to keep yourself awake and try to grab whatever you can, and then we are here to answer you. Today I'm going to talk about, very a different kind of subject, which is, if you look at the title of my talk, is Epistemology and Molecules of Ayurveda. So, I come from, my background as molecular biologist. I'm professor in the School of Biotechnology. My interaction with Ayurveda is with my different Ayurvedic physician friends and the people who are working in this area. But I was very curious to know what the Ayurveda is really all about. One thing which has dawned upon me is the epistemology of Ayurveda. And if you think about how epistemology is synthesized, it's based on the observation, which you just saw, which Dr. Babita showed you, and it's a heavily observation-based. And then what she told you, there are some inferences. She made some inferences. And then she did the reasoning. And then on top of that, there is a philosophical insight. And that's how the epistemology of Ayurveda is synthesized. To understand Ayurveda, these things are very critical, and we need to understand it. And especially when we were trying to understand Ayurveda in a context of modern knowledge, which we have. It integrates empirical practices with the spiritual wisdom. Understand the health has a dynamic balance between the individual and the universe. And this morning you saw Professor Jaya Kumar showed very nicely that how the environment is built, individual to the environment, and there was a sync between that, and that's the integration, or empirical practices are integrated, and then it based on the sensory experiences, logical reasoning, authoritative testimony, and the philosophical insight. And I will just talk about more about this in a bit. If you look at the core principle, which is a foundational sources, are called pramanas. There's a different type of a pramanas, pratyaksh, which you can feel, perception, anumana, where you can do the inferences, then aptavachana, where somebody has said something. Like you publish something in some paper and you are talking about, that's the aptavachan. Sushrut and Charak, they have described the wisdom and the knowledge, that's the aptavachan. Not only that, but all the different type of literature which is available is part of that aptavachana. And then the yukti. And this framework gives us a flexibility to reason, to create and the next is the upma, which is analogy. So if you look at the foundational source, which is the pramanas, it gives us enough framework so that we can think about, we can reason. Sometimes when think about, Agni, how you can think about that Agni is gut microbiome. It's an analogy. It's an inferences. So we are using a core principle of Ayurveda where, pramanas are the part. So extrapolating our knowledge from what is in Ayurveda to the modern is going to come into the part of yukti and upmana, which will be supported by your anumana, pratyaksha, perception interferences, and then also utilizing authoritative testimony. Then ontology, which is a theory of realities, we will talk about that. Then holistic and the empirical approaches, integration of a philosophy. This is a pretty interesting part, which when I learn about the Ayurveda, this is the thing which appeals me most. Integration of the philosophy, Sankhya, cosmology, Purush and Prakriti. So once you will start reading those things, so in Ayurveda, what it calls is, what is your body is, that's what the cosmology is. So universe and body there is a correlation between that. So it's a pretty philosophically pretty strong foundation this knowledge system has. And then the validation through practice and efficacies, spiritual and the ethical dimensions. If I look at the pramanas, there is a direct perception, observation through sensory organs. It's like heartbeat. Heartbeat is your perception because you are listening it. Nadi gyan. When I talk to my physician friends in Ayurveda, they tell me like what they really listen when they were doing the Nari Pariksha. Those are the sensory perception. And that is a key in diagnosing and understanding the symptoms. Then Anumana, inferences, which is a logical reasoning based on the observed data, help in predicting disease progression and treatment outcome. Authoritative testimony, which help us to understand some contextual connotation of any disease. And then Yukti, logical reasoning, synthesis of data from multiple sources used to design a personalized treatment strategy. This frameworks provides a methodological approach to understand traditional knowledges and ultimately will lead us to the drug discovery, personalized medicine, and holistic medicine, because it's allowing us to use the knowledge or wisdom which is available in Ayurveda can be utilized using inferences, authoritative testimony, direct perception, logical reasoning to conclude something, connotate something, and that will be pretty useful. Then ontology of Ayurveda, which is a core principle, which depends on the Panch Mahabhuta, 5 elements, you have heard about it. Tridosha, which is a Vat, Pitta, and Kapha, balance, imbalance, how it creates. Prakrit, which is extreme body constituents, and then Agni and Ama. So, these are the things which makes the core principle. So when we were trying to do taking it to the, trying to understand something in a context of modern medicine, these things need to be taken into the consideration when we talk about this. And how we are going to integrate with the modern medicine, there are some approaches we can take. Systems biology, chronic disease management, and the mind-body medicine. And then these are going to be integrated into the modern medicine, and these are the points where we are interacting with the modern medicine. This is something you saw this morning. Neil showed you that how from abhyakt to vyakt and matra mahabhut creates a prakrit. And then any imbalance into tridosha causes a disease. I added a couple of more things in there, which is the cycle of Agni, Ama, and Shotha could be a main integrator which can define the, a disease. And I call it the grand unification theory of disease. These three, with all other things combined, we can definitely will be able to explain many of the things which might be happening as either disease progression, disease etiology, or disease manifestation. And I think in the morning you saw this one also, how the healthy person is defined in Ayurveda. It's a sum, it's a homeostasis of doshas, Agni, Dhatu, Mal, Prasanna, Atma, Indriya. That's called the swasth. And any disturbance in that equilibrium or homeostasis will cause a disease. So Ayurveda does not define a disease, it defines the swasth. Anything deviation from swasth is the disease manifestation. Interesting part is if you look at the Soth, how the Soth is defined. This is defined by in a Madhavanidan chapter 41. The one thing which is pretty interesting here, if you look at, one thing is they all define that what is the Soth and what form of the Soth is there. I think they talk about 6 different form of Sothas. One is Agantuj, Nij. So those part are there in a Soth. But one thing which was very interesting is when they talk about Soth being unripe, ripening, and ripe. And if you look at the continuum of inflammation, initiation of inflammation, exacerbation of inflammation, and then resolution of inflammation. Are they talking same thing? And this is where epistemological principle allows me to reason out. Is it possible? So this is what we are trying to do. Interestingly, if you look at rheumatoid arthritis, Madhav Nidan defines Viruddha Ahar Chestasya. It's not a compatible food, not compatible behavior. And then if you look at Karot Saruzam Sotham Yatra Dosha Prapadyate, Soth is the one of the part. So that makes it pretty interesting to understand how this etiologically it's embedded into the disease progression. So this morning you saw Neil presented our study, what we have done, and he showed the data with metabolomics. I'm just showing you data with RNA transcriptomics. And this is the data coming from PBMCs, means it's from your peripheral, for their peripheral blood, monocytes. And what we found, for each Prakrit, what we see, the RNA transcriptome landscape is it showed a pattern. Means there is a similarity between a same type of a person with the same Prakriti. And you can see here that how it's beautifully distributed, this pattern is. We also used each transcriptome analysis as a single cell, and we used a single cell pipeline for the analysis, and you can see we can see that it's segregated in 10 different clusters. These are some markers which we looked at, and you can see that there is a big difference in terms of the immune cell type, and especially if you look at the 9, which is a Vat Pit Dwandaj Prakrit people, they have a low level of a CD4, CD3E, CCR7, but CD68 high. Shows that these people might have a higher level of innate immune response compared to adaptive immune responses. So just to give you some background, PBMC constitutes your major cell type which is involved in immune cells. It's a complex network of cells, molecules that work together to defend our body. The key components are T helper cells, and they play a crucial role in immune regulation. It's responsible for coordinating a very precise immune response. If you have an innate immune response, it's like more non-specific. Then you go to the T cell. It's a more precise and very coordinated response for any different type of antigen. And they could be characterized as a Th1, Th2, Th17, and T regulatory, depending on the nature of those subpopulation. But what is most important? Important is accurately classifying how the T helper cells is, and that will help us to really understand a given disease. It's a very plastic. T cells has really a lot of plasticity. If you look at how the mature DC presents any antigen to naive CD4 cells, it's activated. Depending on what kind of cytokine malleole is there, it can be activated as a Th1, Th2, Th17 T regulatory. It's a very plastic in terms of lot of canonical, non-canonical pathways are operating there. There's a redundatory pathways are involved. So sometime you will see without this malleole, they can activate STAT4 and then activate a TBET, which is a marker for Th1. Sometime without any activation of a STAT4, you can have a TBET expression. Sometime without even TBET expression, you can have an interferon gamma IL-12 and IL-27 expression, which is a hallmark of the Th1, showing that great level of plasticity. How we measure it is state of the art. T cell phenotyping, either you do the cytoph or you use the flow cytometry, and they both are antibody dependent, so it has a limitation how many you can look into that at one time. But there is another way you can do it. You can do the single cell transcriptome analysis or TCR repertoire analysis, and you can find out what type of a T cells are there. The which is in the right side is expensive, cannot be utilized all the time, whereas the cytotoph and the flow cytometry has a limitation of how many markers they can show. So the way what we did is, in our lab, we got the RNA-seq data from PBMCs, and those RNA-seq was collected by activating the PBMCs. And by the way, this data is not from my lab, it's coming from a public database. We are just analyzing it. We use the 298 samples where different classes are, the samples are there. We just looked at if there is any pattern. You can see that there is a pattern just based on their RNA-seq data. Then what we did, we used some AI ML magic, and we tried to predict just based on different type of, just based on a whole transcriptome analysis, what kind of subtype will be there. So this is called support vector machine classification, and you can see here that we were able to predict what type of dominant subtype is there just based on the RNA sequence. And you can see the accuracy for training data set, validation data set, and test data set is pretty high. But that's not good enough. We want to know what is their relative contribution in terms of each subset in there. When you do the flow cytometry or other techniques, you just know what is there, but their relative contribution is not known. So to do this, two of my postgraduate students, they worked on this. They have created this model. They said, instead of using a support vector machine classifiers, we should use the regression to give a certain weightages to certain markers, and then try to predict their amount or their contribution, relative contribution in any given situation. So these are some methodologies have been used where we gave the weighted mean expression. You select the markers, then you assign the weight depending on their significance or expression. Then you calculate the weighted mean expression for each subset. Then you compute with the total your expression, and then you look at the compute the percent contribution. This is why this is important, because it provides the biological significance. These are some markers which we selected based on classifiers as well as based on our literature, and you can see that these are some markers, and then we provided them some weight. And now utilizing this data set, we started doing a subset proportion prediction models. And you can see here that it was able to predict, train, and then predict in a very accurate manner by looking at the R square. And then how good this model is working, it depends on the two factors, which is the MSC and the MAE, and you can see that for the training, test, and validation data set, the MAE and MSC is low, showing that this model is working pretty accurately for not only training, but also for test and the validation data set. So what is the use of it? If you look at the rheumatoid arthritis, now all these numbers which you see in a sample across the data set, it's each sample from rheumatoid arthritis patient, and you can see that we are able to, for each patient, we are able to find out the relative contribution of TH1, TH17, and T regulatory. And then this is the one which is predicted, showing that the majority of them patients were showing TH17 and TH1 are T regulatory response. Interesting part is when we just looked at the PBMCs. One will expect that there will not be, if it's not activated, non-infected, apparently healthy person, you would not see a TH1, TH17, T-reg, TH2 response in a majority, because you are not expecting, because these things happens in either some kind of a disease or some type of a infection. But when we looked at the subpopulation in a PBMCs, what we did, we just took the PBMCs. These are the some data we got from outside. They have taken the PBMCs and they just expanded it by activating a CD3, CD28 and using IL-2 for the clonal expansion. So when the clonal expansion happens, you can see whatever the inherent T subpopulation is there, it's going to go proliferate, and you can see that each individual has a different type of preponderance for having a different type of a T helper cell. And you can see that 3 people showed the TH1, another is a T regulatory. You see that there is a 6 people who has predominantly T regulatory, TH2, TH17. So this is a kind of thing which gives us an idea that each individual PBMC, even at the basal level, is very different. Contribution is very different. So the response to disease is going to be very different. But we are taking it in a different way. We are going to use this model for a different purpose. If you look at a concept of, in Ayurveda, there is, for any formulation, there is a concept of Ras Panchaka, which depends on the Ras, Guna, Virya, Vibhaka, and then Dosha Ghanta. Can we use these parameters? Can we use this information, knowledge, to really find out a formulation which is being used? What is the active compound? What are those compounds which are really acting at? So this is a work done by our Ayurveda biology student. What in their class they did, I had some data from Tulsi, where mass spec was done. We got a number of compounds there. Which one to look at? There was 1,000 compounds. And even if we look at the high abundance, there is like 100 compounds there. So then we went and thought about, okay, fine, let's look at what the Nighantus are talking about the Ras Panchaka. And we just started with one thing, which was Rasa, where it's a Katu, Tiksh, and Kas. It's almost everything is like a Katu. And so if you look at most of the formulation, all the Nighantus talk about Katu. So when it's a Katu, so it means, is it possible that, and look, this is again, I'm doing the reasoning, which is allowed by the Pramanas, which is epistemologically I'm right when I'm practicing my Ayurvedic knowledge. So now so we started looking at, okay, fine. See if we have compounds which are Katu and then Madhu, and see how they are targeting a certain pathways, and especially in a T subset differentiation and polarization. Tulsi, if you look at, it has been shown majorly in a disease where TH2 dependent disease is prominent, like a skin and others. So they started looking at the TH2 differentiation pathway. If you look at that pathway, which, so if you see the TH2, which is here, GATA3, STAT5, STAT6. So they started looking at, doing some docking experiment where they looked at how these different molecules, which has a bitter test, binds. And if there is a Madhu test molecules, how it binds to the STAT5. And you can see that there is a good difference in terms of their docking score. This is a very preliminary data. This is a kind of a data we are trying to integrate. Now, next thing what we are trying to do is, the model which we have created, AI/ML model, we are planning to integrate this Ras Panchaka attribute into our model to create, so that we can simulate what kind of different molecules can regulate the relative contribution of different type of T cells. So this is how we were trying to do. This is how we are trying to, I will not say cannibalize the Ayurvedic concept into the modern, but trying to synergize it and get the better resolution or get the better idea. And these are the markers we were trying to include in there. In the end, what is the goal? Goal is creating an intelligent medicine which utilizes the inherent property of our body and makes us healthy. And that's the goal of our research. Thank you for listening me. I will be more than happy to answer any question if you have. Thank you. Yeah, sure. So this is not a cell type. This is a PBMC. So what we did, we had from each prakriti. I think you in the morning you missed that. Neil showed we have done studies where we collected a PBMCs from different prakriti people from 7 different parts, and we did the RNA sequencing for those people. Now what we are doing when I said single cell analysis, we considered individual RNA landscape as a single cell. And then we utilize the single cell analysis. TS70, yes, that's right, yeah. So now this is what we were trying to do. Yeah. Now we are trying to take that and create a relative contribution and from inherent T cell population. So the students are working in the lab right now. They are going to take a PBMCs. They will just activate it and polarize it, and then they will try to see what is the inherent component is and how much we are forcing it to polarize. So Prakriti people are most of them apparently healthy, yeah. We can still see the difference. Yeah. You can see this. This is just a. Not yet, ma'am. This is what we are planning to do next. Yeah. So I think that's the one thing we need to do. So this is just to create a baseline. Now we are going to move on to and look into different, disease, and then what kind of a dosh is there. Can we utilize these markers to identify a dosh? Based on the Prakriti inferences there, yeah. We also worked on the T cell polarization by using different milieu. And then see the effect of Neospora polycylia on which kind of cell type it is actually showing anti-proliferative. And we saw both TS70 as well as Treg depending on the milieu. But if it is naive, it doesn't actually have anti-proliferative effects. So that's true, yeah. So that's the thing. If you have and that's why the data is which you saw was the public data available where they took a PBMCs, engaged CD3, CD28, and expanded using IL-2. So they are whatever the inherent clonal population is there, it's just got expanded, and that's what we were trying to do. First expand, look at the inherent population, and then try to proliferate it. Yeah.
So anybody else?
Yeah.
Sir, my question is, during sampling you have collected sample only across the India. You have not taken the other part of the globe. Why? There is any specific region? Because you have only shown in your earlier speaker, they also showed, he also showed the data sampling of, sampling is from the across the India only, not other part of the globe, means other country or other continent.
Yeah, so it's a
because there is a huge amount of genetic diversity across the continent.
So the problem is problem for us is Prakriti assessment has not been validated into the other part. So the basis for this study is our ability to, do the Prakriti assessment.
Even sir, in country, suppose it seemed to be like a randomization of the population because you have taken the sample from the different region, and it is different geographical region, ecological, even ecosystem wise, food habit wise, too much diversity, disparities.
So that's good.
Yeah.
You know, that's good.
So here
so
yeah.
that's very good, because you know, we were showing that
Yeah.
Prakriti is something which is a keystone kind of a thing,
So,
rather
sir,
than getting affected by
but,
your food, geographical location and anything.
But why you are biased for the Andaman? Andaman, because it is a special zone of our country. Andaman, because tribal communities, now government of India, they are protecting the tribal communities.
Yeah.
What do you not think about that region?
No,
Because
but you
it is
can-
also very important.
No,
Yeah.
every
This is
community.
very special geographical
No,
location.
it's a very, you know, specific community we are talking about.
Yeah.
What we are trying to do,
Yeah.
we need to understand this
Yeah.
thing. We are trying to look at some generalized hypothesis rather than being very specific.
Okay.
If you talk about Andaman indigenous people,
Yeah.
okay,
Yeah.
they are going to be differently evolved.
Yeah.
Now the question is, before we doing these studies,
Yeah.
we need to validate the way the Prakriti is being assessed.
Yeah.
First, we need to validate in that population.
Yes.
What the validation we have for Prakriti is the generalized population.
Yes.
So for a specific population, we need to do that first, then we can do
So,
these
but
studies.
earlier speaker also mentioned there is no need of the validation in Ayurveda.
Yeah, so still I'm saying it's
Yeah.
it's not a validation.
Yeah.
So you are getting confused.
Yeah.
When I tell a validation,
Yeah.
when we were talking about validation, validation is not that Prakriti is something using an NMR we are trying to validate.
Yeah.
What I'm saying validation is validation of a principle of Ayurveda.
Okay.
So first we need to validate the principle.
Yes.
Does the Prakriti assessment, what we were doing here, is it good for those people, or it need to be tweaked?
Okay.
If you look at the ECG, okay,
Yes.
there's a generalized ECG.
Yes.
And then there's a certain community which shows always a higher ST elevation.
Yes.
So if we will look at the generalized one and then use that,
Yes.
we are going to say that they are diseased,
Yes.
but they are not.
Yes.
So each and every technique, whether it's a molecular biology, it's a modern medicine technique, or these traditional, they need to be validated
Yes.
into the particular circumstances. Once it's done, then this is the second leg of work. It's not the first leg of work. Okay?
Okay. Thank you very much, sir.
Yeah. So thank you so much, Professor Rupesh Chaturvedi, for nice talk and elaborated much on how this Ayurveda is going to be connected with modern medicine. And the validation is very much required, what he said in the last. So now next speaker in the session is Dr. Bhavna Prashar, Principal Scientist from CSIR Ayurgenomics Unit, Trisutra, CSIR IGIB, Delhi. So I welcome you, madam, for your talk.
Thank you very much for the invitation and also the introduction. Is it coming? Yeah. So I just thought, Dr. Rupesh, I just went through a small surgery and I was not sure whether I'll be able to make to it. But Dr. Rupesh said, even if you just come and talk about for some time whatever work you have done, even if you don't want to use the slides, it's also fine. But I think it helps sometimes, you know, to have the slides so that even if, yeah, I'll do that. I can do it from here also, but for the pointer, I'll use this. So as you all know, most of the audience is perhaps Ayurveda, but I also am happy to learn that a lot of molecular biology students are also there. And the kind of description that Dr. Rupesh gave about the kind of evidence and the methodologies that are followed in generation of knowledge as well as generation of evidence, I would say that culmination of all of it if you were to ask, they not only did the observations, inferences, reasoning, but they gave a three-way cause and effect relationship between external causes, internal causes, clinical features, and corresponding medicine. So there's a three-way cause and effect relationship, which is called as Three Sutra Ayurveda. So having done large-scale experiments, large-scale observations, and making the theory out of it, they have been able to give us this beautiful framework of Three Sutra Ayurveda. And I'll just take you through more details of it, that for the individual aspects, there are individual specific parameters of description, but there is an interconnectedness between all of them through a common organizing principle called Three Dosha, which is Vata, Pitta, and Kapha. So causes can be diet related, lifestyle related, environmental stress, many things. Medicines can also be cleansing medicines or oral medicines or many mechanisms of, but they all talk to each other through a common language called Tridosha. So now there was a question whether we need validation. It's not about the question of validation, but what we talk, this whole gamut of Three Sutra Ayurveda in terms of a common language called Tridosha. Likewise, so many a times the Aushad related descriptions or modern researches have happened, but those modern researches have explained the effect of those medicines in modern mechanisms as to whether it lowers down sugar or whether it lowers down glycosylated hemoglobin or whether it does this or it does that on the modern medicine related biological mechanisms of disease. But here entire description of the similar kind is available in terms of its effect on dosha, dhatu, mala. So if we were to explain this whole thing to modern science, or where is the bridge required? Why is the bridge required? Because individually these things can be discussed or detailed analysis can be done. But if the crosstalk needs to be understood in the modern scientific language, like we have Vata, Pitta, Kapha as our biomolecules, similarly modern science also has biomolecules, receptors, pathways, signaling pathways. So where is the convergence? Do we really worry about that? And if what is the benefit of having that convergence? We will go through that with the example of the studies that we have done at our lab in CSIR's Institute of Genomics and Integrative Biology. So why we came together, first of all? Because the collaboration happens only if both sides have to gain something, both sides have some questions to ask, and at the end of the whole process, either side is benefited and they feel that their efforts have, their questions are getting addressed. So we all know that normal disease, pre-disease, disease onset, these are the stages of health to disease spectrum or a transition. But even though we explain it in detail at every step, is there a way to predict a priori as to who will progress to what kind of a disease, who will progress to complications if the disease is got? There are some people who they have sugar for like 25 years, but they do not progress. There are people who immediately progress to next stages. So is there a way to, that was something which was bothering, it is even bothering now to the modern scientists. And during the human genome sequence get published, people were thinking that we will find answers in the genetic markers. So whether we can find genetic markers corresponding to differential disease susceptibility or differential drug response, is there a way to? So there are many approaches being identified to address that question. And but the clear thing was that predictive markers for early prevention and personalized approach in medicine is required. So, and many efforts globally were going on, catalog of human genetic variations and HapMap project, and then subsequently many such initiatives have happened just to catalog genetic variations. Still, cataloging is the only thing we are talking about. Then comes the association as to this genetic variation is associated with this disease or this drug response or this kind of an environmental response. That's called phenotype to genotype relation. Or there were approaches, later on the approaches were PHEVAS approach, where a set of genotypes were tested for multiple phenotypes. So there is another approach which is being taken is for extreme phenotype, that having this phenotype and not having this phenotype then just compare what is the difference. So there are multiple newer and newer approaches and newer initiatives globally that were happening. That happened. I'm talking about 2002 when we thought of this. That time only human genome sequence was out, and subsequently HapMap project happened, and then GWAS also came up. First of all, genome-wide variations were catalogued, and then genome-wide variations were then thought to be associated with certain kind of health outcomes. So where do we stand there? The question that was posed to us, and especially I took long period to have a discussions to how to start this Ayurgenomics. So on one hand, we have a Tri-Sutra Ayurveda, which has a clinical application of predictive medicine, preventive, personalized, but is there a research application? Can we have newer discoveries of biomarkers? Now the question from modern side was, can we bridge these two disciplines to identify some markers for disease prediction as well as disease drug response and disease progression? But why should I be worried about that? Because that was their question that they were asking. My question was that whether Vata Pitta Kapha have a molecular basis, whether Vata Pitta Kapha can be explained in the modern scientific language, and whether the Vata Pitta Kapha can have a explanation in the genetics as well. So now you have a question from both the sides, and both the sides want to address it. Now if I were to do Vata Pitta Kapha at a molecular level, what is my model? Is there any petri dish in which I can put Vata Pitta Kapha and check? There is no other way. So one of the ways was that Prakriti, because naturally people of different Prakriti have a higher expression of respective dosha, which means they become a real kind of a window into Vata Pitta Kapha's physiology. So that was and also not only that they are having Vata Pitta Kapha high in their physiological expression, but they are also described to have differential susceptibility to certain diseases. So there is a premise that these people, if identified, can have higher probability of getting into some disease or the other, which means they are already having a disease susceptibility. So analyzing their genetic and molecular patterns can kind of heighten our chances of getting the disease predictive markers. So this is how the Tridosha framework works. Tridosha is a common link between Tri-Sutra, and in a homeostatic state it is called Prakriti, it is healthy. And in the same, if they get perturbed, they become disease. So which means here is a thing in Ayurveda which connects biology with medicine. It connects health with the disease as well as therapeutics. So it was good to go further with this. But the natural models of dosha are Prakriti. And that's why Prakriti has been described in Ayurveda also in multiple contexts, especially in the clinical context always. Many times people feel that Prakriti is a basic principle and it's just a, it's a fundamental principle. But this is an example of a fundamental principle always described in Ayurveda in clinical context. May it be for the selection of the drugs, may it be for the progression or prediction of the progression of the disease, or may it be for the recommendations related to healthy lifestyle living. And why is it so? Because they are patterned in physiology. They are patterned right from the time of developmental times, and they have been very well described to have a genetic basis. And this is also another description. This is the third point which actually coincides with the modern scientific literature, is that they also talk about genetics being contributed by ethnicity, geography, familial characters. Everything comes as inherited. At the same time, there are certain things which influence the overall expression of that Prakriti, and that's what Ayurveda describes as many factors that contribute to development of Prakriti. Shukra Shonit Prakriti, Kal Garbhashay Prakriti, Mathur Ahar Vihar Prakriti, and Mahabhut Vikar Prakriti. That means that they are also talking about some kind of a population genomics, some kind of a environmental influences in terms of epigenetic changes. So that way also there is a perfect example of Prakriti being a concept which coincides with the modern genomic and molecular biology. So we asked the question, is there any way we can bridge Ayurveda with modern medicine? Number one, for predictive marker discovery, as the original question was, can we find modern biological and molecular correlates of Tridosha and Prakriti? And can we also then further move on to develop new medicine or molecular mechanisms of the medicines explained? So first thing that we did is deciphering Prakriti from population genomics to molecular patterns. We identified normal, healthy, working in the population, get different Prakriti individuals and analyze there. So PBMC, as Dr. Rupesh was mentioning, that's the only accessible tissue we can take to identify the genetic and molecular differences. So subsequently, we also went on from genetic and molecular to physiological patterns, the cellular patterns, to identify patterns across different levels of organization in biology, if the footprints of Prakriti are visible at every stage. So for that, we developed a questionnaire to first of all identify Prakriti of individuals coming straight from Charaka, Sushruta, Vagbhata. Again here, just wanted to point out for the molecular biology students that here the description of each phenotype of Prakriti is also linked to biology. It's not that, okay, these are the 10, 15 features, consider them as Vat. These are the 50 features, consider them as Pitta. No. Why those features arise? There is something in biology going on because of the higher expression of those biological mechanisms, this feature arises. So it's a pretty much collection of, every feature has a tag along with it, whether it is because of certain dhatu or certain dosha. So it's a complete biology-based medicine, we can say. There's a huge potential for deciphering biological variability using these approaches. So we converted it into an assessment tool, very much like a clinical assessment tool. It never says anywhere this is Vat, Pitta and Kapha, because it otherwise generates biasness amongst the observers as well as even the responders. So, and then combined occurrence of certain features only allows you to, put a label whether this is Vat or Pitta or Kapha. So Prakriti ascertainment is based on the combinatorial occurrence of phenotypic features that are captured through the questionnaire. And then, so as we were also talking about different dimensions, different contributors of Prakriti. So in science, if we want to test the effect of one thing, we have to keep every other thing constant. So the same process Ayurveda is talking about multiple factors that might contribute to individuals' Prakriti. So to be able to examine that, okay, every other factor, genetic homogeneity, so the ethnicity, geography, age group, everything remains the same, and still you identify different Prakriti individuals in that age group, in that ethnicity, of the same geoclimatic condition, then you can safely ascribe any difference to be because of the Vat, Pitta, Kapha Prakriti. And for this, we also used to do a genetic homogeneity using the markers that we had identified through Indian Genome Variation Consortium to be coming from Indo-European North population. So to ensuring it through genetic analysis as well. Having done Prakriti analysis, we identified extreme Prakriti individuals because those would be the people who will have a higher chance of expression of respective dosha across different systems. So what we first did was genetic expression, genome-wide gene expression. At that time, it was only 19,000 19,000 transcripts that we analyzed. And we also, these are healthy individuals classified by Prakriti, age group 18 to 40. So very, very narrow age group coming from the same Delhi NCR region, and also ethnicity-wise they were same. So we found that certain biochemicals and certain gene expression profiles were different. And those gene expression profiles were then looked for enrichment analysis, whether these are randomly coming some genes or these genes are enriched in certain biological processes, and whether those biological processes have got anything to do with description of Vat function or Pitta function or Kapha function. So as you can see, cell cycle transport related genes were upregulated in Vat in comparison to Pitta as well as Kapha. In Pitta, we had an innate immune response like NK cell mediated cytotoxicity and all. They were higher expressed in comparison to Vat and Kapha. and cough had a cellular biosynthesis related genes upregulated. So this told us that there is a baseline pattern, and they were also instructed to take a similar kind of food previous meal, and were abstained from taking any food or smoking, nothing before the blood was collected, so that you are also removing any effect of any immediate change in the diet or the lifestyle pattern. So then amongst this there was one gene called EGLN1, which is an oxygen sensor gene, which was differing between pith and cough at an expression level. Then we went about looking for whether this expression pattern have a stable signature at a DNA level. So we had also done DNA analysis of certain candidate genes, and there are many which differed, but one I'm discussing over here is an EGLN1. So in this, if EGLN1 is high, it actually degrades HIF1 alpha, and hypoxia inducible genes are not switched on. But if there is a EGLN1 down, hypoxia inducible factor is up, and that switches on several hypoxia inducible genes, as if there is some hypoxia going on here, we need to cope up with it. So what we found was that kapha had a higher expression of EGLN1, so low expression of hypoxia inducible genes, whereas pitta had a higher expression, lower expression of EGLN1, leading to higher expression of subsequent hypoxia inducible genes which might have happened. One of them was hemoglobin. Erythropoiesis is also high in response to hypoxia, and that we saw as a high hemoglobin level in pitta also. So this shows that not only at the expression level, but we also got a genotypic signature which would have contributed to that kind of a difference in hypoxia profile amongst these individuals. Then we wanted to see whether this genetic variation that we are talking about, whether it is only limited to this Prakriti and our population, or it is also having any impact. So now if the hypoxia gene is differentially expressed, then can we hypothesize that these people, if they were to go to high altitude, they might have a differential response to high altitude. So we wanted to check that, and for that we had another collaboration in our institute, Dr. Pasha's lab, who used to look at high altitude population as well as natives of high altitude and people who go to high altitude and they fall sick. So what we found is that those people who go to high altitude from sea level and fall sick because of high altitude pulmonary edema, their genotype was similar to that of kapha, and those who were natives of high altitude and adapted well, their genotype was like a pitta. Now again the question came whether is it only limited to Indian subcontinent. So again there is a publicly available data set of Human Genome Diversity Panel at GDP, where 52 populations genotypes data is freely available. So what we did is that, so I'm just telling this for the benefit of students, that how collaborations help at that on the course of your research, and how a publicly available data can also be availed or used to be able to potentiate your observations and hypotheses. So there we found that globally also those populations out of those 52 who were residing at high altitude, their genotype frequency was similar to pitta genotype, and those who were not residing at low altitude or were at a low altitude, there was no relation with the pitta. So this confirmed our observation that the variation that we have identified to differ between pitta and kapha, it actually has a lot of value in terms of its high altitude association as well as high altitude adverse outcomes. So this was a discovery. So again, it's not just a validation of earlier known genetic variation, but it's actually helped us uncover a novel genetic variation which was earlier never published for high altitude adaptation. So there's a potential for novel discoveries as well. So then we have subsequently looked at whether there is only hypoxia related variation, but hypoxia is also associated with thrombosis, and we observed from our data set that a thrombosis associated genetic variation in von Willebrand factor gene frequency was more in kapha and low in pitta. So the bleeding associated variation was more in pitta, and thrombosis associated was more in kapha. And then we saw that whether this is having any combinatorial presence. So combination of T allele of EGLN1 and ancestral VWF was high in kapha group, which might confer them more susceptibility to hypoxia-associated thrombotic outcomes. So now, so that's about the variability. Now you captured the variability at a genetic level amongst different Prakriti individuals, but since ours is a 3-sutra Ayurveda, can we do something about it? Whether we can alter that kind of a probability. So that those are the kind of things one can take further. But one of the things that just wanted to show for the Ayurvedic, that whatever observations we make, does it map back to our Ayurvedic descriptions? Because Ayurveda also says that Vat, Pitta, Kapha have a different blood-related parameters, ranges, where clearly they are saying that even amongst the healthy individuals, there's a prolonged bleeding time possible for Pitta individuals, whereas for Kapha, the blood is more of a thick nature. So, that tells that they will have a respective disease susceptibilities, and that is why they have been advised to take a different kind of dietary precautions, even in the native place, but if they travel anywhere else, they have to be more cautious about it. So this is what is the summary of the work. Now, another collaboration that we ended up doing is that EGLN1, hypoxia sensor gene, responsible for high altitude adaptation. But is it only relevant in high altitude? What happens if we are all the time living in sea level? We never get exposed to high altitude. But hypoxia being a very core kind of a biological phenomena, can we have any other place, any other disease where hypoxia can modify the disease condition? So we actually collaborated with another colleague of ours, Dr. Anurag Agarwal, who was working on a mouse model of asthma. And we asked him to tweak this gene in the mouse model of allergic asthma, and we found that if EGLN1 was to go beyond, was to be inhibited beyond a limit, and if HIF1 alpha gets exaggerated quite a lot, then it actually exacerbates asthma so much that it becomes severe and non-responsive to even steroids. So now here we have a biological system or a mouse model where we have modified hypoxia, and now the test was whether we can alter it, whether we can modify it. So that's what the question that we asked, whether we can, so if Pitta imbalance is showing hypoxia differences, and if that is also modulating asthma, can there be a Pitta modifying or a balancing medicine which can alter asthma severity? And we selected Adatoda vasica as one of the plant, which is Pitta Shamak. It is called again Tiktharas, as Dr. Rupesh was telling, that Tiktharas is there dominantly, and Tiktharas is commonly hitting both Pitta and Kapha, so it actually does not let any one of them get exaggerated. In fact, it balances both of them. So we use that rigorous examination on asthma phenotypes at tissue level, at phenotype level, at molecular level. And we also did a transcriptomic profile of lungs of the mice who were treated with Adatoda vasica. And we found many known pathways being hit of the asthma, but we also found hypoxia-induced mitochondrial, so hypoxia-induced changes were also altered. We've subsequently validated whether this particular Adatoda vasica is also hitting HIF1 alpha in particular, not just the pathway. So for that we did a cell line model to induce hypoxia chemically and then see if the effects of hypoxia are altered even in the cell line by Adatoda vasica whole extract. And then subsequently we also showed that hypoxia induces mitochondrial morphodysfunction, and whether hypoxia, if HIF1 alpha is being targeted by Adatoda, whether it is also affecting the subsequent downstream phenotypic variations. And we have shown through seahorse assay and GFP tagged all that pathway to show that mitochondrial morphofunction is also benefited by a use of Adatoda. So this is what I just wanted to tell, that we started from phenotype or a lakshana to identify people of contrasting dosha prakriti types. We identified one of the molecular marker kind of for the Vat Pitta Kapha, and then subjected it to the medicinal alteration to see that HIF1 alpha axis is also kind of a targeted by the Pitta Kapha modifying thing. So with this, I think in the interest of time, I'll stop here. Just wanted to say that we are, since we are working from more than like 2 decades now, and students are here, they should know that importance of collaboration. So lots of collaborations from different diverse disciplines. To begin with, I and Mitali started in the mentorship of Dr. Brahmachari, but we expanded to multiple disciplines. We also run a PhD course on Ayurgenomics in ACSIR since 2014. And this is our lab, many of you might have seen. And these are our team of people and funding. So with that, I thank you all for your kind attention, and if you have any questions.
Nice talk. So anybody have any question?
See if the student has any question.
Namaste, ma'am. Ma'am, I have a question that जब हम लोग जीन की बात करें या कोई भी बायोमार्कर को अगर एज अ कैंडिडेट हम लोग सेलेक्ट करना चाहते हैं, तो हमारी बॉडी में जैसे ह्यूमन बॉडी में क्या होता है कि प्लेयोट्रोपिक जीन होते हैं, और इवन इफ कोई और बायोमार्कर भी ले तो मल्टीपल पाथवेज के थ्रू वो फीडबैक मैकेनिज्म से वो कंट्रोल होते हैं. तो आखिरकार जब हम लोग यहाँ पे जैसे फिनोटाइप पे हम लोग लिंक करके देखना चाहते हैं, तो हम लोग कैसे कैंडिडेट सिलेक्शन करेंगे हमारे जीन ऑफ इंटरेस्ट का या बायोमार्कर ऑफ इंटरेस्ट का?
So, एक ऐसे कैंडिडेट जीन ऑफ इंटरेस्ट ढूंढना या किसी भी किसी भी आयुर्वेदा के कॉन्सेप्ट के लिए, चाहे दोष हो या कुछ और हो या धातु हो, तो उसके कॉन्सेप्ट उस पर्टिकुलर फंडामेंटल बायोलॉजिकल एंटिटी जो आयुर्वेद में डिस्क्राइब्ड है, उसके लिए बायोमोलिक्यूलर बायोमार्कर ढूंढना. सबसे पहले तो हमारा बेस जो है वो डिस्क्रिप्शन जो आयुर्वेद में है ना, और उसके बाद फिर हम आजकल तो इतना ज़्यादा लिटरेचर भी अवेलेबल है कि यू कैन एक्चुअली लुक फॉर लॉट ऑफ थिंग्स कॉरस्पॉन्डिंग टू दैट डिस्क्रिप्शन इन द मॉडर्न लिटरेचर, क्योंकि बहुत सारा रिसोर्स भी अवेलेबल है. बट फाइनल टेस्ट ऑफ द पुडिंग इज इन ईटिंग, राइट? सो यू हैव टू एक्चुअली आइडेंटिफाई पीपल विद डाइवर्स प्रोफाइल्स विद रिस्पेक्ट टू मे बी दोषा और एनीथिंग एल्स दैट यू आर टेस्टिंग. सो उनको लेके, उनका सैंपल लेके, जैसे बताया भाई आप एक्चुअली करके दिखाइए आरएन, जैसे हमने भी दिखाया 2008 में पब्लिश किया आरएन ट्रांसक्रिप्टोमिक डेटा. सो ऐसे ही आज भी जैसे अगर हम बोलते हैं कि इम्यून प्रोफाइलिंग करके देखें, जैसे सर ने दिखाया कि CD38, CD4, CD8A वगैरह सारे उन्होंने सेल्स प्रोफाइल्स करके देखे. सो एक्चुअली पहले कुछ हाइपोथेसिस बिल्ड करने के लिए यू हैव टू डू लॉट ऑफ लिटरेचर वर्क, एंड योर सबस्ट्रेटम शुड बी योर आयुर्वेदिक डिस्क्रिप्शन on which you can map the modern literature, but ultimately you have to show it through doing it by clinically analyzing, because Ayurveda का कोई भी biological concept ऐसा नहीं है जिसका कोई clinical manifestation नहीं है. उसका clinical manifestation है ही है. तो आप उस clinical manifestation के base पे लोगों को identify करो कि कौन contrasting manifestation show करते हैं, तब फिर उनमें जैसे हम case control करते हैं ना, healthy versus any given disease, तो वो भी तो एक कॉन्ट्रास्टिंग एक तरह का हो गया. ऐसे ही उस फिनोटाइप के लिए जिसमें वो फिनोटाइप है, जिसमें वो फिनोटाइप नहीं है. बस आयुर्वेद में ये है कि कोई एक ही फिनोटाइप नहीं है कि भाई बाल काले हैं या नहीं है, या स्किन ऐसी है या नहीं है. देयर इज अ कलेक्शन ऑफ फिनोटाइप्स विच फाइनली टेल्स द स्कोर ऑफ दैट पर्टिकुलर बायोलॉजिकल एंटिटी इनसाइड द बॉडी. सो आई थिंक फिनोटाइप टू बायोलॉजी वाला जो लिंक है वो आपको एक्सप्लोर करना पड़ेगा, तब आप मॉलिक्यूलर कोरिलेट्स ढूंढ पाएंगे.
Ma'am, I think decade-long experience and then your work, it's pretty nice work. My question is regarding the EGL1. So EGL1 has as a hyperbaric hypoxia, and then when you did the in an animal model, I don't think so. That's a hypoxic.
No, so
hyperbaric hypoxia.
So what happens in the hypobaric hypoxia is that EGLN1 goes down, so that HIF1 comes up, it stays for a long, and then HIF1 response genes. So that's a molecular phenotype of environmental hypoxia. What happens in environmental hypoxia at molecular level is this. Now, if we were to mimic that in the laboratory, by actually depressing EGLN1 by using siRNA or the chemical inhibitors, and we are producing high HIF, which is the condition similar to hypoxia.
So it will be interesting to look into the into the cancer patients where
who have hypoxia. Yes, absolutely.
So there is a hypoxia, and how the EGL1 might be playing a role.
Absolutely.
And depending on a different dosha, because if you look at the literature, there is some evidence that certain group of people are prone for
Absolutely.
cancer.
So one of the work that we followed up after this was we actually developed the lymphoblastoid cell lines of the extreme prakriti individuals to look at dosha in cellular level. And because our idea was this, that if there is a HIF high or HIF low, and if I want to test something or subsequent effects of that, I cannot do it in a human being, so we can generate the cell lines of those individuals. So we actually find that HIF1 alpha to be, at the normal level also high in pitta individuals. And then we have subjected them to more chamber-based hypoxia to see if different prakriti individuals would respond, different prakriti cell lines would differently, respond to that hypoxia. So that one thesis is also done and paper is in bioarchive in case you are interested. And the premise was that for certain individuals, because HIF1 being high in cancer cells is like a established thing. You can, and there's a tricky thing also that if you do it too much low, then also it is not very good for cancer, and if it is high, it induces angiogenesis and it survives the cancer. And whereas some people have been advised to undergo hypoxia preconditioning before they actually take up the chemotherapy or anything else. So there's a set of people who require that, not everyone, but nobody can predict it a priori as to who would be requiring it. Maybe there are now methods of doing personalized medicine based on the sections of cancer tissue as well. But that's the most, kind of a immediate application one can think of differential hypoxic response. Same way, different cell proliferation response is also observed in, so as I showed about the gene expression pattern, we have seen it in the cell lines also, that Vat, Pitta, Kapha, Prakriti have a baseline patterns of cell proliferation, like a doubling time is also different, even without any stimulus.
Thank you so much for a nice talk. So more discussion, you can take questions during the tea time. So I think now tea time.
Yeah. Thank you. We thank all the speakers, and we invite chair and co-chairperson Professor Kashyap Dubey and Dr. Ditendra Varsakia to give the memento to Dr. Babita Yadav. Dr. Babita Yadav. Oh, sorry. It was Professor Rupesh Chaturvedi. And Dr. Bhavna Prashar. With this, we end this session. Yes. And then we also invite Professor Rana Rupesh to give the memento to chair and the co-chairperson. Thank you everyone for this session. We have a very short tea break and we'll meet again at 4 o'clock. Hope to see you soon on time. all to the session 3. We have 4 talks in the session, and we have Chair Dr. Puneet Mishra, Professor of Community Medicine, AIIMS Delhi, co-chair by Professor Bharat Bhoyar from CB PACS. So we'd like you to invite the speaker.
So, good afternoon, everyone. Now we are going to start this session, which is session, I think last session before valedictory. So, may I invite Dr. Jitendra Varsakia, who is assistant professor at CB PACS, for his talk.
Hello everyone, dignitaries of the dais, respected chairperson and co-chairperson, respected professor and his colleague, and dear students. Without wasting much time, I am directly going to my talk. Ayurveda concept of the arm in context of disease and modern medicine. Important to know the subject. Arm is a key concept in the Ayurvedic understanding of physiological, pathological and therapeutical process. So we can elaborate arm in a 5 manner, in a simple term. Whatever classical description are very deep in a Sanskrit language, but I try to elaborate these things. First stage is impaired digestion and metabolism due to the factors like poor diet, stress, environmental factors. The digestive and metabolic process can be hampered by these etiological factors. Then after the formation of arm is represent by toxins, by toxins is present, unprocessed or improperly digested food leads to the creation of arm, which is toxic and uneliminated part of the body. वो eliminate नहीं हो सकता है या तो digest नहीं हो सकता है, इसकी वजह से वो channels में रहता है. Then after that it accumulate in the body, arm accumulate in the body because it cannot be neutralized or eliminated by the body. That's why. Then it travel to the systemic small channels. We can say in our classics like strothas, small opening channels are there. And then it manifests in a symptoms form like indigestion, like imbalance of the body, etc. The concept of Agni and arm. According to classics, Dehagni, Jatragni, this is the digestive fire we can say. The reason for these things we can assess by Ayu, means lifespan, age, Varna, color, Bal, strength and immunity, Swasthya, health, energy, Utsah, Upchai, bulk, Oj, immunity, and Tej, complexion. These things are very, very useful to assess the Agni in a classical way, Ayurveda. Let me discuss ahead. The proper maintenance of the Agni helps a person to live longer, and its impairment gives rise to disease. अगर आपका digestive fire ठीक है, तो आपकी जो body की mechanisms है, metabolism है, वो systematic चलेगी. If the digestive fire is hampered, अगर आपको भूख नहीं लग रही है या excessive भूख लग रही है, तो this thing is lead to your disease condition. Anyway. Therefore, Jatragni is considered to be key regulator of the most important sustaining factor of the living being. Improper dietary habits like adhyasan means एक खाना खाया और दूसरा तुरंत ले लिया. विस्मासन, खाने के टाइमिंग फिक्स ना होना. मॉर्निंग में लिया 7 बजे, शाम को लिया 5 बजे, रात को लिया 11 बजे. This thing the Vismasan. And wrong behavior pattern like the Vegdaran, suppression of the urges. If you have urge of the urine, you hold it and not go there for. If you have urge for the motion, this whole thing, suppression of the urges, also vitiated the dosas, 3 dosas mentioned in the classics, and it leads to the impairment of the Agni. In Ayurveda, it is being believed that Agnimantiya is the root cause of all diseases like diabetes, obesity, autoimmune disorders, etc., etc. Agni partially digested, due to the disturbance of the Agni, the digestive food will be partially digestion or not properly digestion. That's why Mandagni and our Agnibal lead to the pathogenesis of arm pradosaj vyadhi, which disease leads by the arm. When arm mixed with the dosas, dhatus and malas, this is the key component of the Ayurveda principles. It develops the certain complex adverse reaction which is basis of the disease. There are two states of dos dhatu mala. There is a sam state and there is a niram state. When the dosas collaborate with the arm, this is the state of the sam condition, and then it comes to the niram condition. Ayurvedic text explains the sam and niram on the basis of sign and symptoms of the mala pariksha. We have unique method of the mala pariksha, sam and niram. The symptoms of the mala pariksha is also given in a classics, which can be benefit to we take as a biomarker of our classical terms. Factors. Which factors can be hampered your digestion system? The classic says aharaj means dietary factor, lifestyle factor, etiological factors for the vitiation of the different dosas. Vitiated kapha dosa, mainly cough, combined with the vata and leads to the development of samprapti of the different diseases. Among the measure of the aharaj, dietary factor is the main, because we depend on the diet. Whatever we think do in a difference in the diet. Once the sharir is abolished, there is a quotation in our classics, all things keep inside, first focus in your body. आपको अपने बॉडी के टाइमिंग, मेटाबॉलिज्म, जो भी टाइमिंग है सब फिक्स करने हैं. In the tradition of the Ayurveda, arm has been defined from three viewpoint. We can elaborate this arm condition in 3 points. This perspective represents the different aspect of the arm, shedding light on its formation effect and nature. Here is the elaboration in a 3 manner. First one is the arm as a product of impaired digestion, byproduct of impaired digestion. This is the gastrointestinal viewpoint. From the digestive perspective, arm is primarily seen as an undigested or partially digested food that cannot be properly processed by the digestive system because of the low level of Agni or low level of the digestive fire. When the digestive fire is weak, food is not broke down or not properly digested. This results in a formation of the ama, which considered as the toxin or waste product of the body. And this improperly processed food is called am. This undigested food cannot be absorbed or eliminated properly, causing a building up toxic material that circulate by the small opening channels like strotas, which given in our text. It settled in various tissues, organs, leading to the digestive disturbance and create different symptoms like bloating and discomfort, all these things. Second one is arm as an imbalance in a metabolism, metabolic viewpoint. Arm is not limited to just a GI tract. It extends to the body's metabolic function. Wherever the unexpelled product or toxins travel through the channels by the strotas, wherever they go, they create a hamperness there. From this viewpoint, it represents the systemic imbalance in the body, body's metabolism pathway. The arm produces here is a more systemic in nature. It does not only result from poor digestion, but it also break down in the body's ability to convert food and energy into the healthy, usable material. This leads to the accumulation of the toxins in a body, which contribute to the fatigue, low level of the energy, and different type of the chronic diseases. Third one is the arm as a manifestation of the imbalance in the entire system. It is the holistic viewpoint of the arm. From a holistic perspective, arm is seen as a manifestation in imbalance of a whole body system, affecting the both physical and emotional state of the body. The viewpoint emphasizes that arm is not just production of the digestion or metabolism, but it byproduct of overall systemic imbalance, which includes the emotional, psychological, and environmental factor also. This aspect of the arm leads to its spread across various tissues, organs, impacting not only physical health but also emotional well-being it too. As a result of individual may experience disconnection between the mind, body and spirit of our human being, leading to a symptoms like, now the symptoms is physical and mental both are there, like anxiety, depression and other chronic symptoms according to disease also is created. Now the interconnection between the three viewpoints. The three viewpoints, digestive, metabolism and holistic approach, represent the different angle of the SAM concept of the arm. This is the SAM concept. When one of these systems, digestion, metabolism and oral body of the health is disrupted, it can lead to the production of the arm. The totality of the arm can be understood only by considering these three perspectives together or it interact with it and fit to each other. In Ayurveda, it is essential to address the arm from all three perspectives to restore the balance and prevent the disease, lead to the treatment aspect also. So improving the digestion to reduce the formation of arm, first one. Second one is the regulating a metabolism through diet, lifestyle and herbal remedies. And third one is addressing the emotional and environmental factor that contribute to systemic imbalance. So I'm also represent the transition from the physiology to pathology. Now, what role in the physiology to pathology in arm? The role of the arm in Ayurveda from physiology to pathology in disease condition is fundamental concept describing the accumulation of the undigested or improper processed metabolic waste which act as a toxic substance in body. It is often referred as a root cause of many diseases arising from impaired digestion like Agni dysfunction, which lead to systemic dysfunction also. First one is the arm in physiological formation. So arm is produced when the Agni is weak, means digestive fire is weak, resulting in incomplete digestive food that leads to accumulation of the undigested food in the stomach and intestine. Second one is poor nutritional assimilation, that weakening the bodily tissues, which we can say the dhatus. Production of the metabolic toxins which circulate in the body all over and it go to the holistic level. Factor leading to the arm formation, so many factors are mentioned in our classics and practically also we can see that there is a weak digestion due to the low digestive fire, overeating or irregular eating habits, consumption of the heavy, oily and incompatible foods like viruddhahar, stress, sedentary lifestyles, lack of physical activities and suppression of the urges like Vegdhara. Now coming to the pathological role of the arm in disease condition. Once the arm accumulates, it begins to obstruct channels, trothas, leading to the various pathological conditions, and it is the starting point of the santrapti, means disease progression, disease formation process in the classics. There is the 6 stages given in our classics for the disease formation. There is the Sat Kriya Kal. There is one more fundamental principle which involves the disease formation process, Sat Kriya Kal. First one is the Sanchay, accumulation. Initially built up of undigested waste in a GI tract, it is a Sanchay. Then it Prakop, aggravation. Arm starts spreading to all over the tissues like dhatus. Third one is the Prasar, dissemination. Moves to the circulatory channels, blocking bodily channels. Third one is the sansrai, means deposition. Arm settled in the weak tissues or organs. Last one, fifth one is the vyakti, manifestation. Symptoms of the disease become apparent. Now the symptoms arise. Last one is the bed. The complication stage is there. Chronic disease stage with severe systemic effect. So this is two concept of pathology to physiology, the arm how react. Therefore, it is the precursor of the inflammatory, degenerative and infective process that eventually manifested in a full-fledged disease. That arm may also include the infective process it implied in a certain description mentioned in our classics like. So the same things mentioned in our classics like that improperly or undigested food which is located in Amasai. That's why our stomach, we call stomach in a Sanskrit language Amasai. This is the sthan of the arm. A fundamental principle in Ayurveda is that by tackling the arm in an early stage, progression of the disease can be hampered or arrested at that place. Now the role of arm in Ayurveda from physiology to pathology disease. This already I have discussed in previous slide. This 5 stage I have elaborated arm how work in a 5 stage. The first one is a stage involved. Arm progressively undergoes various transformation. In the process is developing to full-fledged disease. First stage is the formative stage. In this phase, arm interact with the 3 dosas, become more complex and active. In this stage, which arm form accumulate in a body, in this stage clinically symptoms hardly manifest as. Clinically symptoms present नहीं आ पाएंगे. All indigestion does not lead to the arm, but it does only when the cough is involvement. Only when cough is involved, indigestion leads to the arm. So, what about the other dosas? In case of the other dosas, when the Vata is involved, indigestion is known as the Vistabd. The results in a drying up and degenerative state. Then when Vata combined with arm, it is a state of drying up and degeneration. When Pitta is involved, this stage is Vidagdha. The stage of the leads to inflammation. This is the precursor of the inflammation. Next is the interactive phase. In this phase, arm interact with the 3 dosas and become the more complex and active. Arm interact with the Vata, arm interact with the cough and Pitta also. Arm interact with the Vata create a condition known as the Sam Vata. Arm interact with the Pitta and cough, respectively condition known as the Sam Pitta and Sam cough. Now next stage is the phase of the complication. Phase of the manifestation. This phase is a degenerative, inflammatory and infective stage. In this stage, technically this stage is known as a SAM, and which is an acute condition, we can say. 4th stage is the phase of the complication. This is the phase of the development of the arm wish, the toxins which produced by the arm. It is the stage of the crisis that even lead to the death condition also. And final one stage is the phase of the restoration. If body recover in this stage, this is the stage in which the active interaction between the arm, dosa and dhatus have resolved, and the body may recover mild, moderate and severity on the basis of the disease symptoms. The disease may also persist in a chronic phase, this known as the Niram stage of the disease. So this is the 2 concept of the arm in a Sam and Niram stage. Importance. The concept in Ayurveda that if treated in early stage, it is possible to interfere with the degenerative, inflammatory and other progressive pathologies for early prevention of the disease. There are also specific interventions which can administer in a classical way for the treatment of or manage of the arm dosa. These 3 main important things are there, depending on the severity, quantity as well as the type of the arm. A 3 prolonged approach has been advised to tackle the arm dose. First one is the Langan, second one is the Langan Pachan, and third one is the Sodhan. Out of this, Langan involves the restriction of the nutritional intake, even to the extent of a complete fasting to rest the digestive and metabolic apparatus of the body. Whatever जो भी organs या body के जो भी tissues digestion और metabolism में involve है, उसको completely rest देना. This is the Langan. Langan Pachan involves prescribing active medication also, which will neutralize and transform the arm by digestion or metabolizing it helps to render inactive. Specific medicine are to be used depending on the stage of the arm. Sodhan is a purification process which removes the arm dose from the body altogether. When used in the appropriate context, these 3 measures can help to neutralize inactiveness and eliminate of the arm from the body. Now validation and research portion of the arm. Many studies could be visualized for the experimental validation of the concept of the arm. One approach would be identify the biochemical pathway and process that underlie the degenerative and inflammatory pathogenesis in the body. So, how we can assess arm with the help of the Ayurveda classical literature. So, there is some questions I have depicted here on the basis of the classical symptoms given for the arm and Agni. First one is the do you notice coating over the tongue? Second one is do you have a indigestion? Third one is the do you have a adverse dislike to food? Do you have a constipation? Do you have a water or loosely stool pass? Does your stool have a pungent smell? Does your stool sticky? Does your stool sink in water? This is the mal pariksha, sinking water. Does you feel tired or lethargy? Do you feel sleepy the whole day? This is also one symptom of alasya given in a previous lecture. Are you suffering from a headache? Do you have a pain in your joints, multiple joints? Do you feel the feverish whole day? And last one is the, do you suffer from body ache? So this is the symptoms of the arm. On the basis of this, we can assess the arm and we can take as a biomarker of the Ayurveda classical point of view. Agni assessment. The effect of the Agni will be assessed by a 6 parameter. This one, the Ayu, Varna, Bala, Swasthya, Utsa and Upchai. These 6 parameter we can elaborate in 18 different questions, the grade 1, 2 and 3. On the basis of that, we can assess the Agni, which I have depicted here. Now the experimental validation of the arm. How we can validate arm as a modern point of view and our Ayurveda point of view. Validating the concept of the arm through experimental means involving studying in the context of modern biomedical sciences. This would like to be a complex task as the arm is not directly translatable into specific measurable entity or biomarkers in the way that we understand disease today. However, I am trying to elaborate this thing here. First one is the biomarkers of incomplete digestion. So, modern research could aim to identify biomarkers of incomplete digestion or the presence of the undigested food particles in the bloodstream, stool or urine. This could involve using a modern diagnostic tool to tackle change in a metabolic pathway or markers related to digestion. The digestive enzymes like amylase, lipase, protease, inflammatory markers like CRP, cytokinins, gut microbiota profiles which influences the digestion and immune function of the GI tract, metabolomics to study metabolites that may be produced by fermentation or breakdown of the food in the GI tract. Second one is the gut microbiome studies. Ayurveda emphasizes the importance of the gut health, which may related to the balance of the microbiome in a term of digestion and the prevention of the arm formation. Experimental studies could be focused on the gut microbiome composition and its effect on digestion and metabolic byproducts. The relationship between a dysbiosis, imbalance in a gut microbiota, and the formation of the toxins or metabolic waste that could be related to the concept of the arm by questions I have depicted in the previous slide. Third one is the immunological studies. Arm is often associated with the toxins and inflammatory state that could be lead to the disease. How undigested food or toxins trigger immune response, such autoimmune diseases or chronic inflammatory conditions. Investigating the gut permeability, also known as the leaky gut, and it connecting to the immune activation, which could mirror the process of the arm accumulation, which can do the study on this base. Intervention study. One of the primary Ayurvedic treatment of the arm is a panchkarma. That is depicted in our classics. A detoxification process or use of the specific herbal formulation, which we can implement in this scratch. Clinically, trials with the Ayurvedic treatment aim to the removing toxins or harm from the body, which we can study in a larger scale. Digestive health studies, understanding the how stress, diet, and lifestyle factor affecting the gastric and production and digestive enzyme level, we can tackle in this stage. And last one is the pharmacological validation. We can testing the effect of the herbal medicaments which depicted in our classics on markers of the digestion and markers of the metabolism and markers of the arm. Clinical trials to determine the efficacy of the Ayurveda to reduce the toxin load or improving the digestion. It can help to do the validation of this time of the Ayurveda fundamentals. So, clinical studies to assess the variations in a gut flora depending on the constitution of the individuals and how Ayurveda intervention help to normalize the gut flora, and the prevention of the formation of the arm is the another possibility in the field of the Ayurveda and contemporary stage. Validation of the concept of the arm would be a significant step in the process of improving our understanding how the Ayurveda physician diagnose, classify, and the stage of the progress of the disease and the tackling of the arm and this type of disease. We can focus on these specific therapeutic decisions and made it in a clinical practice. Thank you so much.
Am I audible? Okay, thank you, Dr. Jitendra. It was a really wonderful presentation, and I'm surprised to see that how much Ayurveda know that gut health is so important, that most of diseases they do arise from the gut. In modern medicine, now we have started talking about the gut health in connection with the other disease. But here, what he has shown that Ayurveda physicians, for centuries they were aware about the gut health, that it is of prime importance which can lead to many diseases. So now this is open for one or two questions if there are any from the audience. If not, then I'd like to invite my co-chair for his comments before moving to next presentation.
थैंक यू सर. आज का ये जो सेशन हुआ है इसमें डॉक्टर जितेंद्र वर्षाकिया जी ने जो आम का कंसेप्ट बताया है, अग्नि का कंसेप्ट दिया है, उसके अंदर जो सर ने अभी कहा कि गट हेल्थ की कितनी इम्पोर्टेंस होती है, उसके अंदर जो डिजीज़ फॉर्मेशन होता है, प्रोग्रेसिव डिजीज़ फॉर्मेशन में जो 6 फंडामेंटल हमारा जो कंसेप्ट है वो जैसे ऊंचाई प्रसर व्यक्ति भेद कितना इम्पोर्टेंट है ना, और उसके बाद में गट हेल्थ कितनी इम्पोर्टेंट है ये उन्होंने यहाँ पे दिया है. उस बारे में बहुत ही अच्छा इनका प्रेजेंटेशन था, उसके लिए मैं इनको धन्यवाद करता हूँ. और मैं धन्यवाद करता हूँ स्कूल ऑफ जितने भी कोलैबोरेशन में जो आपका आज के कंसेप्ट वो हुआ है सेमिनार वर्कशॉप, उसके लिए मैं सभी को धन्यवाद देता हूँ. थैंक यू सो मच. ओके, next speaker for this session is Dr. Shwetank Sharma, Institute of Liver and Biliary Sciences, New Delhi. I invite Dr. Shwetank for his presentation. Thank you.
Good afternoon, everybody. First, I'm not an Ayurveda expert, so my lack of knowledge may please be excused. And what we have been doing actually is some collaborative work in collaboration with CARI, Punjabi Bagh and All India Institute of Ayurveda, and that is what I'm going to present. I was asked to make a presentation at a very short notice, so whatever work we have done, I'm going to present that. And I thank Dr. Jitendra for enlightening us, at least me, with what ARM is, because without my knowledge, I was working on that actually. So it's an interesting thought. And what I'm going to just briefly talk about is the gut microbiota response to Virochana. As Dr. Jitendra just said, that Panchkarma is a very important aspect of recovering from arm abnormalities. So we have been actually trying to understand how does the gut microbiota actually change or vary when in different stages of the Panchkarma process. But there was a slight difference in our approach. instead of taking diseased individuals or people with digestive disorders, what we took were healthy individuals. And the reason was very simple. It started with the question, how do you define a healthy individual? So if I may take just 1 minute and ask anybody in the audience to define a healthy individual. We know how to define a diseased individual. How do we define a healthy individual? We have to perform a case control study. We have to capture healthy individuals. Anybody. Okay. So let me summarize it as not any apparent presentation of a disease. Right? Very good. But then, if we take this definition, it's almost perfect, but we do not know what is happening in our enteric system. We may have problems with the liver which are not progressed to a stage where they are presenting, they are in the developmental stage. We may have problems, as Dr. Jiten said, it may be at a very low level in the digestive disorder. It is not presenting itself very strongly. So, one component that is contributing to all these factors, both liver as well as digestive, is a new organ that has been defined as a new organ, and that is the gut microbiota. I'm not going into the details, but gut microbe, the number of bacterial cells, just the bacterial cells, the microbe in the gut actually constitutes bacteria, viruses, and fungi. So, but the number of bacterial cells in the human body, in the gut, in the human body, is the same as the number of cells of the human body, which means the number of human cells is same as number of bacterial cells. So with my students, I always often joke, now you decide whether you are human or bacteria, because it's actually, for me, it's a whole ecosystem that is working together. 2 equal number of cells represented in one body it's very difficult to define whether it's human or it's bacteria. But again, when it comes to the brain, the human parts takes over everything else. Anyhow, coming back to the point. So this was a study supported by CCRAS, and thanks to the team of CCRAS and Carrie and Ayya Panjabi Bag and Ayya, that we were able to complete it. We are still in the last stages of analysis of the Ayurvedic clinical parameters, which I am not going to show here because that is beyond my comprehension. Thanks to Dr. Shezi for she has been interacting with me and coaxing me to improve upon the analysis. So I'm not going to discuss too much on what Panchkarma is, you already know. But the questions that we started was that does Panchkarma have any effect on gut microbiota? And if it does, then what is the aspect of Virechan, which is very popularly used for cleansing of the body? How does it work? And the idea for Virechan, emphasis on Virechan, came from the recent findings in various studies where fasting itself was showing that it has beneficial effect on the gut microbiota, and the individuals start feeling better just by fasting. So I'm not questioning the whole process of Panchkarma, but these are two connected things. Then how are these changes in the gut microbiota actually changing the functional aspects of the bacteria? Change in the bacteria is one thing, and then functionally how is it changing is the second aspect. So the primary objective was to determine the effect of the purgation therapy, that is Virechan, and the gut microbiota profile, and then correlate it with the nutritional, biochemical, and general well-being of the individuals. So it was a single-arm, open-label trial with a sample size of 60. Now, if you see at the bottom, there is written that we'll take 75 and with about 15% as dropout or excluded. I'll come to this. And of course, this slide lists the various parameters that we were investigating. From at our end at ILBS, we did not analyze any Ayurvedic parameters. We were looking at the individuals from completely a biochemical allopathic point of view. And all the Ayurvedic analysis was done by Carrie and All India Institute of Ayurveda. Now, one thing, again, I will keep on mentioning this again and again, that these are all healthy individuals. There was no diseased person in this, and all these individuals are undergoing the Panchkarma, the Virechan therapy and all that. So again, some additional parameters that we took, and there were other Ayurvedic screening series of analysis that was done to understand what the patient is, what that individual, we always confuse, what that individual is feeling like before the therapy, during the therapy, after the therapy, what they're eating, what is their behavior like. And the inclusion criteria, of course, was kept that these were healthy both from an Ayurvedic standpoint as well as from an allopathic standpoint. So effectively, I can assure you that just barring a few small analysis that is left, we have been able to identify how to actually determine a healthy individual, a healthy Indian individual. Whatever we have been reading in any study, I'm not questioning the Ayurvedic practices, but in most of the studies, we have been following the Western literature of what a healthy individual looks like or should have the parameters. But this is the first study where we can identify, using both Ayurvedic and allopathic principles, where we identify the individual as a healthy individuals. And I'll quickly skip all this. So this is the study protocol, and the ones that are marked with the red arrows is whatever studies we were carrying out at this Institute of Liver and Biliary Sciences. This is the standard protocol that we followed for the Panchkarma at the Ayurvedic centers. These individuals were undergoing these processes at these two centers, and this was done only after they were cleared from one round at the Ayurvedic center and the second round at our center. So they were completely free of any disease, any disorders or anything. Again, the steps continued, and the arrows actually also suggest that we were collecting stool samples of these individuals at these various points. So the investigations that we took, we had a huge list of performers. This actually list of performers runs into about 55 slides, and I'm just skipping that, removed all that, and just left with 3-4 slides. So we covered all the aspects of what diet they were taking during the process, what diet they were taking after they were discharged from the center, and we took a reading of their microbiota even at day 90. That means how does the microbiota actually remain or change after the process has been completed. They have gone back to their original lifestyle. Whatever they may be eating, they can as well go to market, have golgappas, change the bacteria back to something else. Then we correlated with how long does the effect of Panchkarma actually stays on these individuals. So there's a huge long list of this. So we screened about 95 individuals. We were thinking of 75, enrolled 60 individuals, 30 coming from each of the two centers. Now, what we noticed was that instead of the 15%, that it's not dropout actually, these are not patients, so this is excluded. So of the 15% that we were thinking, there were 35% individuals who came from Ayurvedic centers to our place marked as healthy. First the Ayurvedic center was approving them, they are healthy, then they are coming to our place, we are screening them. And then what interestingly we found was that many of them have deranged LFT lipid profile, thyroid profiles, abnormal fibroscan. Fibroscan is a small sonographic assessment of the fibrosis in the liver, and ultrasound also showing fibrosis. So this actually was a very surprising fact for us also, which shows the amount of burden of the disease that we have, that many individuals have early stages of fibrosis, can be protected with lifestyle changes, even at with Ayurvedic treatment, no need to come to allopathic centers and go through expensive treatments, but they are not diagnosed. So if I may recommend, maybe a recommendation from this meeting can as well go that ultrasound and fibroscan screening may be carried out at Ayurvedic centers so that they can change or modify their practices. to benefit such individuals who are getting missed and they come at later stage. An individual is not going to the hospital unless there's a pain at 12 o'clock in the night. There is a simple fact, all the doctors know this. So if this is done, then definitely we'll have a very good healthy Indian population in future. So we carried out, collected a lot many samples at various stages of the treatment. I'll come to the microbiota part. Sorry for having to show this just after tea. So we collect the stool sample and we solubilized it, pulverized it kind of a thing, filtered it and extracted the suspension. Now, if you look at these samples that we see, I call them beautifully rainbow colored samples, but these are actually representing healthy individual samples, but with different labels of bile acids and bilirubin content in them. That is why they have different colors. And this, from my discussions from Ayurvedic experts, I know that this also represents at what physiological state that healthy individual is, because the amount of bile is increased, bilirubin is increased, or something that is getting escaped in the stool. So what we do is we take the bacteria from this, isolate the bacteria, take the DNA, and we analyze what is called as a 16S RNA. The complex structure on the left that we see is that of a rRNA, which sits in the ribosome where the protein synthesis is taking place. This part of the bacteria is actually conserved, and it does not change from one type of bacteria to another type of bacteria. So that is why we can use this to mark the bacteria and identify which species is present in the individual. Now, it has 9 regions. What we took was about a V3, V4 region, and that shows about 80 to 90% similarity compared to the whole strand. Now, this is a profile of the amount of DNA that we collected in these individuals at various stages of the Panchkarma process. Now, if you look, very interestingly, the DNA amount is about 200 nanogram per microliters in the left 2 boxes. But when it comes to Virechan, the DNA amount goes down dramatically. It was very difficult for us to capture in one instance the DNA in some samples, so we had to repeat the sampling. And that means that the bacterial load is almost vanished at the stage where the individuals are at the Virechan stage. So this was some new finding for our teams. And when if you look at the right side, post-Virechan, we see that this amount actually comes back. So bacteria actually restructure themselves, and I will show you the slides, what happens eventually. So what we found initially was that the boxes that we see in the middle, it is Deepan Pachan, DP, Virechan, Samsarjan Karm, SK, SP that is hidden behind Snepan, day 30, day 15, day 90 is the time. These are the time points at which the samples were collected. And what we see is that there is a very clear difference between the bacterial composition, the dots that you see. Now, if you take any dot on the left-hand side, any red dot on the left-hand side, and if you take any red dot on the right-hand side, it belongs to, let's say, day 15. But then day 15 individual does not have the same bacteria in the gut, even after the Virechan process, which means that bacteria in a constant state of remodulation after Virechan. Now, which bacteria are good, which bacteria are bad, needs to be worked out, but this actually indicates that Virechan process is immediately affected once the individual is released from the treating center. Now, when we compared it between different stages, what we see is that if we look at the top left, what we see is that day 0 is the baseline. When the individual first came, we collected the sample. Then Deepan Pachan and Snepan. So Deepan Pachan and Snepan are conducted consecutively during the Panchkarma process. But you look at the distance between them, they are far apart. The day 0 being somewhere in between the Deepan Pachan and Snepan, which means that the bacteria is actually moving in composition very, very dramatically when this Panchkarma process is going on, even between 2 to 3 days, there is a change in the gut microbiota. Again, I will keep on repeating this. These are all healthy individuals. There is no disease. They never had any disorder during the process. They never complained of anything. All the individuals who were enrolled completed the whole process from start to end. Now, if you look at the diversity of the microbiota, it is like, you can consider this like what kind of a forest each individual is having inside the gut. The forest of bacteria this is. Now, if you look, with the red line is actually the average of all the readings that we have, and Shannon index on the top, Simpson at the bottom, and Chow index on the right actually shows these diversity index. Shannon is basically how many types and what numbers of different types are present. Simpson is how evenly the numbers are presented. If species 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, they have all 100 counts, or one has 2 count, other has 20, other has 50. So if we look, variation is one part in each of these graphs which actually deviates and goes down very dramatically, suggesting that, again, confirming that there's a reduction in the bacterial taxa that are present there. So this slide actually shows what is the composition of these bacteria in the various stages. And if we look at the one in the middle, the VRI, so that is the variation stage. Now in this, if we see the red bar, which has suddenly increased in number, so this is relative abundance. The numbers have increased of the red bar, and the red bar actually represents a taxa called Escherichia shigella, which is a pathogenic taxa. Does that mean that when we do Panchkarma, we are activating the pathogens? Now this is the interesting part. If you follow it up, follow the red, it vanishes within the Samsarjan Kamp, the SKA and SKB process itself, it dies out. During this process, there was no complaint by these individuals of any disorder at any time, which means that because there was a change in the dietary pattern during the Panchkarma process, the bacterial population adjusted itself such that some bacteria came up, some bacteria died down. So we come up with, maybe somebody has already said it, we have now it has now become a common practice at our place, that a pathogen, these are pathogens, the Escherichia shigella, a pathogen is not a pathogen until it is pathogenic. So we have streptococcus in our gut, we have staphylococcus in our gut, we have E. coli in our gut. Every time we say E. coli is the one which is causing dysentery. Why? Because E. coli is such a species that it comes up on every culture every time. It may as well be a viral dysentery, but E. coli will always come up. And what is the percentage of E. coli in the gut? In the bacterial population, 0.1%. So we have to be, probably we have to elaborate this process of identifying or correlating bacteria with your digestive disorders, and with ARM, as Dr. Jiten said, we probably have to elaborate it and understand it in a much stronger sense. So when we look at which bacteria actually changed, we find that between day 0 and Deepan Pachan, day 0 being the baseline, on the left side, there is no difference. The gray bars actually show that there is no significant difference. But as soon as Snepan is done, there is a significant difference in the taxa that are changing. And what we see is that Lactnospiraceae UCG 004, if you see, is a beneficial bacteria, which is also now being prescribed allopathically in certain medical centers. This increases with Snepan, which is a very interesting finding which already exists, a treatment which already exists with Ayurveda system of giving. Snepan is nothing but ghee for those who are not from Ayurvedic background. Now, if we look at what happened with Virachan, we find that between baseline and Virachan, there is a lot of reduction in the good bacteria which are marked with star, and certain bacteria like Escherichia shigella, Enterobacter, they increase. Again, they are increasing, but they are not pathogenic. Whereas in Samsarjan Karma, if you look, the greens have gone down, the numbers have gone down, and the orange has increased, which means there is a sudden shift between the bacteria during the process of this, between just the two stages of Panchkarma itself. Now, if you look at the left, the day 15 graph and the baseline, what we see is that if I just go back one slide, the number of bacteria you see here, it's a 20-30 bacteria which are significantly P less than 0.05, etc. But when we look at day 15 days after being released from the hospital, there are just about 4 bacteria which show significant changes, which means this change is with the baseline. So which means that by day 15 after Panchkarma, the bacterial population becomes similar to when they were at baseline. But again, I repeat, these are healthy individuals. So if it comes back to normal, it means that this healthy individual has come back to a normal healthy state as it was. Panchkarma was a process that altered the bacteria according to the treatment that was given. But at day 90, day 30, if we see, there is no significant difference between baseline and the day 30 bacterial population. Surprise came to us at day 90. 3 months after the treatment, these individuals have significantly changed species, but these significantly changed species are only increasing. There is no one which is decreased, orange, all on the right side. And there are about 3 species which are good bacteria and they increased. So, which again goes on to tell that of course there are some immediate alterations when Panchkarma is occurring. Then there is a readjustment of the bacteria during first month, second month. But at third month, it changes into a different type of combination from the baseline when the individuals are coming. We did not follow up with these patients on what their dietary pattern was at day 90. It was like we were already having a record of initial records, but at day 90 what their pattern was. But then we suspect that this is probably because of the changed habits. Once a healthy individual undergoes Panchkarma process, feels happy, goes home, says, okay, now I'm happy, back to Golgappa and Chaat Papdi and I'm fine from anywhere else. That is again taking in additional load of additional virus, bacterial, fungi load which is altering the gut bacteria. So we then looked at how these changes actually occurred over a period of time and classified it on what is called as the linear discriminant analysis score. And what we see is Bacteroides, Bifidobacterium, Ruminococcus, Musicola, Ligilacto, all these are good bacteria. And you look at the graph on the heat map on the right side, the blue is low numbers and red is high numbers. So the good bacteria are actually increasing with the Panchkarma process. And this is a comparison between the days, and this also confirms the same fact that the bacteria eventually go on to improve. The composition actually, the Panchkarma process itself resets the bacterial composition so that it favors the growth of what are called as good bacteria. We then identified what is the metabolic change that is occurring because of these bacteria, and which bacteria are associated with these change. So we find that during at the baseline, Snepan, Virechan, Samsarjan Karma and day 90, there is a distinct combination of metabolites which are going up and which are going down, red being up, blue being down, which means that again it feeds into or gives support to the ARM concept that in ARM the metabolic factors are equally important and they are regulated by the change in the bacteria alone. Again, these are healthy individuals. Standard diet for everybody. And when we look at the PCA plot on the top left, what we see is that the metabolic profile during the Virechan process is very distinct from the metabolic profile during Snepan or Samsarjan Karma or baseline day 90. Of course, this is a transient profile. You can call it fasting or I don't know how you explain this. I'm not an expert on that. But then during that Virechan process, this is a very distinct profile. On the bottom, we have certain metabolites and the pathways which these metabolites are from and where they actually show a very strong change because of the Panchkarma process. And I would like to end here, and I'm sure you would be curious about other things. So if you have any questions, we can take that.
Thank you for the very elaborate presentation. I really enjoyed it. Just wanted to ask if the healthy individuals, you did not see any variability in the response
Response to?
To Pirigen therapy.
Variability is only in the form of the metabolites that we analyzed.
But variability with respect to stage of treatment or variability with respect to individuals?
So it's a preliminary analysis that we have done. And in that, what we have found is that the bacterial composition that we see here in few texts are changing. It is not really as simple. And there are about four types of bacterial compositions that we have at baseline alone, which means that for us at least in the allopathic center, a healthy individual can be of any four types based on the gut microbiota. And these bacteria are associated with different metabolic
profile of the
components, including dopamine. One group had increased dopamine
you saw
right at
four
baseline.
categories in the
Yes.
baseline itself.
Yes,
Yeah, that's what I just wanted to know. Because you mentioned about the health assessment that they did.
Yes.
Because Ayurveda describes not only that absence from the disease, but it describes how a healthy person should be looking like. Not like the biological definition of health, which is samadosha, samagnisha, but how a clinical profile of health would look like.
Right.
That is also given. And in that, there are some 5-6 features related to gut health and the excretory health. So we also assess health assessment using those parameters.
Correct.
But having said that, still within that there is a scope of looking at variability as per Ayurveda. Because Prakriti is of course about that only.
Right.
Second thing is I also wanted to know, was there a duration of the treatment? I mean, Virechan was administered in any particular season or any time?
No, so these patients, I'm always sticking to patients. These individuals were treated either in the springtime or during the autumn, the Sharad Ritu time. So that was the time that was identified as where generally the human body is in homeostasis. So.
Thank you, Dr. Sadank. It was really wonderful presentation, and many times people they question that different processes which we do in Ayurveda, do they have any scientific value? And here I was surprised to see like the Virechan which he has demonstrated that how does it affect the bacteria, gut microbes, which in the earlier session was described that gut health is probably one of the most important factor. So, sir, thank you for enlightening us with your presentation and showing the real-world evidence from the gut microbes after the Virechan this thing. So, these are my comments. We have taken questions. If there are any further questions, they can be taken after the tea break so that our next speaker would be Dr. Savita Bastavadi from, he is Ayurveda expert from Vedita AI. But before inviting Dr. Savita, may I request my co-chair if he have any comment for this presentation done by Dr. Shotha.
Very elaborative, knowledgeable, practical presentation made by Dr. Svetang Sharma. I congratulate CCRS and team of ILBS team of Dr. Svetang for this research study. Thank you.
we have this team of researchers, and I was a practitioner like any other till the time of COVID hits. Then the thought came, we really want to work for some bigger mission in life, to really empower students to read and understand Samhitas better, and young practitioners to exactly practice like the one experienced. You know, I understand reading Samhitas or practicing in the field of Ayurveda is very challenging. So, our AI scientist since last 5 years, even though he's into different startups in technology, he read, understood Ayurveda and created this technology of interactive books. We have put these interactive books, all Brihatrais and Lagutrais, in this website Vedita AI. So, wherein during this presentation, I'll show you how it works. It is easy to discuss the chapters and different conditions, and that takes the test also. Unlike earlier time, classical way of reading, now we should really take advantage of this artificial intelligence. And we have this patient simulator wherein while patient is explaining about the complaints or the case, so this system takes the whole study, puts it in a system and give you the auto suggestion. It is like an assistant so that you are not missing out on anything. Helps to what are the different types of investigation you might need, what are the different types of examinations you should do, and different tests you might suggest, and differential diagnosis. Of course, you need to use Yukti to come to the final conclusion, and that suggests the treatment protocols also mentioned in classic texts with reference. Because we already have these ancient texts with us, that any suggestion is also coming from is through the text. And we have that's in the pipeline, the patient the simulator, wherein you will be practicing on dummy cases so that when exactly you are in your clinic to see the real cases, you are not afraid. You already know what are the different cases you already practice, and it will be easy. So let's go to the presentation about the today's topic. It's about Ama and Shotha. This presentation explores the Ayurvedic principle of relationship between Ama and Shotha, highlighting chronic inflammation as a key factor in degenerative disease. We will also discuss how Vedita AI tools helped us in researching content of Ama and Shotha from our Samhitas. When we searched in our AI tool, that almost gave us 5,530 sutra references from different texts. You can see in Ashtanga Sangra almost 1600 plus references we found. Video we need to play. There's a video we need to play. Because that's showing the different. This is actually the video, the different references that searched and presented. It does the work which seemed to be humanly impossible, for any researcher to find in such a short period of time. From that, we took this concept of Ama and Shotha from the smart search of this Samhitas, Vedita AI tools, and case discussion with patient simulator. Again, this is a video. I just want to show how it is, how it works. This is a screen recording. We need to play this video actually. Huh?
If anyone.
So this is the Madhav Nidana from our library. Right now we have Brahatrayas and Lagutrayas there. So we can play the video. This auto playing, right? Yeah. These are the different references we got in Vedita books. It's a gold mine for researchers. Yeah, we concluded these things from there. And yeah. For example, we took this Ama Vata. This is one of the condition due to Ama. You go there and ask the question, what is Ama? This is the answer within few seconds. From there, we got this, what is armor? Like, since morning we are hearing different lectures, very beautiful presentations about arm and all. So, through Vedita, we got this information, put it here nicely. I don't have to, I think, again and again explain what is armor and all. So, we'll go to next slide. Yeah. How is armor formed and what are its characteristics? That's giving you the reference also. Where is it deriving from? The smart way of learning, understanding, researching. Yeah. So we came to this conclusion, understanding armor, definition, the formation, the characteristics and symptoms, along with those references. Next, how apakwa ahara rasa in amasya results in armor formation. This is the next question we asked. This took the reference from Charaka Samhita and describing. That will beautifully give you in a tabular form, in a flow chart. So less of effort, more productivity. Yeah, that also suggests the pathogenesis. So we were able to produce this slide from the results given. The effect on dosha. It is clearly distinguishing like between vata, pitta and kapha in a beautiful table. We can ask doubts, we can discuss, that will take the test and ask questions, and out of 10, how many you were able to answer, that's going to assess also. This is like, one aspect is you take the membership of a gym, but you don't have a coach. You have books, but you don't know what to read, how to read, you know, how to prepare yourself for exam. It is like a coach. No, I want to have bicep, I want to have, you know, particular way that's going to train you on different samhitas, different disease aspects. So then we came to this conclusion, amal, which can be can combine with other biological entities like dosha, dhatus, malas and all. Ama mixes with vata, then what happens? And with pitta, what is happening? With kapha, then different things it is able to explain from there. Smart search about with Vedita about how Srotodushti is happening. Since we have the authentic source of Samhitas, so we can 100% rely on this, unlike the ChatGPT which normally we use. Sometimes that hallucinates and give random answers. So this is the explanation about Srotodushti that was able to give the verses also with the reference and the definition. What are the defining characters of Shotha? That's searching about Shotha now from Sushrut Sutrasthan. Shotha, again, as you all know, swelling, inflammation caused by dosha imbalance. Detailed explanation about that. Now we want to know about the preliminary symptoms of Shwetu. So I want to search about that using AI assistant. Like we have different options there, ask questions, we are asking questions right now. We have the option of test knowledge, and third option is discuss also. The third one is discuss and learn. We can eat up Samhitas, ma'am. Yes, in English translation. We have translated all the Samhitas. I am going to show that now.
Okay.
All Brahatris and Lagutris we have right now in our library. Next, we are going to.
Library, this is like even PV.
It's an AI library, ma'am. Vedita library.
So PV Sharma and others have translated.
Yes, yes.
You're taken that or you're translated yourself?
Archived.
Yeah, so taken that also.
Yeah, yeah. And next project is we are translating Siddhi Nandan Mishra says all the books. That's our next project. That is now giving us the idea about Nidana of Shotha, prodromal symptoms. You all are smart people. I don't have to, teach you the disease, about the disease, but I just want to show how easy it is to learn, understand, research, and everything.
One more question I had. You
Yes, sir.
showed one Excel sheet kind of a thing. How did that come?
That research page?
Yeah, so you showed Excel sheet where there are multiple columns.
Yeah,
In the columns, cause, disease, treatment, all of that is also written. Disease is also written. So how does that
In
come?
the beginning, when
Yes,
I
yes.
said when we searched about the armor
Huh.
in all the Samhitas,
Yeah.
around 5,500
Yes.
references came.
Yeah.
That is because of AI. AI, we have integrated.
Can you show that?
Ma'am, for the interest of time, can we keep questions at
Questions
the last,
we'll
at
keep
the
it.
end of the presentation? Thank you.
Yeah. Types of Shotha in Brahatris. So that is taking reference from Charak Sutrasthan, Sushrut Chikitsa, Ashtang Hriday Nidan Sthan and all. And beautifully it is giving in a table. So types of Shotha, Ekanga, Ardhanga and all Sarvanga. So according to different Acharya, what are the types and all. Even making presentation was so easy using the AI tool, using Vedita. Sushruta Sanhitas reference it is giving about types of Shotha. As you all know, it's 6 types. Samanya Lakshana of Shotha. Common signs and symptoms: heaviness, instability, elevation, heat, thinning of veins. So, beautifully it is describing. You can ask doubts. You can eat up head like how we used to do to our professors. Till the time I am convinced, totally, It's a smart tool basically.
Your time is almost over.
Okay, so I'll try to finish faster. How we were able to treat the patient also. See, knowledge is one thing, but implementation is very, very important. So we tried to solve that problem of patient consultation, which I'll show you. I'll quickly go to the last. General pathogenesis, Samprapti Ghataka and all those. List of diseases under Shotha. Shotha as a symptom and also disease. Complications. Ama and Shotha in disease pathogenesis. Case studies. So we just as an example, we took this Grahini, that is irritable bowel syndrome, and Ama Vata, 2 cases. how we were able to put the case history and then derive the results, and whatever the medicine also it has mentioned, gave the reference from the Sanhitas, like Samprapti Ghataka also, and diagnosis, how we reached to the conclusion, and treatment plan, whatever the treatment we suggested, it is going to be very, very helpful for young practitioners, you know. And that will exactly give you the explanation why that suggested that particular medicine. For example, in Grahini it is saying Kshayaja Grahini, that Saptamrita Parpati or Mahalakshmi Vilasras, or the combination of Jatamansi and Shitopaladi. Why? You can always ask why and learn. For learning also, learning cases also, and treating the new patient also become easy now. Then this Amavata we took, and the treatment plan that suggested Amavata, Yogaraj Gugulu, 5 minutes, okay. Mahavata Vidwansan. So, one second. Yeah. This is the QR code. There are a lot of job openings in the field of research in artificial intelligence. Anybody want to be in touch with us, please do register, and also for the books. So thank you so much. I just want to show the library. Can you, this app, can I show this app from here? Can you connect here?
We are just showing the last thing, till then we can proceed for the questions. Continue with the questions.
Not a question, we are running out of time, so I'll just take the next speaker.
Quickly we have to shift on to the last speaker.
Yeah, that's what I'm asking. No question.
So thank you, Dr. Savita. Since we are running out of time, so in this session we will not going to take any questions. The questions can be asked during tea break. Now we are going to invite the last speaker, Dr. Saurabh Kumar Sharma, who is Assistant Professor at School of Computer and System Sciences, JNU, New Delhi. So Dr. Saurabh Kumar Sharma.
Are there any participants for the workshop, for the tomorrow's workshop? Can you just raise your hand? Yes. So we request you to stay in the hall after the this session, so that we can have a word with you. Okay? So thank you.
Short of time, I won't be able to show that patient consultation software and the test reports, 7-dimensional test reports, and the app. Maybe next time. Thank you.
So good evening to all of you. First of all, I would like to thank the organizer, Professor Rana Pratap sir, Professor Rupesh Chaturvedi sir, Professor Neil Sarwar Bhaves, Dr. Hemant Kushwaha ji, and other organizing committee members. And also, I would like to thank to our chair and co-chairs for this session. So this is the something different topics related to the what we are listening from morning about the Ayurveda, but it is a special, we can say the one special sector where AI can be used in the Ayurveda, and what are the opportunities, what are the different methods that currently other researcher, or we can say the scientific community, are working in the particular domain. So I will not take more time, just till 10 to 12 minutes for concluding this my talk. So when we talk about the Ayurveda or something, so first of all, the 3 or 4 terms comes in our mind or in our general conversation, that Bharat, ancient Kal, Ayurveda, disease, and then when these all are things, then the data will come, and when the data will come, then AI will come. So in a layman language, the main we can say the dependency of AI is the data. When we have a small data, when we have a general data, we cannot apply the AI. So we have to think that when the complexity of data will increase, when the large amount of data will be there, then AI will come in the picture. We cannot say generally that AI, suppose as a bioinformatician, or we can say that as a doctor, when we call the AI, so people think that this is a big word. So how AI is going to work? So just we have to understand AI, it is nothing but it is a dependent on the data. When the data, and even in the data, when the data is too high, or we can say the nowadays big data, and the complexity if there will be there in the data, then only we can apply artificial intelligence. So how it is going to work? Just next to 5 to 6 minutes. So originating in India, Ayurveda is an ancient system of medicine based on the concept of holistic healing, taking into the account of mind, body, and spirit. We have talked about the Ayurveda, then how the AI is going to be used in this particular sector, and when we talk about the Prakriti and Vikriti, so these are the some genetic, simple terms, or we can say the definition in a layman language, that what is a Prakriti and what is a Vikriti. So that we are not going, I'm not going to explain in a detail, but when we talk about the personalized medicine matters, when we talk about the particular role of AI in this particular sector, so we know that since 2003, when the completion of the Human Genome Project started, then personalized medicine came in the picture. Scientific now have started to understand correlation between the individual characteristics and health, and allowing doctors to develop a personalized medicine and treat diseases like leukemia, breast cancer, and all other things. In modern conventional medicine, doctors generally don't consider our unique body type, genetic, or characteristics when treating us. This was the, I'm talking about the 2 or 3 years before. But now the doctors and the scientists are going to start. So when we talk about the one new drug, so it will take the around 10 to 15 years, and the billions of the funds will required for that. So how AI is going to be help us in this particular sector? So the convergence of genomic and Ayurveda, AI is next frontier in medicine. Scientists are now discovering a link between the Ayurveda Prakriti classification and specific gene. This opened a fascinating opportunity to interest Ayurveda insight into the modern healthcare through AI-based personalized medicine, and also incorporate traditional holistic preventative care, including yoga and meditation. So in medical institute must do more research and data-driven study to full comprehensive and effectiveness of Ayurveda treatment in contemporary healthcare practices, and that is where AI emerged as an invaluable tool. So how our AI will be the crucial will play a crucial role, and what are the different methods when we talk about the AI? So so basically the data. Without data we cannot do anything. So collect all 3 main types of data: genomic information, Ayurvedic Prakriti classification, and healthcare record. When we have all this data, then high quality data is paramount for this exercise. Data integration and training, that's all required for the AI. So AI need, that is why we do the 70% for the testing or training. So if we have 100% data, or we can say the, suppose around 10,000 data set, then out of 10,000, 70% for the training. When we start train the data, then our model, or we can say our algorithm, that is called the AI. Because AI is nothing but it is only a source of code where we have structured in such a manner that it learns like a human being, like from a child, that we learns some language, then we start some modification that particular language, and after some time we do the influence in that particular sector. So that is why AI will work accordingly. When data will be good, when data will be fine, then only we can apply AI. And apply AI, it is nothing but it is a source of code, our algorithm, that how we have given the instruction to our application, our computer or machine, that how it is going to be adopt all the data. And based on that, then we do the 20% testing. Then whatever it learns, test is going to be good or not. And then 10% for the final result. So these are the right-hand side, we can say the data collection, data pre-processing. Pre-processing data is so important. We cannot apply AI or machine learning directly on the data. We have to pre-process the data, that this data is doable or not. Then next stage is feature extraction, where we apply the different classification, segmentation for extract information. For example, like identification of the breast cancer. Then data integration. Then after that, there is a predicting ML modeling, then model validation, and individual treatment, and then last one is the continuous learning. So by this way, the process will start. So the data is going to be feed, trained, segmentation, all. Then the tool will apply. Tool means algorithm will apply. Then final we will get the result, and then we will compare that what this is our expected outcome or not. If not, then we will again go and check our methodology and the data. So based on that, whatever we have done, this is our work. So we have developed a database that is called a traditional medicine database, and we are focusing only on the northeastern region of the India. So where we have collected the data, and these data are basically consist with the phytoconstituent, with the disease where it will be cured, and the gene with the gene information, so that it will be help for the research work. So what is the data distribution? So 945 botanical information, including taxonomic, graphical, and therapeutic uses by specific communities. 93 whole genomic sequence of plant till now we have updated, and still it is going on. Phytoconstituent 150 ratio 40775 means out of 40,000 we have done 150, and still it is going on. Database development and modification is going on, and we are going to, since this is a project is funded by the DST, with the help of DST officials, we are going to open for a public domain, so that anyone can take the data and use, or we can say do the research based on that. So how it is going to be basically this database work? So explore the world of medical plants. So this is all the information that we have received till now, and the important part is that this is the secondary database that are basically already registered in different papers, or we can say the different literatures. But the important part that I am going to mention here, that the ethnic information, or we can say the ethnic group that is practicing in this particular region, so that audio, video, and the language, like when we go in the Manipur, then there is some language that will be converted into Hindi as well as in English. So we want that their information should be safe, and the community that are going to be in danger, or the plant which is very important for the medicinal purpose that are going to be in danger, we can basically save that particular society and community. So by this way, we are working here. So, so we have also developed a tool that is called the genomic analysis of plant through algorithm. So that is where we apply the AI, since the different plants from the different region have also some mutations, and which phytoconstituent and which DNA, which sequence, nucleotide sequence is going to be changed, that we have also developed a tool so that from North India, from South India, from East or West, is there any mutation within the plant is possible or not. So we found that from the one region to second region in the same plant, there will be a mutation. So what will be the advantage for mutation? If there is we found a mutation, so it will be, when we do the bioinformatic analysis, then it will be helpful for identifying the new drugs. So these are the some data, because I have already explained you the data is more important. So from the Ayush Ministry, these are the data that they have till now we have on our public available database. So based on that, now what we have worked, so we have started work 2 years, 3 years before for the breast cancer. So we have started with two process. First one is through genomic, and second one is through image form. So when we have started through the genomic, so these are the process where we have done the bioinformatic studies and found the top 5 gene involved in the breast cancer, and that is for triple negative breast cancer. And the right hand side, that is for the image. So we have taken the histogram data, MRI data, then we have applied the tool, some algorithm, and we have enhanced the precision technique that are identification for the early detection of breast cancer. That was published in just one month before, I think in this month also, that is published in Health Information Science and System, that is a Springer Nature journal. And in the same way, we have done a brain tumor using the optimized deep learning methods, and also in the breast cancer for the drug discovery. So this is the part of the AI, but how it is useful with the Ayurveda. So we have identified the 2 and 3 plants that we are herbs, we can say, and that we are doing the bioinformatic study, then we will go for the animal trial. And when we talk about all things research part, then budget and other opportunities important. So thanks to the Government of India that they have increased the Ayush Ministry budget from last year to this year, 14% morely increased. And when we talk about the budget, then what is the market value? Is any industry is going to accommodate with this particular research or not? Is there any market value? What are the global value? So these are the, we can say that these are the open market, and I think it will be very useful for upcoming days for Ayurveda, for Ayush Ministry, and for the scientists who are working in this field. So my sincere acknowledgement for GIC Hub, IIT Bombay, DST, and Department of Health and Research for funding this work, and our team members, Professor R.K. Brojan Singh, Dr. Nidhi Verma, and Professor Neil Sarwar Bhaves. They have tremendously working very hard and giving all the inputs and suggestions for completion of this project. Thank you so much.
Thank you, Dr. Saurabh, for giving insight about the AI and how we can use in Ayurveda. And we were amazed to see your work on the brain tumor as well as in the breast cancer, and hope your research in the area of Ayurveda will further improve this practice of Ayurveda and its understanding in the, not only in the country but all over the world. So with this word, I give it to my co-chair if he have any comment before we conclude this session.
Thank you, Dr. Saurabh.
Now
Okay,
we
next.
request chair and co-chairperson to give the momentum to the speakers. Dr. Saurav Kumar, so we request you to give the momentum to the speakers. They'll give to you. Dr. Siva Batsuri. Dr. Savita Bastavari. Dr. Jitendra Valsakya. Dr. Jitendra Vasakia. Dr. Shwetang Sharma. And we are very thankful to the chair and the co-chairperson. We also invite Dr. Hemant for giving the momentum to chair and co-chairperson. There will be a very short tea break again. We are again apologize for this delay. The valedictory session will start after 6:15. So let's have a tea break.
Yeah, we'll start the session at by 6:15, but 6:10 we'll request you to be here. The workshop participants who are there may kindly be seated.
Hello all. Since we are in the end of this remarkable conference, we welcome you to the valedictory session. First we invite Dr. Y.K. Gupta to the dais, and also Dr. Ramesh Vishanti to the dais, and Dr. Rana Pratap to the dais. Dr. Y.K. Gupta is professor and head in AIMS Delhi. Dr. Ramesh Vishanti is a director at ICGEB Delhi, and Professor Rana Pratap is an SLS JNU New Delhi. Now we request Dr. Rana Pratap to provide welcome as a plant to Dr. Y.K. Gupta. We also request him to provide a plant as a welcome to Dr. Ramesh Shanti. Thank you, sir. And now we request ICGEB Director Dr. Ramesh K. Shanti to give a chair address to the conference.
Should I speak from there or here? Okay, yeah, it's fine. Good evening, everybody. I'm really happy. I'm really happy that this meeting, ICGEB has had this opportunity to host this conference on Ayurveda and how best to move this, to validate Ayurvedic principles, to do the compositional analysis in an effective way. I think it's really great that you have chosen ICGEB for this particular one-day meeting. Now, we all know that all over the world, at least half the world's population uses traditional medicines. They just, that is what they've been used to for centuries, and even today the access that they have is for the traditional medicines. So ICGEB is an organization that works primarily for the benefit of the developing world. So our job is to actually promote technologies, validate technologies that are beneficial for the developing world. So it is very appropriate, and it is for us, a real privilege for us to be able to contribute to this task of, validating traditional medicine, making traditional medicine more acceptable all over the world, not just in India. And I was referring, just mentioning to Professor Y.K. Gupta that I think last year, and many of you know it, is that the Indian government has actually offered to support the setting up of a center on traditional medicine in collaboration with World Health Organization in Jamnagar. So this is the importance that the government of India actually, gives to traditional medicines, and to take this traditional medicine all over the world. And correspondingly, there's also a signal from the World Health Organization that they take this particular technology, this particular, these systems of medicine, alternative traditional medicines, seriously. And I think the objective of this particular center is to validate traditional medicine, to bring scientific rigor. See, this is all through experience and wisdom, traditional wisdom, that we have come up with knowledge about which plant to use, what formulations to use, what kind of people need what kind of medicines. But all of these require some amount of scientific validation so that you can actually bridge the gap with allopathic medicine, and down the line bring traditional medicines to the common persons. And the traditional medicine itself although I mean we are all too small people to say that we validate traditional medicines, but in the current scenario, it is actually important also to bring this scientific rigor so that more and more people can actually take it with greater and greater confidence. So it's really wonderful that we are doing all of this, and India is the home of traditional medicine, I would say. And we have, through generations, we have built up from maybe thousands of years. I was reading a book by William Dalrymple, it's called The Golden Road, and it lists all of the things that ancient India did to promote knowledge across the whole world. And this knowledge spans from mathematics to astronomy to surgery to botany and medicines. So we rarely realize what we have actually contributed. So to me, Ayurveda is synonymous with traditional, with India, with our culture. And we can do whatever we can to bring this medicine to our people with greater confidence. And then from our people to the rest of the world, I think it's a great achievement, and it's an important activity that has been undertaken in this conference. And once again, I thank you all for joining in this and giving me this opportunity to be with you today. Thank you.
Thank you, Dr. Shanti. For this valedictory session, we had chairperson Dr. Ramesh Vishanti and co-chair Professor Rana Pratap. So I request you to start the session with the valedictory address and concluding remark. The valedictory session would be by Dr. Y.K. Gupta, and the concluding remark will be by Professor Rana Pratap. So I hand over the mic to you.
So may I request Professor Gupta kindly for the valedictory address.
नमस्कार, यहाँ पे बैठे हुए मेरे दोस्त डॉक्टर रमेश सोंती, whom I respect as a great scientist, Dr. Rana Pratap Singh, and शायद आप लोगों में से बहुत कम लोगों को पता होगा, हो सकता है सबको पता हो, ये भी हो सकता है कि आज एक बहुत ही इम्पॉर्टेंट डे है, ख़ास तौर से आप सबको एक खुशी शेयर करने के लिए। और डॉक्टर राणा कल से he will be joining as a vice chancellor of Ambedkar University. So आपको बहुत बहुत बधाई, and I am really fortunate to be here on such an auspicious occasion that he is not only my friend but I just came to know, and then I could not resist कि ऐसे में तो अगर उनसे मुलाकात भी हो। और गोपेश ने हमें फ़ोन किया और हेमंत ने फ़ोन किया, तो मैंने कहा अरे अगर ये पहले बताते तो हम सुबह से ही यहाँ क्या बैठ जाते। But I really congratulate you. I think this meeting which I am addressing is a very timely, very important. और अगर आप में से how many people of you have gone to Prayagraj? Very good. और वो प्रयागराज में जो लोग नहीं जा पाए वो ये भी एक प्रयागराज ही है, where we are meeting the two streams, I would say. One is the traditional, or we can say Ayurveda system, and then you are talking of a modern system of medicine and including molecular biology, and talking of a science, high medicine, and traditional medicine, which is also of a deep science. So I think ऐसा मौका वहाँ तो 144 ईयर्स में आया है, बट यहाँ पे हमें लगता है ये जो हमारा सिनर्जी का एफर्ट है, दिस शुड कंटिन्यू फॉर 144 ईयर्स फर्दर। ये इज समथिंग विच इज़ ग्रेट इनिशिएटिव and great effort and need of the day. When I was governing council member of Ayush for a couple of years long back, I always used to say कि ये जो हमारी जो देश में धरोहर है जिसे हम आयुर्वेदा या आयुष कहते हैं, इसको जितना इसको ड्यू क्रेडिट या मिलना चाहिए था वो शायद हमने वो मास्क हो गया, वो नहीं मिला। एंड गलती किसकी? गलती हमारी है, क्योंकि हम जो साइंटिस्ट हैं, जो एक दूसरे को अगर मिलते हैं तो फिर हम कहते हैं अच्छा तुम आयुर्वेद से हो, तो आयुर्वेद वाला कहता है अच्छा आप तो मॉडर्न मेडिसिन के हैं, न तो हमारी दोस्ती तुमसे न तुम्हारी दोस्ती हमसे। रास्ते अलग अलग, रास्ते अलग अलग नहीं है, रास्ता तो दोनों एक ही जगह जाता है। वो रास्ता जाता है वेलबींग ऑफ ह्यूमन बीइंग, एक टोटलिटी में ठीक करता है, और दूसरा मोर ऑफ ए सिम्टोमेटोलॉजी और बेस्ड ऑन द जीनोमिक अप्रोच, बेस्ड ऑन द मैकेनिस्टिक एस्पेक्ट, वो ठीक करता है। बट अल्टीमेटली बोथ आर वर्किंग टुगेदर। एंड हाउ कैन इट बी दैट दोनों जने अलग अलग चले? So this is what we say is the shake hand approach is the one of the most important thing. And I always believe that when you say the hundreds years of wisdom, maybe thousands years of wisdom which you collect from Ayurveda, which has matured, and particularly an Ayurvedic system of medicine, Ayurvedic system of treatment option, maybe Panchkriya, maybe anything else, which has matured by experience and documentation, and then it has been refined. Does it require any evidence? Does it require any evidence? And this has been often debated. And I always say, if you want to survive in a contemporary period, if you want to survive in a day when everything is science-based, you have to generate evidence. And need of the evidence is not necessarily that you go to subgenomic level and you go to the subcellular level, submicro level. It is, the evidence can be generated different types. The epidemiological evidence, now we are talking of a real world evidence. WHO has given the recognition for real world evidence. आप इकट्ठा करिए दादी बाबा के नुस्खे. Nobody can prevent you, but provide the statistical analysis of that, clean the data, and then you say with 10% certainty, with this much of confidence interval, and then we can say with these caveats, this drug or this system is effective. But if you say that this drug is or this system is effective in each and everybody sitting here, or if you say this medicine or this herb is effective in each and everybody and without having any side effects, the credibility or understanding of this will become weak. And that is what the evidence is. Evidence can be the real world evidence. Evidence can be the simple evidence of epidemiologically. Evidence can be radiological evidence. Evidence can be biochemical evidence. Evidence can be the statistical significance, or maybe it is not statistical significant. Evidence can be the genomic level. I just inaugurated one of the lab in AIIMS Jammu, which I chair, personal medicine and genomic, high genomic center, sequencing. And what I said, यार ये जो sequencing अभी हम कर रहे हैं हर center में, हर AIIMS में या referral AIIMS में, for what? Ultimately, we are looking for genomics and we are looking for sequencing, and based on the sequencing, we can say that whether this drug, particularly anti-cancer drugs, anti-epileptic drugs, anti-tubercular drugs, or drugs of narrow therapeutic window to begin with, whether these drugs will be effective better in this patient or will produce more side effect in this patient, and what should be the dose titration for a person, what should be the concomitant medication which should be given to reduce the side effect or increase the efficacy of this drug in a particular group. This can be better done, better predicted if you have a genome analysis. If you have understanding of genome analysis, ये genome analysis करना आसान है, पैसा लगता है अब, लेकिन करना आसान है. And this creates a lot of noise. So I always say big data means big noise. But the important thing is you convert this big noise into music. That means you generate some information. And if this information can be translated into your wisdom, that how to treat a patient, how to best get the best effect of this. This is what? This is personalized medicine. This is personalized medicine. Today it is not possible to have a personalized medicine कि हम आपको एनाल्जेसिक के क्रोसिन की डोज दें, एस्पिरिन की डोज दे दें, आपको 300 मिलीग्राम दें, आपको 355 मिलीग्राम दें, किसी को 260 मिलीग्राम एंटीबायोटिक दें, किसी को 200 मिलीग्राम है. ऐसा नहीं पॉसिबल है. And therefore, generalization of this largely has been done. So now, you have to have a personalization of the medicine in where the dose calculation is done based on these very specific parameters and the drugs are of therapeutic window. Now, if you just go, this existed in our Ayurveda long back. We had Vayu, Pitta, Kapha. What is this? And we have a Vayu Pitta, we have Kapha Pitta, we have Kapha Vayu, and also sub-classification of different type of Prakriti. Now, if this Prakriti can be characterized by way of how you are taking ahar, what is your Prakriti, what is your Pravritti, what is your aspas ka Vatavaran, that will determine, and what is your other habits, that will determine, and what is your psyche, that will determine your exactly Prakriti. And for that, इस प्रकृति को आइडेंटिफाई करने के लिए लोगों ने बहुत सारे टूल्स भी बना दिए, एआई टूल भी बना दिया, डिजिटल टूल भी बना दिया, कई सारे क्वेश्चनर भी बना दिए. इंडियन सिस्टम ऑफ मेडिसिन के इंस्टीट्यूट ने भी एक सिस्टम बनाया, डीएसटी ने भी फंडिंग करके भी एक सिस्टम बनाया. अल्टीमेटली फॉर व्हाट? अल्टीमेटली फॉर सब क्लासिफिकेशन ऑफ दिस प्रकृति. What is that prakriti? And then based on these prakriti, can you identify the most effective, most optimum treatment or therapy? And this is now, if you, those who are here, there was a project when I was in CSIR long back. There was a project which was Ayurgenomic project was there with CSIR. कि हम उनका genomic analysis करके and then correlate with their prakriti and then correlate with the drug effect or drug adverse effects. That is also a possibility. And that is what you have to validate it. तो अगर हम कहें कि हमारे पास molecular base आज है, use करना चाहिए हमें. And अगर हम उसको isolated हम एक कमरे में बैठ गए, दूसरे में कमरे में बैठ गया, तो I think that will be the underutilization of our resources, our science, and that is what the synergy between the two systems should work. And that is what I think is very important. I have seen, and of course I know Dr. Sonti is working such a high science. Dr. Rana has been working on organ and chips. Now, if you are having organ and chips, if you have the organoids, now it is also possible that हमको जरूरी थोड़ी है कि हमने हाई मॉलिक्यूल, स्मॉल मॉलिक्यूल एंटी कैंसर ड्रग को ही टेस्ट करना है. Majority, many of the drugs of anti-cancer drugs have come from plants. But we have extracted them, and we have found out the single molecule of that. Now that can also be done, studied in organ on chips. Future clinical trial will not be in human. Clinical trial will not be human. After 15 years or 20 years, these all clinical trials will be done on human on chips. हम तो खत्म हो जाएंगे ना, क्योंकि हम क्यों आदमियों को देना है? तो that is what the things are there. And that is not only that it is to do the molecular events only for the modern system of medicine. It can be done for this is also. We have also been talking of pharmacovigilance program. Adverse event, nothing is there which has an effect, will not have a side effect. जो ये कहते हैं आयुर्वेद के लोग, I am a strong believer in Ayurveda, कि ये आयुर्वेद की drug है, this will be 100% free of toxicity. I think such type of overstatements dilutes the science. We should know it is what you call as any drug, whether it is Ayurvedic or this, is a by-edged sword. और हम क्या करते हैं, हमको मालूम है कि ये इसका जो है, जो दूसरी तरफ का है, इतना मोथरा है, इतना चौड़ा है, that the risk associated with Ayurvedic medicines are much less as compared to the therapeutic benefit. But if you say it is no, then it is wrong. And therefore, there is a need also to have the constant vigil of what drug can produce side effects. And that's what we started a couple of years back, pharmacovigilance of Ayurvedic system of medicine. And this is, Jamnagar was the center. That was the excellence center. Couple of centers are there. So you can also know that this drug can produce side effect. Now if this can produce side effect, why it is producing side effect? And then how can you reduce the side effect? Whether you do the different putta kriyas, maybe you can do 10 putta, maybe you can do 100 putta, maybe sahasra putta, to reduce the toxicity or to increase the efficacy. And if you are creating bhasma, people have been talking that the bhasma is a nonsense. Bhasma is the science. And then if you create a bhasma, then what is the contemporary science is bhasma? Is it a nanoparticle, or it is a microparticle, or it is a particle of a different range which does not cause toxicity? And we published a paper some time back, which was a very important paper landmark, when people said this bhasma in India, Indian bhasma have created lot of renal toxicity, the Ayurvedic drugs. And we found that if you give these medicines, particularly bhasma, abrak bhasma, it did not cause toxicity. Of course, this was an animal experimentation, but this was eyes opener. Though concentration was present, that means you reach this such level that it isn't present, but perhaps it goes into the tissue, does not stay there and gets out. So you have to, there can be two ways. That if you say कि हमने भस्म खा दिया उसको renal toxicity होगी, but you have to see that renal toxicity उसकी हुई, was that because of the right quality of bhasma, or this was because of the poor process of creating bhasma? It may be inappropriate formulation, preparation, or inappropriate use. But if you say कि हम किसी को भी कुछ नहीं हो सकता, then also it is inappropriate dose. So many times, I think the going into the intricacies, science of this is very, very important before we accept and before we reject. And I always talk about, we have to have 3 baskets. If I say that this product has an anti-cancer effect or anti-epileptic effect, or if you say हमको तो वहाँ पे बाबा जी ने एक दिया था और वहाँ से 10 लोग ठीक हो गए, इसके खाने के बाद में उसके एपिलेप्सी ठीक हो गई, मिर्गी ठीक हो गई, बहुत सारे लोगों को, बड़े सारे लोग खाते हैं, वे मिर्गी समझते ना? आप जाओ यहाँ से मेरठ के पास आगे जाओ तो वहाँ पे एक मिर्गी बाबा बोलते हैं, and he is famous that people go for a long distance and make a queue, and then he gives some pudiya and people say that they get cured. But then we have to put in 3 baskets. Whether such claim, you examine that claim. Whether this claim is garbage, rubbish, foolish, people are being or maybe gullible people are being used that. Put this, examine this and put this in the garbage box. The second thing is, if there is some evidence or some truth, put this into the box for evidence generation. Evidence generation means epidemiological evidence generation, and then work on the science. And then if this is worth doing, accept it. Accept it with evidence. Now that is what we say, 3 boxes. And that is what I think when you say the sangam of these 2 systems, I think this system should be done on this basis. If I just give you a simple examples, अगर आप लोगों को डेंगू तो किसी को हुआ है? हुआ है कितने लोगों को हुआ है? कई लोगों को हुआ है। कभी पपीते के पत्ते का जूस पिया है? 2-3 लोगों ने पाया. And this was the myth created. This was a false advertisement created. कि पपीते का जूस है, उनके हमारे घर में पपीते के पत्ते ही सारे तोड़ के ले गए, पपीता ही नहीं बचा. और वो उस, but then it was so much hyped, but then we investigated that and then we found that this has nothing to do with the viral load reduction, this has nothing to do with the antipyretic effect, and therefore this must not be. So we investigated that. Because इतना सारा लोगों ने इसको खाया पिया, पत्ते खत्म कर दिए, तो इसको, this makes a case to investigate it and go into the high science into this, go into the antiviral property, go into the replication of virus, go into the antipyretic property, go into the fractionation, and then see it works or not. So far it was not found to be. Maybe it does not, we don't say that कि ये होता ही नहीं है, but we say we did not find. We did not find. And a lot of molecular biology was done on that. So this is what I am saying, if we interact with these things, it will be important. Lastly, I will also, one thing I will say. हमको कई बार हम कहते हैं कि यार ये है ना, इसका हमने ginger है, is very effective for anti-cancer vomiting. I published paper on the ginger for anti-emetic in cancer. अब लोगों ने कहा इसका एक्सट्रैक्ट निकालो, इसका इसमें मॉलिक्यूल निकालो. अरे भाई, there are two approaches. One which you call as a reductionist approach. You go on reducing and then make a single molecule, and by the time single molecule is, then it becomes toxic. There are several examples then. Or it may be in a very low concentration, most effective. So you have to see very critically whether and how much reduction is required. How much single molecule is deliverable. And that is where the wisdom should be. In some places where if you are doing anti-cancer drugs, I will definitely like to go for reduction approach, because I have seen that at least 20% of the, not now, about 5 years back, जितनी हमारी drugs थी anti-cancer की, 20% of the drugs came from the plant. But then it was converted into single molecule. But then there are several things which have a highly immunogenic, immunomodulator, immunostimulant, anti-arthritis, and maybe anti-aging, liver, it may be for Alzheimer's, neurodegenerative disorders, where the multiple drugs or a formulation which acts on the multiple receptor, multiple targets is the best effective. And therefore, I don't recommend that you go unnecessarily to single molecule identification. So you have to do a balanced approach, how much and to what extent you have to go into the deep and deep, because there is no end of that. So there is evidence, real world evidence. That's why WHO has also given recognition for WHO for real world evidence. So real world evidence, vis-a-vis real world evidence makes a case for investigation in detail. Epidemiological investigation or maybe molecular investigation. So real world evidence is now, each and everybody who is practicing here, I came to know that a lot of you are practicing also. And real world evidence, only thing which you have to do, evidence, real word, means what you are doing, document it. Document it with as much as possible details, as much as possible variation which you are seeing. And then give a, ये नहीं कि हमारे दादी बाबा ने और वहाँ पे एक आदमी ने ठीक कर दिया, एक बाबा ने वहाँ पे मेरठ के रास्ते पे, investigate it, bring that pudiya, investigate it here, and then come to some conclusion. So I am really happy that I am in middle of the top scientists of the country, Dr. Sonti and Dr. Rana, and of course Dr. Rupesh and all my colleagues here, Hemant. So I feel that I could not attend the deliberations, which must have been very exciting, but I am fortunate that I could address at least this. Thank you very much. all the organizers to giving me this opportunity, and congratulations to you all, and I wish all the best to those who have taken bath in Sangam in Prayagraj, and those who are here in this Sangam.
Thank you. I mean, you yourself is a sangam of both the traditional and modern medicine, in a way. But nevertheless, since you shared the good news, we'll request Professor Y.K. Gupta and Dr. Sonti to felicitate Professor Rana Pratap Singh on his appointment as Vice Chancellor, Gautam Buddha University, by presenting a bouquet. I'm sure Dhanvantri ji has provided a good luck and will continue to provide a good luck to him. So it has been a long day, and so now since I hand over the chair session to Dr. Sonti, so can I invite Professor.
Actually, it's a very happy day. Professor Rana is the Vice Chancellor of Gautam Buddha University. So glad you could find time to come again and be with all of us. So please, your concluding remarks.
Thank you so much, sir. I am always there for research. So I am very much thankful that we have Professor Sonti and Professor Gupta here. And actually, they have been our mentors for a long time. And if you know about the research, whatever is going on in terms of whether it is plant sciences or animal sciences. So we have one stalwart in plant sciences, other in the animal sciences. And Professor Gupta has showered his blessings on us at several occasions in terms of guiding the research or also directing the research and also giving us or suggesting us the new ideas what can be done. Sir, I am very much thankful for your blessings and the support. Related to our one-day conference today, I hope in the beginning when we started the inauguration, and if you compare yourself right now at the end of this day, I am sure that you will find yourself a bit more educated with respect to Ayurvedic concepts. I hope it is true, because for me it is always and every day is the learning. And still we are learning, although we are working from last several years. And we are fortunate that the scientists and the experts from CCRS and CARI and International Institute of Ayurveda, we have here to share their wisdom and knowledge with us so that we learn about our ancient knowledge of various alternative medicines. And Ayurveda is one of them, what we are discussing, but all of you know about the Ayush system, what we have in terms of having a holistic medicine system, whether it is Ayurveda, Unani, Siddha, homeopathic, or yoga. If we just look at the benefits of this Ayush system, Sir has already elaborately told about the drugs or the medicines, what we have and where we lack. And mostly Professor Gupta emphasized that although we have results, but sometimes we don't have evidence. So that is what you emphasized. और हिंदी में इसको बोलते हैं कि परिणाम तो है लेकिन प्रमाण की भी ज़रूरत पड़ती है, क्योंकि आज यदि आपको किसी को कन्विंस करना है, so you need both. Otherwise, even if you go on the medicine shop, buy any medicine,
we quickly look at in the Google and see what it does. So you find the information. So right now it is the age of information. And in this age of information, if we are working on something or developing something, unless we disseminate to the people or the public, it may not go too far. So now it is a big challenge before the scientists that whatever we are doing in the laboratory, it has to go to the people and the public as well. So this is a big challenge before us. So conferences or the meetings are, I would say, this is a celebration of research. If we are working in the lab whole year, whatever you have generated the data, get a time on one day just to discuss your data, and that is nothing but you are celebrating your research and trying to disseminate whatever you have done, how it could be beneficial or it can benefit to others. With respect to Ayurveda, sir, as we know the potential, the Ayurveda has very immense potential in terms of preventive medicine and maintenance of the diseases as well, along with the curative ones. When you talk about the prevention, whole day you have heard the lectures, and once you know the concept, as sir told about the personalized medicine. So personalized medicine or personalized wellness, it will come only when we talk about how to identify a person, how one person is different from another person. Unless you identify, you will not be able to prescribe a particular medicine which is going to suit that person, or the particular dose of that medicine which is going to be effective for that person to cure that disease, particular disease. As we know that all of us are different, and that is the concept of Ayurveda, how to identify or recognize each people's constitutions, bodily constitutions. And as sir said that in the beginning, in addition to the modern medicine concept, what we have out psychological factors, psychological and social factors. So in Ayurveda, as we have the holistic system of medicine, it is also including various aspects of our day-to-day life, which may not be considered when you go to a medical doctor who is practicing allopathic medicine. But if you go to an Ayurvedic doctor, they will be asking a range of questions, which will also cover many aspects other than only what you have in the form of disease. So this holistic system, that is what it is going to give you the real benefit, that is what we think. And that can happen only when we understand the system. So there are many concepts. Here, two concepts were discussed today, the Ama and Sooth. And now all of us are a little bit more educated with these two concepts. So understanding the other concepts of Ayurveda as well, I think it is going to be a very remarkable development, a remarkable achievements once we have. And this is also a process to convince those researchers who are just reading the textbook or the research papers being published on the modern medicine and modern medical concepts, to get sensitized and see whatever knowledge we had or we have, how to take it forward and get the most benefits out of it. As sir said that in healthcare, ultimate goal is the well-being of the people, well-being of the person. So in this respect, Ayurveda has immense potential, and we are thankful to all the scientists and the Ayush Ministry, the advisor and the Director General for their support. As we see that in last decade or so, there are several centers which have been initiated to work on a particular disease or a particular concept. And we are very much sure that in next few years, you will be seeing those results in the form of publication, and which is going to convince each and everyone, whether they are in India or abroad. So once we have at that level of scientific knowledge and inputs out of our research and the activities, whatever we are doing, ultimately we feel that our effort will go in right direction and we'll be able to contribute something. So this conference was also one small step in this direction. And I am very much thankful to Professor Rupesh Chaturvedi, Professor Neil Sarovar Bhavesh, Dr. Hemant, and Dr. Pallavi for working on these two concepts, this team for the Center of Excellence, and trying to contribute our little effort to see how it could be taken forward and it could be beneficial in terms of managing the diseases. So with this, I am thankful to all of you for spending your time here whole day and gracing this occasion. Thank you very much.
Thank you, Professor Rana. We also thank our chair and co-chair, Dr. Ramesh Shanti and Professor Rana Pratap. We request Professor Rana Pratap to present a token of thanks to Dr. Y.K. Gupta. We also present We also request Professor Rana Pratap to provide token of thanks to present the token of thanks to ICGEB Director Dr. Ramesh Shanti. Thank you very much for the wonderful valedictory session. Now I invite for the vote of thanks to by Dr. Hemant K. Kashyap.
Honorable dignitaries, esteemed guests, participants, and my dear colleagues. It is indeed a great privilege for us to deliver the vote of thanks on behalf of Center of Excellence on Ayurveda and Systems Medicine at Jawaharlal Nehru University in collaboration with Central Ayurveda Research Institute, Punjabi Bagh, and International Center for Genetic Engineering and Biotechnology, New Delhi. We extend our deepest gratitude We extend our deepest appreciation for Dr. Ramesh V. Sonti, Director, ICGEB, for graciously hosting this conference and providing the excellent platform for fostering collaboration between Ayurveda and modern scientific research. We also sincerely thank Mr. Biju Matthew, Administrative Officer, ICGEB, for his unwavering support in ensuring the seamless coordination of this event. And special thank to Mr. Ravindra Ravi, Manager of IT Services, along with his dedicated team, including Mr. Mayank Gupta and Mr. Anil, for providing flawless technical support throughout the conference. Our heartfelt gratitude goes to Mr. Naresh Dabral, Manager of Component, for his excellent organizational support, and Ms. Shweta Gupta and Mr. Sachin Mehta and the entire ICGB administration, housekeeping staff, and security team for their valuable assistance to make this event a resounding success. A special acknowledgement goes to Dr. Anu Singh for her outstanding effort in handling and arranging all aspects of the conference, ensuring everything ran smoothly and efficiently. We are equally thankful to Dr. Neil Sarovar Bhavesh, Group Leader, ICGEB, for his valuable scientific insights, as well as Professor Rana Pratap Singh, School of Life Sciences, and Professor Rupesh Chaturvedi, School of Biotechnology, JNU, for their academic contributions and continued support throughout this initiative. A special note of appreciation goes to all the distinguished speakers for their thought-provoking presentations, which have greatly enriched today's discussion on the Ayurvedic concepts of Am and Sot in context of modern medicine. We also extend our heartfelt thanks to organizing team, technical staff, volunteers, especially the students from my lab, Professor Rupesh's lab, and Professor Rana's lab, for their unwavering and very hard work done for the last 3-4 days. They have been really working hard to make this a success. As we wrap up this event, let us continue to foster collaborations between traditional knowledge and modern sciences, driving advancement in holistic, evidence-based healthcare. Thank you for all being part of this important milestone, and we wish you continue to success in your future endeavor. Thank you, and look forward to seeing you at dinner. Have a wonderful evening ahead. Thank you. There is a small announcement. Those participants who have registered for this event may collect the certificate now, and the participants for the workshop will collect the certificates after the workshop itself. Thank you.
So you all are invited for dinner outside the ICGEB lawn. That's it. So you can come. Rajkumar ji, Rajkumar ji.